Good morning, everyone. I, Dr. Jaswinder Kaur, extend everyone a very warm welcome to the day one of our much awaited virtual conference on neurosciences, NeuroEunoia 2020. It is my immense pleasure to have all of you amongst us and much more for the elite speakers, each one a stalwart of their respective fields, who agreed to grace this occasion. Talking about neuroscience, Professor V.S. Ramachandran said in one of his BBC Right lectures, science tells us we are merely beasts. But we don't feel like that. We feel like angels trapped inside the bodies of beasts, forever craving transcendence. Since the very inception of the human race, people have been wondering about what makes us humans, what makes us behave like humans. It was the observation of the normal behavior and the varied behavior that led philosophers to dwell upon the existence of the mind and then eventually drawing a very thin line between the mind and the brain. The field of neuroscience as it is today is a product of vast and meticulous observation, complemented by scientific advancements. It has come so far that as of today, several artificial intelligence applications are being developed to try to reach the elusive goal of these applications, functioning exactly like the human brain does. Stemming from the steeply growing importance of neuroscience happened the conception of NeuroEunoia 2020. As the subject of neuroscience was introduced for the first time in our college, it very quickly wrapped us in its wonderful and beautiful intricacies. The more we dwelled upon it and the more we saw its imprints in our daily lives, in the people we all interact with, and ultimately telling us who we are. The idea of this conference was conceived mainly to increase awareness about mental health and the ever expanding parameters of the mind and the brain. It is indeed a dream come true for all of us that we see NeuroEunoia having so many registrations and people expressing their unprecedented interest. We are, however, very sorry to inform you that Professor Ramachandran was unable to join us today because of some unforeseen medical reasons, and we pray for him to recover very quickly. And also, it is exceptionally kind of Dr. Sanjay Singh who accepted to grace the occasion once again as the chief guest of the event and to deliver the keynote address. Without further ado, I would take this opportunity to start this event with the hopes to learn and get bewitched in the light of the miracles that our brains can perform. I would like to invite Professor Rehan Khan Suri, the chairman governing body, to address the gathering. Please, sir.
Today's chief guest, Dr. Sanjay Singh, Dr. Pramila Kumar, principal of Madhuri College, my dear colleagues of Madhuri College, teachers and staff of the Madhuri College, delegates of this international conference, a very good morning to all of you. It is indeed my pleasure and privilege to be amongst this gathering, and I would like to congratulate entire team, organizing team of this international conference, NeuroEunoia 2020 in Neuroscience Affair. I must pray for first, as Jaswinder mentioned about our chief guest who could not join us today, Professor V.S. Ramachandran. May Almighty give him a fast recovery. I thank Dr. Sanjay Singh for accepting our request to be the chief guest today. It is indeed my pleasure as I attended his talk on the pre-conference event also. As we all know, this difficult time, we all are struggling, we all are facing these challenges during this pandemic, especially our students and teachers, because we are not able to interact each other personally, and we are just using the mode of online, which is really, sometimes even most of the time we are missing out lot many emotional touches. Physically, when we are meeting, we are really exchanging our energies also. When we are meeting online, those energies, exchange of the energies are missing. And you know, this the biggest issue during these days was mental health. And as the very relevant topic Gargi College has taken up, and this is one of its kind of this international conference which would really bring a good understanding about human brains and the issues and challenges of mental health. The human brain is wondrous organ. We use it every moment of our lives, and yet so much about it remains a mystery. How do our minds make choices? Why is it so easy to remember the lyrics to our favorite childhood song, but we forget important passwords? Do we really use only 10% of our brains? It would be fascinating to know about the wonders of neuroscience and the significant breakthrough that we can hope to see in the next 5 years, or even next in the coming time, future times. So, as this is a very relevant topic, and the kind of craft of this program, this event, the international conference is so good. It covers almost all of the areas related to neurosciences, and especially I would like to mention about the kind of this event of the poster competition during this virtual conference. It would be a great learning to all of us as academia as well as our students also. So I wish the deliberations would be there during last like next 2 days. That would become a knowledge to all of us. So I must congratulate again. I wish all success to this event. I again thank Dr. Sanjay Pratap Singh. I welcome you all for these 2 days. Thank you very much.
Thank you, sir, for your kind words of wisdom. I would now like to invite Dr. Pramila Kumar, a respected principal and the patron of NeuroEunoia 2020, to take over and say a few words. Ma'am.
Good morning, everyone. It gives me immense pleasure to welcome you all to NeuroEunoia 2020, the first of its kind of a virtual e-conference which Gargi College is proudly hosting. I would extend a warm welcome to all the dignitaries who have joined us here. Our chairman, Professor Rehan Khan Suri, the chief guest, Dr. Sanjay Pratap Singh, Professor Chair, Department of Neurology, Creighton University, United States. Guest of honor, Professor Pravat Mandal, Senior Professor, Division of Computational Neuroscience and Neuroimaging, NBRC, Gurugram. And Professor Roop Lal, Nasi Senior Scientist, Platinum Jubilee Fellow, the Energy and Resources Institute, Delhi. Right at the beginning, it is imperative to understand the importance of this conference. Mental health is very misunderstood term, rather a term less understood. We at Gargi College have always strived to create a conducive and a compassionate environment for everyone. In such distressing times of the COVID-19 pandemic, the importance of the well-being of mind has come to the forefront of each one of us. Although it is a neuroscience affair, the topics covered in these lectures by our eminent speakers are sure to affect all of us. Many times, a discussion about mental wellness makes us look into ourselves and tends to make us feel a bit uncomfortable. We aim to abolish this taboo so that we nurture the mindscape to look for reasons, finding solutions, and not to shy away from talking about it. I would like to send out a special note of gratitude to Dr. Sanjay Pratap Singh, who so kindly agreed to join us as our chief guest. I must mention that it was indeed our pleasure to be able to host you at the pre-conference super preview of NeuroEunoia 2020. The feeling is the same and even heartier now, although the request to him was sent at a very short notice, and we are indeed awed by his benevolence to have agreed to grace the occasion. We would also like to share this piece of information with the audience that Professor V.S. Ramachandran faced certain challenges on the health front, and he has very kindly allowed us to play one of his TED Talks. Thank you, sir, for that. Nonetheless, we have our eminent chief guest, Dr. Sanjay Pratap Singh, with us. I must also mention that the talk by Dr. Singh on the pre-conference super preview was very well received by our audience. It is our immense luck to have him here with us again today. Dr. Singh is the chairman and professor of the Department of Neurology at Creighton University School of Medicine, Omaha, USA. He is the director of Neurological Institute at CHI Health, Creighton University School of Medicine. He is also the president of Association of Indian Neurologists in America and a fellow of the American Academy of Neurology and the American Neurological Association. Dr. Singh received his medical degree from MLN Medical College in India, where he was first in order of merit. He completed a neurology residency at George Washington University Medical Center at Washington, D.C., where he was awarded the Best Resident Prize. He then did a 2-year fellowship in epilepsy and clinical neurophysiology at the Gilbert Glasser Fellow at Yale University School of Medicine. He has written a book on neurology and also several book chapters in other medical texts. He has authored several scientific research articles in prestigious medical journals. He is the recipient of several awards, like the A.B. Baker Teacher Recognition Award by the American Academy of Neurology, the UNMC Chancellor's Gold Hue Award, and Kudos Award of the Board of Regents of University of Nebraska, Lifetime Achievement Award at the Indo-Global Healthcare Summit, Hyderabad, and numerous other awards for his professional and teaching excellence. He has been invited to lecture at many national and international institutions and conferences. He has also given talks to the community in TEDx, science cafes, and Simply Science programs. He has also been featured in an expert in many TV programs in the USA. I hand over the screen to Dr. Singh without further ado. Welcome, Dr. Singh.
Thank you, Dr. Kumar. I'm going to try and share my slides. Hopefully that'll work. Okay. And let's see. Okay. So I hope everyone can see my slides. Is that true, Dr. Kaur?
Yes, sir, we can see it.
Okay. And you want me to start now, right?
Yes, sir, that would be perfect.
Okay. All right. All right. So I want to thank the organizers for inviting me back again. I guess I did not bore you to death the last time. So I'm happy to be back and to share more about the most fascinating subject, according to me, the human brain. And so today, I thought what we would do is really go on a different kind of journey and learn about some novel concepts that are beginning to explain and unravel the mysteries of the brain. My approach would be still somewhat similar to my last talk, in which, because I'm fundamentally a clinician first, I talk a lot about patients and their stories. And so I'm going to use some of that as we go forward in this. So as you heard the eminent speakers before me, it is said that we don't know a whole lot about the brain. But I often discuss the fact that how much do we actually know about things that are not strictly the brain, like our genes, the universe, and stuff like that. So just in a couple of slides, maybe go over how much do we actually know, right? So here's the human genome, right? The secret of life. We have uncovered it. We can do a whole genome testing. But if you really get into the nitty gritty, only 1.2% of the human genome encodes for proteins. And that's the part that we actually know about. We have a very interesting term for the rest of the human genome, or the human genes, the 98.8% of human genes. We call them junk DNA, right? So really, we know only 1.2% of our genes. And then this junk DNA, in the last couple of years, they've found that if you take even a tiny part of the junk DNA out, life is not sustainable. I would not refer to it as junk DNA, right? Just because we don't know much about the 98.8% of human genome, I would not use the term junk for it, right? Especially if it's life sustaining. So that's as much as we know about the human genome. Now let's look at the universe as a whole, right? So about 68% of the universe is dark energy, and about 27% is dark matter. So 95% of the universe is formed by dark matter and dark energy, right? We know very little about what is dark matter and what is dark energy. We have some idea what it is not, but we do not have a good idea about what it is. So clearly there are about 95% of the universe that we don't know much about. All we know is the 5%, which is all the planets, the comets, the stars, and the galaxies. Now, the third thing that I always discuss is the neutrinos. It's a subatomic particle. It is one of the most abundant particles in the universe, right? 100 trillion of these neutrinos pass through our bodies every second, right? And it is really incredibly difficult to detect it for us, right? So what I mean to say is brain is not the only part that we don't have a complete understanding of. We do not know about 98.8% of human genes. We call them junk. We don't know what makes up 95% of our universe. And we find it extremely difficult to find one of the most abundant particles, neutrinos, right? Even though trillions of those pass through our body every second. So clearly our knowledge of the physical world is very, very limited. And we have to acknowledge that. And brain is just one part of it. We have not done very well as a global society, because if you were to ask anyone, and if you put some thought into this, any of us could have told you that there are two basic immediate threats to humanity. I say immediate because I'm not talking about climate change, because that'll take some time. Two immediate threats to humanity, and we have known this because it has happened in the past, is a pandemic. And so we are going through one, and you can see how well we are doing as the world. And one of the prime reasons is we have not prepared for it. This should have been the number one thing we should have prepared for. The number two thing is it's called technically a major impact event, when a large astronomical object collides with us, the Earth, right? 66 million years ago, an asteroid came in and had an impact with our planet, and the dinosaurs became extinct. Almost all life form became extinct. So these are the two major threats that our species faces. And if I was to ask you, how much time do we spend on trying to prepare for something like this? Not much. How much of international collaboration is there in these two areas? Not much. So really, we have a long way to go in these areas of knowledge. All right, now let's come to the brain, the final frontier. So the first thing I wanted to discuss is a very fundamental part of our human existence, which is how do we understand more about this final frontier, the brain? So the one thing that has really allowed us to understand this is functional brain imaging. So you have functional MRIs, and then others, more recent developments like the magnetoencephalogram or the MEG scan, those kinds of things. This functional neuroimaging helps us. So how does the functional MRI work? When we put you here into the MR scanner and we ask you to perform a certain task, right? Like solve a mathematics problem, we can tell by functional MRI what areas of your brain become active when you're doing this particular math problem, right? So that's how the functional MRI really helps us. The MEG scan is how we look at the magnetic activity of your brain cells. And then this can also tell us, give us information through which we can map out the active regions of your brain when you're doing. So these functional brain imaging tools have really given us great information. And that's what has really helped us as we go forward on this journey. So let me first talk about vision, right? Let's discuss this very basic human attribute and need. So I'm going to talk about two patients here. The first one is a patient DB, and he has what we call blindsight, and I'll explain what that means. And the second one is CY, which is someone who has visual agnosia. Ignore these neurological terms, but I will try and explain to you why these things are so fascinating. So what is blindsight? So blindsight is when one half of the back of your brain is damaged, like it is here in a stroke, right? You lose vision in half of your vision fields. So say if you had this brain damage on the left back of your brain, you would not have any vision in the right half of your vision field. Now, understand this. The patient says he cannot see on the right side. We confirm he cannot see, as neurologists and neurosurgeons. But despite this, an experiment was done where a pointed object was kept there. Even a finger could have been kept there, right? And we were asked, and this patient was asked, to go and touch that object, right? So that is kept in the blind half of the vision field of this patient. The patient says he cannot see on that side. We confirm he cannot see because of the anatomy, right? But he goes and touches the object correctly 99% of the time, right? So let's understand this. The patient says he's blind on that side. We say he's blind on that side, but he is definitely not blind because he's touching that object 99% of the time correctly, right? That's what's called blindsight, right? And I'll explain how this works. So it's about how your brain is connected. So we'll take a look at that in a little bit. But let's go on to the second patient. The second patient, John, he sees he can see things very clearly, no problems there, but he cannot recognize them. So he cannot name his friend, right? He can tell you that he's the person he grew up with, he went to school with him, but after he suffered this injury to his brain, he can't identify him by name, right? And then some of the things he cannot even identify as to what those objects are. He can draw them out for you, but he cannot identify them. And as he put it very, very well, this is out of focus in his mind, but not in his eye, right? So kind of fascinating. So let's see, how does this happen? What is this visual concept? So we're going to look at a little bit of the structure of your brain. So this is your eye right here, and the eye sends the nerve fibers back into your brain, and they go to this area called the superior colliculus. So we'll just refer to it as SC. And then it also goes to the back of your brain, to your cortex, the cortical part of your brain, right? And this is where you see from. That's what we knew, right? But what has happened is these are two separate pathways. This pathway is present in what we refer to as other animals, right? Some of the other animals do not have this pathway, the new pathway in human beings, but they have this old pathway. So even smaller animals can see because they see food and danger in their vision fields. And so that does exist. But in humans, we have this old pathway, evolutionary pathway, and we have formed a new pathway that takes it to the cortex. What makes human beings different from most animals? It is this cortex right here. So this big structure, the upper part of your brain, which has developed the cortex, is what makes us different. So this new pathway really is what is responsible for this cortical vision. So let's see. So what is happening in this region? So in blindsight, what has happened is that, as I showed you, the cortex, the back of your brain, the cortex, the occipital cortex or the visual cortex is destroyed, right? Now, what is the cortex responsible for? Cortex is primarily responsible for conscious awareness. So what happens is this pathway is no longer working, the new pathway, because this is damaged, right? But the old pathway is still intact, the superior colliculus, right? This pathway that exists in other animals, smaller animals, is still present. So this is the pathway of vision, which suddenly becomes active because this one has become inactive, and therefore you develop blind sight, meaning you are not consciously aware of your ability to see in a particular part of your vision, but you can see because of this older pathway that still exists, right? You cannot see as well as you would have seen if your cortex was working, your visual cortex, but you are able to see. Now, this is somewhat fascinating. Why is it fascinating? Because now we are beginning to learn that there are conscious abilities in human beings, and there may be abilities that we have that we are just not conscious of because they do not have a cortical connection, right? So is vision the only thing, or there are other things that we may be unconsciously aware of or have an ability of that we are not aware of because that doesn't have the cortical connection anymore? So that is kind of fascinating. That is an area that has not yet been explored. But Blindsight has told us that there is a concept of conscious vision and unconscious vision, right? Both, there is vision, but there is conscious and unconscious vision. So the question is there conscious and unconscious other abilities that we have, where we are only aware of the conscious abilities and not aware of the unconscious abilities. So that's one. I see that people are raising their hands. Just like last time, we will have question time at the end of my talk, and I will try and take as many questions as possible with them. All right, so hopefully Blindsight has taught us this concept of conscious abilities and unconscious abilities that we may have, that every human being may have, which we are unaware of, these unconscious abilities. All right, now let's look at that other concept where he couldn't recognize. But what happens here is, this is your eye, the vision nerve fibers go back, you see the scene, right, the object, but then there is a connection that goes from this vision cortex to an area called fusiform gyrus. So essentially another part of your brain where you recognize. Now, if this connection from your vision cortex to your recognition cortex is damaged, you can see, but you cannot recognize. Right? So this is a very interesting concept where just disconnections between your vision cortex and your facial recognition cortex, well, if that connection is broken, you can see someone, but you cannot recognize them because that connection has been broken. So it teaches us that in the brain, you have these areas of connectivity, right? And if these connections are broken, you develop these deficits, which are somewhat difficult to explain. In the past, these people were not even taken seriously because people thought, oh, they're making this up. How can this happen? How can they see someone, they know about the person, but they cannot recognize the person? So clearly, this concept is an interesting one where this disconnection syndromes are teaching us a lot about how your brain actually works. Now I'm going to show you a few things which play tricks on you in terms of your visual ability. So it tells us the limitations of our visual ability, even though it's a fascinating and a fabulous ability that we have as humans. So if this is a classic picture, if you look at it, some will see a younger lady. You can see the eye, the nose, and then the face right here. And some will see an older lady where you see this is the nose, this is the eye, and then this is the mouth, right? So again, one picture, and depending upon your visual perception at the time, you'll see a young lady or an older woman. This is another very fascinating area. So what I'm going to ask all of you to do is focus on the red dot. You see these rings around the red dot, right? If you focus on the red dot, these rings will disappear, right? So let's do that. I'll give you a few seconds to do that. Focus on the red dot. Completely focus, and you'll see the rings will disappear, right? So your visual apparatus is somewhat limited because of how your visual system is built up. And you don't see through your eyes. The part that actually sees is your brain, right? So that's where the ability has to be there. Now, the next illusion is pretty common. This is obviously a stationary picture, but you see a circling motion because your eyes are, this has fooled your visual perceptual abilities. So you know that this is just a picture, but you're seeing the circular motion in this. And that's also, how your limitation of your visual apparatus. Now here, some of you probably see, white columns and then white dots, right? And if you were to make your eyes a little smaller and look at this, you will see the word bad eyes in this, right? So clearly, the way you see the world, the way you see the world depends very much on how you perceive the world visually. And our visual perception is somewhat limited. And it's also just in the Vibgyor spectrum. So ultraviolet and infrared spectrums could have a lot of things going on in the world, but we would be completely unaware of this because our visual apparatus is somewhat limited in what we can do. So that has to be borne in mind as we perceive the world. Now let's look at this visual phenomenon. So if I was to ask you to focus on the face of this woman, right? And I want you to just focus in on it, you would think your eyes are completely stationary, right? But they're not. If I put a high definition camera on your eyes while you're focusing on this picture, your eyes would be darting about like this, right? These are eye movements of which you're not conscious, but they're continuously happening, right? All right, so this was fascinating. When this was discovered, the concept was, all right, can we truly stabilize your eyes, right? Fix them literally on this picture. So they used a technique called retinal stabilization technique. The technique itself is not important, but when you put that, the eyes do not move and they focus on that picture. What was discovered was that after a few moments, everything goes black in front of your eyes. You cannot see, right? And when that retinal stabilization is removed, you start to see again because these darting eye movements start again that you're not conscious of. So what is this about? This taught us a very important thing about the human brain. This taught us that the human brain considers change in input as input. A fixed input is not considered any input at all. Right? So let's think about this a little bit. Because your eyes are darting about, you have change in visual input, and that is regarded as visual input by the brain. Once the visual input does not change, the brain thinks that there is no input at all, and so you stop seeing. Okay? Fascinating. But the question is this true only of vision, or is this true of other things too, like sound? So if there was some sound in your environment which never changed, right? Never changed, never became louder or softer, or the frequency never changed, right? We would be completely deaf to it. Or if there was something in our environment that never changed, despite the little darting of our eyes, then we would be blind to it, right? So this is telling us that the brain really considers change in input as input. Otherwise, it doesn't consider any input at all. Why would this? What is the advantage in evolution with this? So let me give you an example. So some of you might have experienced, unfortunately, dizziness, right? So you get dizzy. So the room starts spinning around you, right? A common cause of that is your inner ear is being damaged, right? And that's why it happens. So what happens normally? Your inner ear from both sides gives your brain, say, 100 points of input, right? When one input is destroyed, right? Then what happens? Your brain interprets that as 0 from this side, 100 from this side. That means you're spinning, right? Because it should be 100 and 100. Now, over time, the brain then determines that this difference in input is not changing. 0 from here, 100 from here, right? So it interprets no change as no input. That is so important. That's how we recover from dizziness, right? From a damaged internal ear. So this is very advantageous to us, because if one part of a sensory organ is damaged, we don't have permanent deficits from it. We can recover from those. That's how we recover from these deficits. So we learned that through this visual experiment. Now let me tell you a story of a patient. So this was a patient in Europe. She had an injury to her brain. Both sides of the back of her brain got damaged. And this area that we call MT got damaged. Now remember, at that time, we did not know what MT did, this area. So both sides, this vision area, MT, was damaged. The patient suffered this brain damage, recovered, and then came to her physician. The physician asked her, "How are you doing? Any problems?" And she said, "Yes, I cannot cross the street anymore." And so the physicians, if they're nothing, they're at least empathetic, right? So in his empathy, he said, yeah, you've suffered such major brain damage. So, it can take away your confidence. And that's why you are probably having difficulty crossing the street. And the patient said, no, that's not what it is. And the physician said, then what is the problem? And the patient said, well, the problem is that I cannot see motion. And the doctor said, what do you mean? She said, when I go out on the street, I see the car here, then the car here, and then the car here. I don't see it moving. And he said, that's kind of interesting. Any other problems? And she said, yeah, I can't pour water from a jug into a glass. And he said, why not? She said, because I see the glass this full and then it's just overflowing. I don't see it filling up. Now that physician thought, she has had some serious psychological issues, because these kinds of problems have never been mentioned before. Well, but he was good enough to actually consult a neurologist, and that neurologist took it upon himself to investigate the matter further. And what he found was that this MT area was damaged on both sides. And what we later learned was this MT area is what detects motion. So if you destroy this area in your brain on both sides, you will never see motion again. So you see things like a car at this point, then at this point, then at that point. You never see the car moving, right? So clearly a bizarre sort of presentation, but it taught us that there is a brain area that is actually responsible for perceiving motion. Similarly, the V4 area right here is a color vision. And if that gets destroyed bilaterally, you may literally have black and white vision, and the color vision may go away. So as we are learning more about the brain, we are discovering that there are specific areas that are responsible for specific functions, and then there are networks that are responsible for functions too. But it is still an open sort of work. The work is still in progress as we are doing this. Now, before I get done with talking to you or giving you a little whirlwind tour of vision, here's the last part that I wanted to mention. So this is an animal model where this is the eye, this is the brain, right? The blue is the vision fibers, which go to the vision cortex of the brain. And the red are the hearing fibers, which go to the hearing cortex or the auditory cortex of the brain, of the animal. This is how the brain develops in the animal, right? Now, what they did was that these group of scientists, they eliminated the hearing input. So the hearing red fibers were taken out, right? And then they found in development that the vision fibers not only went to the vision areas and vision cortex, they actually went to the hearing area and to the hearing cortex, and the hearing cortex started to behave as the vision cortex. So is this a big find? Yes, it is a major find, because this is telling us that the hearing part of your brain can start functioning as the vision part of your brain. This violates the gospel truth, right? That if one area of your brain is damaged, right, like the vision area, then you lose vision forever. this is bringing to us the possibility that we might be able to convert another brain area to function as vision cortex, right? So it was a very major find, and it still needs to be developed further to see what the true impact of this finding is. Okay. Now, before I go on to the next topic, which is sleep, I wanted to give you a brief idea about this, about a small microbe called Toxoplasma, right? How it takes over your brain. So here's a very interesting interlude. So the Toxoplasma, I will just call it Toxo, essentially it reproduces in the cat gut, in the gut of a cat, right? But it's a parasite that infects rodents or mice, right? Now, mice are scared of cats, and so they will not go near cats, so they cannot be eaten. Therefore, the parasite cannot get to the cat gut where it needs to reproduce, right? So how do we solve this issue? How does this parasite, Toxo, do this, right? Also, remember, 1/3 of the people around the world, could have some kind of infection like this. So what happens is that the Toxo, this parasite, gets to the brain of this rodent, the mice, and it actually takes away the fear of the cat by its actions on the brain of this mice. And so these mice that are infected are not scared of the cat, and therefore they do not stay away from cats, and therefore they get eaten by cats, right? So this parasite has taken over the brain of this mouse and has fulfilled its need for the mouse to be eaten by the cat. How does it do that? How does it take over the brain? It actually produces the neurotransmitter of reward, right? Dopamine. Dopamine is the same chemical, the reward chemical, that is connected with addictions with cocaine and other psychoactive drugs. So it changes also structurally, it changes the brain of the mouse, some dendritic retractions in the amygdala, which is the fear center, right, of the brain. So it alters that. So it alters the chemicals, it alters the structure. And the result of all this is that it changes the brain and the mind of the mice, and the mice then end up going to near the cat, getting eaten, and the parasite can then develop further. What has been really interesting here is that there is schizophrenia in humans. Has been seen that in people infected with this parasite have a higher incidence of schizophrenia than in those that do not. So we are beginning to learn about human disorders in a way that we have really never looked at them before, because of our new understanding of how parasites and other microbes might be taking over our brains. Now let's study a very important topic, the sleep and dreams. So the first question is, why do we sleep? Well, till a few years ago, we really had no idea why we slept. Why do we dream? We still have no idea why you dream, right? There must be some evolutionary advantage in dreams, but we have no idea why you dream. And yes, people will go to the Freudian explanation of dreams and stuff, but there's really, in hard neuroscience, there's not much evidence for that. Even though I must admit that Freud was a bona fide neurologist who just happened to develop the field of psychiatry. Now, in the last few years, and by few, I mean within the last between 2 to 6 years, we have really learned a little bit more about what happens during sleep. Why do we need to sleep, right? So let's see. We have discovered this whole new system within the brain, which we call the glymphatic system. What is the glymphatic system? The glymphatic system is that these are channels that go all throughout your brain. And they take CSF, right, the fluid around the brain, and that fluid then goes through these channels and literally washes up your brain. So washes the brain of the toxins, right? And this happens primarily during sleep. How do we know that? If you look at this, right, this is the animal when the animal was awake. This is the animal when the animal was asleep, right? And there's almost a 60% difference in these channels, in the size of these channels between wakefulness and sleep. So clearly a greater than 60% increase in that space. Better CSF flow, better brain washing of the brain. So this is literal brainwashing. This is not being done in the other way. This is literal brainwashing. So let's see if that is of some significance, right? So for the audience, let me explain. Amyloid is the abnormal protein that collects in your brain cells when people get dementia, Alzheimer's dementia. And so this abnormal protein, amyloid, when it collects in your brain cells, it can actually destroy brain cells. Brain cells will die off, and then the person will develop deficits like you do in dementia, right? So this amyloid protein, abnormal amyloid protein, is really important, right? So they injected amyloid protein in the brains of sleeping mice, right? And in the awake mice, and found that during sleep, CSF cleared this dirt amyloid twice as quickly like a dishwasher, right? So this is profound now that we understand that during sleep, what is happening is that the fluid around your brain goes through your brain and washes out all the toxins, right? This is something we had not known just till a few years ago. And we have also learned just in the last year that not all sleep is equal. So you have the light sleep, the deep sleep, and then the dream phase of sleep, right? The REM sleep. It is only the slow wave sleep, the stage 3 sleep, right? Non-REM stage 3 sleep, right? That particular part is during that slow wave sleep is when this washing happens most efficiently, right? So it is important to get sleep, and it is important to get the deepest part of sleep for this brainwashing to happen and the chemicals to be taken out. Now, it was thought, if you look at the neurological literature, you will find a lot of publications saying, well, in stroke, your sleep is disturbed. In dementia, your sleep is disturbed, right? Because you have stroke, because you have dementia, your sleep is disturbed. Now we are learning that it's not that stroke and dementia cause disturbed sleep. It's actually disturbed sleep that may lead to dementia and stroke, right? So now we are learning that if you do not get enough sleep and the right kind of sleep, you would be putting your brain at risk because these toxins are not being washed off as they should be. So big information that we have now derived from this. Second function in sleep that has been discovered, as you see here in 2017 science publications, what they found was that there is synaptic scaling. So what are synapses? So your brain cells, they connect to one another by synapses. So that's a connection between brain cells, right? Memory is also basically a synapses. Synapse formation, connection between two brain cells is memory. So this connection is altered during sleep. Altered how? Altered like this. So some connections are broken off, some are made smaller, some the connection is no longer as strong as it should be. So this is what is happening, right? And this is what is being referred to as smart forgetting. So what is happening is things that your brain considers not important. During the day, you form a lot of synapses. The synapses the brain thinks are not important. During the night, those synapses can be broken off or made less connected, right? So this is another thing that is happening during sleep, is that you have what's called smart forgetting. So some of these connections are being broken off. Now, what about this fascinating part of sleep, which is called REM sleep, rapid eye movement sleep? Essentially, what's happening is that your eyes are darting about while you're sleeping, right? During this phase. This is also the dream phase of your sleep. So each and every one of you had 3 to 4 vivid dreams last night, right? How many of you remember all 3 or 4 of your dreams? I would probably say almost none will remember all 3 to 4 of your dreams. The other fascinating part that is happening is that during this phase, of REM sleep during dreaming, your body is completely paralyzed. Almost every muscle in your body is paralyzed. If we put a needle connected to an electrical apparatus, it will show us a flat line. Right? And so you were paralyzed 4 times last night, and now you have come back. So every time when the dream phase gets over, the eyes don't stop darting about. You go out of this REM phase of sleep, and your muscle strength comes back, right? But every night you're paralyzed 4 times, completely paralyzed 4 times every night. That's kind of fascinating and interesting. So what do we know about dreams? We do not know why we dream, but what do we dream about, right? So it is associated with this darting eye movements, right? Under your closed eyes, you can see that the eyes are darting, right? And so the idea was, how do people dream? Almost everyone dreams, right? And REM is when these dreams really occur, right? So if people are these visual, they said, what is these darting eye movements? What does this have to do about it? So they said, well, it's probably your dream scene, right? That's being looked at, and that's why the eyes are moving back and forth. It's kind of difficult because the eyes are darting like this. You don't really see anything when your eyes are darting like this. But they said, okay, let's test this in blind people. Now, in blind people, if they become blind after the age of 7, they will still have visual dreams. If they are, say, blind from birth, they do not have visual dreams. They can have auditory dreams and other kinds of dreams, but they do not dream visual dreams, right? But in these blind from birth, these eyes are still darting about under the eyelids. So clearly, it's not scanning a scene from your dream. It is something more. These eye movements are the key to understanding this, right? So let's see if we can understand this. Some of you may have heard of this term, since we have a mixed audience, anosognosia. So this is a typical neuroscience concept where we give very complicated names to fascinating things. So what is this? Let me first explain this a little bit, since it's a mixed audience. So what happens here is, say the right part of your brain is damaged, stroke, head injury, it just gets damaged, right? And the person is paralyzed on the left side because your right brain controls your left side. So your left side's paralyzed, right? In some of these patients, when we go and talk to them and we say, are you paralyzed on your left side? They say, no, I'm not paralyzed. Are you sure you're not paralyzed? No. Can you clap? They're clapping with one hand, right? Can you hear yourself clapping? They say, yeah, I can hear myself clapping, right? So there's complete denial of deficit. That's called anosognosia. And this was happening really well, and we were moving along because we said that's what happens. Then what happened was that in what we in the United States call hierarchical medical schools, which is almost everywhere other than the United States. In a hierarchical medical school, there was a professor rounding on patients, and this patient had anosognosia. So he was paralyzed on his left side, but he was denying his paralysis. They even demonstrated the one hand clapping and him claiming to hear himself clap. And so that was there. Now, this is not a psychological phenomenon, because this is a biological phenomenon that happens in every race and continent if you have this type of brain damage, right? So it's just there. Now, that patient also complained of dizziness. And one of the tests that they do in some countries for dizziness is they squirt ice cold water into their ear canals. When you squirt ice cold water into the ear canals, what happens is your eyes go into these jerky movements, right? We call that nystagmus, but your eyes go into jerky movements. So it's not a very pleasant test to do in a live person. In the United States, we actually do in comatose individuals, but it's not a pleasant test. So in this hierarchical medical school, the professor announced it, left the room. The senior residents announced it, left the room. The poor third-year medical student realized he didn't have anyone to announce it to. And so that meant he was going to do the test. So the nurses helped him out. He gets ice cold water, squirts it into the ear canal. You get these jerky eye movements. And he asks the patient, how are you doing? Because he wants to know, are you dizzy like you normally are, right? When you have dizziness. So he asked them, how are you doing? And the patient said, what do you mean, how am I doing? I'm paralyzed on my left side. Wow, he said, he was just denying it, and now he says he's paralyzed on his left side. So he's very excited, as any third-year medical student should be, and he runs out to get the whole team. So he goes there and he says, his anosognosia is gone. He's not denying his deficit anymore. Now, let me tell you, we do know that anosognosia does go away. In hours to days to weeks, it does resolve, right? If you ask me, why does it resolve? Well, we had no clue why it came, so we obviously have no clue why it goes away, right? But we know it does go away. But just to humor the medical student, the professor took the whole team back to this patient room, right? And said to the patient, oh, so you're aware that your left side is paralyzed? And the patient said, I don't know what you're talking about. My left side's not paralyzed. He said, can you clap? And he's clapping with one hand. So the poor medical student didn't look very smart. But the nurses there insisted, no, this patient did say that he was paralyzed. So the professor said, okay, do that, what we call the caloric test, again. So he gets ice cold water, squirts it in its ear canal, the jerky eye movements come on, and they ask the patient, how are you doing? He says, what do you mean, how am I doing? I'm paralyzed on my left side. Wow. How long? He says, 1.5 days. You guys have been treating me. Okay. Then the jerky eye movements only stay for a little while and then they go away. So the jerky eye movements are gone. They ask the patient again, how are you? I'm fine. Are you paralyzed? No. Can you clap? He's clapping with one hand again. So what is happening? As long as the jerky eye movements are there, it is taking you into a different plane of human consciousness, right? Where the patient is aware of his deficit and knows that this team has been treating him. As soon as the jerky eye movements go away, you come right out of that plane of awareness and you are unaware of deficit again. Fascinating. Where else do we get these jerky eye movements? REM sleep. What do we do during REM sleep? Dream, right? And as soon as the jerky eye movements go away, you do not remember your dreams, right? So jerky eye movements have the ability in some way to really take you to different planes of human awareness. And so that is what we are looking at. In fact, there's a new treatment for PTSD, post-traumatic stress disorder, that is being looked at with eye movements. So eye movements, in some way that we do not fully understand yet, and is a subject of great research, they somehow can take you into different planes of human awareness. It's also telling us that we may think we are not aware of things, but we are at some plane in our consciousness. Dreams are universal. People who say, I don't dream, if you wake them up during REM sleep, you can figure them out. They will always remember their dreams. There are some dreams that can happen during non-REM too, right? Why do we forget our dreams? There are some neurochemical and anatomical explanations. I'm not going to go into the details of those for you. And then one of the things that has been looked at is that during your dream phase of sleep, your memory is becoming consolidated, right? Major publications on this. What is happening? Whatever you did during the day, that is the same thing you are actually seeing, or related things you're seeing during your dreams. So the idea is the things that you found important, you are actually remembering them and consolidating them during REM sleep. The only problem is that your Prozac, the antidepressants that you take, Zolofts and Prozacs that you take, those actually cause a marked decrease in the REM phase or the dream phase of your sleep, but they do not decrease, they have not shown any real decrease in memory or learning during that period of time. So it's still a subject of research, but it's an important one, because we really do need to understand why we dream. And so at our center, we have a major study that's ongoing on dreams and their neurological purpose and what's going on there. This is big brain damage on both sides, in the back of your brains and in the depths of your brain. And in that, you do have REM, jerky eye movements, but there are no dreams. So there is a particular disorder in which you can lack this. Now, there is this disorder called REM behavior disorder. So what is this? It's a very fascinating disorder. So as I told you, if you remember, your body is completely paralyzed when you're dreaming, right? Now imagine if your body is not paralyzed when it's dreaming. What will it do? It will act out the dreams. So what happens in this? This person who has REM behavior disorder, he's not paralyzed while he's dreaming. So he acts out his dreams. So he may punch through walls, or he may hit his partner who's sleeping with him, right? So this is a very interesting disorder. It's mostly in men. It's mostly in older men. It can be treated with a common medication, but recent data has shown that it's probably an early sign of Parkinson's disease. And so these patients can develop that later on. So interesting bits of information that we must have, but it's a very distressing disorder. You know, I have some patients with this, and they're somewhat high functioning executives, but they can't stay in hotels because they're people who live in rooms around them can actually see them doing these kinds of things, acting out their dreams. Okay. I just shown you some of, for the neuroscience people in the audience, that the SLD is the key area for paralysis during dreams. It's a separate pathway other than your regular motor pathway on how it influences your motor brain cells and causes paralysis. So that's an important thing for people to know. Now let's move on to the evolution of the human brain. What makes us different from other animals, right? The main thing is we have a much larger brain, right? Particularly the cortex, this part of your brain, which is the cortex, but particularly the front of the cortex, the frontal lobe is much larger, right? So we have a much larger brain. How did we get here? So what I'm trying to show in this part is the fascinating fact that just a couple of genes have led to the evolution of our brains. Right? So the first is this gene cluster called NOSH2NL. This gene cluster is found only in human beings, right? And in Homo sapiens and Neanderthals. And this is what is responsible for the bigger brain, right? The question is really, and how does it do that? So how does our brain form? Our brain forms stem cells, right? Stem cells, they give rise to brain cells, right? So it does not allow stem cells to differentiate into specialized cells of the brain. So what then happens? The stem cells just keep becoming, they keep replicating and becoming more and more. And because it delays their specialization, you get a much larger amount of these stem cells. And then when they differentiate, you get a bigger brain. So that's as much as we know today. Now, as you can see, studies on this are just a year or two old. So I'm just trying to give you real recent information. And this gene, we estimate, came into human beings about 3 to 4 million years ago. And that is what has given us this larger brain. So if you look at some of the other primates, right, and you look at their brain size and you look at our brain size, right, and that has happened because of this one gene cluster, which does not allow the stem cells to specialize quickly, and therefore it allows the stem cells to multiply and grow and then specialize and cause a bigger brain. So as I've told you, look at the dates here. September 2020, this is a very active area of research, but literally one gene cluster is able to give us a larger brain. So that's kind of fascinating. Similarly here as to how it started and how it actually formed. So as physicians, we're also interested, does it cause problems? Yes. If you have too much of this, then you can develop macrocephaly, larger heads, babies born with large heads, or too little of this, you'll get microcephaly, which is babies with smaller heads. What we have learned is that in evolution, what caused us to go from a chimpanzee to a human being, a genetic mutation, that genetic mutation remains unstable for millions of years. And so if it can flip and make us humans, if it flips back, it can take us to the older primates, right? So you can have a normal human brain, but if that mutation, if that gene flips back, you form this kind of smaller brains, right? So it is teaching us a lot about how these brains kind of develop, how you can go from here to here and then from here to here. So, but those regions which have created the human brain remain unstable for millions of years, so you can switch right back. And so that can be an issue as we go through this. So one gene cluster for the large brain that we have, which differentiates us from other animals. The other is that we have language. As far as we know, there is no language in other animals.
Please shut down your mics. Is the humble request. Please do not.
Thank you. So humans have language, and as far as we know, the other animals do not have language. They do communicate, but they don't have language like you and I do. What has been discovered recently is that this is through one gene called FOXP2. One gene has given us this ability. And if you look at that FOXP2 gene, the difference between the human gene and, say, the chimpanzee is 2 amino acids. So 2 base has caused this difference, right? Very little difference, but it has given us the ability of language, which is a fundamental ability that we have. I'm not going to go into the details of how it does this. Essentially, this gene affects development of our brain and also development of our speaking apparatus, right? The mouth, the airways that we have. So clearly, it really does have that ability, right? And it's a very key component. There are some theories as to how it probably gives us. It helps us understand and form concepts which you need in language production. But the most fascinating part is this, that it only differs in 2 amino acids between us and non-human primates. And in mice, it differs by 3 amino acids between us and the mice. So it's not a major change. It is something that with today's technology, we can easily do and change this. And people have done. So what have they done? They have taken this FOXP2 gene, our language gene, right, and introduced the human FOXP2 gene in a mouse, right, to see what happens. Well, what happens is that this mouse becomes much smarter. It can go through a maze much more quickly than other mice can, right? It can find food much more quickly than other mice can. So introducing a human FOXP2 gene in a mouse makes it much, much smarter, which is telling us that it is probably responsible for language, and language is responsible for a lot of things we're able to do as human beings, right? So language is the end result, but there's a lot of other intellectual processes that go into doing this. So I did want to share with you one gene cluster, NOS2NL for larger human brain, right? And one FOXP2 gene, right, that is responsible for the whole language and to some extent the intellectual prowess that we have as human beings. Now we move on to the concept of brain function. So what areas of the brain have what functions? How do we determine that? A large part of that is because we put electrodes on the surface of the brain electrically, right, to see what function is in a human being. And then we know that this area is responsible for this function. Why do we do this? When we are treating patients with epilepsy, we can find out that the epilepsy is coming from this area of the brain. But before we cut that region of the brain, we need to make sure that that area of the brain is not responsible for any vital functions. So we stimulate these. And this stimulation over time has really led to us understanding a lot about the functions of the brain. A lot of what we know about different areas of the brain and their functions is because of these stimulation studies. So let me share just a few patient examples. So let's see. So the first one was this patient who the front of the brain was being stimulated. Now, remember this. How does this happen? You have the patient lying on the bed, right? You have the physician, the neurologist standing beside him. The electrodes are inside the head. The patient doesn't know which electrode is being stimulated at what current strength, right? The neurologist is controlling all of that. So they went around stimulating different areas. When they came to these red electrodes, right? Patient has no idea which electrode you are stimulating. But when the neurologist got to these red electrodes, neurologist and neurosurgeon, and they stimulated these red electrodes, right? The patients started to laugh, right? And so the physician said, why are you laughing? And he said, oh, you did something funny, right? And they said, what funny? So he'd always make up something that the physician had not done, but which he found funny. But whenever he stimulated these electrodes, the patient would laugh, right? And if he increased the current by which he stimulated this, they would laugh even louder, right? Now, it would be somewhat disconcerting for the patient to think that he was laughing for no reason, so he'd make up a reason, right? So we learned that there are areas of your brain which are responsible for laughter. We still don't know what is the fundamental use of laughter, but we do know that there are regions in your brain that do this. Even more fascinating. So in another patient, what was done was these are the electrodes placed on the side of the brain. And then whenever they came to these two yellow electrodes right here that I've pointed out, when they stimulated this electrode, the patient said, you know what? I'm having an out-of-body experience. So if this patient doesn't know which electrode they're stimulating, they've went around stimulating all of these, the patient did not say they have out-of-body. Every time they came to these two electrodes and stimulated it, the patient said, I'm having an out-of-body experience, right? Fascinating that there would be an area of the brain that may, at least in these patients, do this. When do we have out-of-body experiences? So if you were to study the near-death experiences, people say, you know, I have this out-of-body experience, right? So if people have near-death experiences and come back, they say, I have the out-of-body experiences. So I'm going to give you a very unglamorous explanation. This area is the border zone of two major blood vessels. One blood vessel supplies this area, one blood vessel supplies this area. So what happens? It's like this. It's like the sprinkler, right? If you decrease the water, the heart stops. What happens? The border zone is affected first. And if the border zone is affected first, dysfunction here can give you an out-of-body experience. So I don't know if that's what everything does, but it's a fascinating explanation based on hard science now. Now, the last one that I wanted to discuss, which is much more, even more fascinating, is the concept of desire. So there are areas of the brain that were stimulated, and the patient said, I have a desire. So right here in the front of the brain, where the patient said, I have a desire to move my hand. Okay. Desire was not something that we had neurologically been able to produce yet. And if you increase the current of the stimulation in the same area, the patient says, I moved my right hand. Now, this is on video, so you know, no, you did not move your right hand. But the patient will insist, I moved my right hand. And if you stimulate it with lower current, the patient says, I have a desire to move my hand. If you stimulate it with higher current, he says, I have moved my hand, right? So clearly eliciting desire from brain stimulation is somewhat fascinating because it's a very fundamental part of who we are as human beings. So some areas that we have sort of found out. Now let me move on to the concept of mirror neurons, right? So what is the concept of mirror neurons? I'm sure a number of you have heard of mirror neurons, but because we have a mixed audience, I'm going to take a very basic approach when we go through this. So what are mirror neurons and how were they discovered? Mirror neurons were discovered in an Italian lab where they were trying to see in the monkey brain, as to how motor activity happens, right? So what did they do? They put electrodes, like I showed you, on the brain of the monkey, and they were trying to see when he performs a motor activity, meaning like if he picks an object here and puts it here, how does this brain stimulation happen? What they found was that when the monkey picks up something and puts it here, the certain brain cells get activated in his brain. But more fascinating is that when the monkey sees someone else doing exactly the same thing, picking something up and putting it here, the same brain cells got activated in the monkey, again, as if he had done that action, right? So he's not doing it. He's just watching someone else do it, but the same brain cells got activated. And so these brain cells were referred to as mirror neurons because they were literally getting active when the animal's doing the action himself or herself, and they're also getting active when the animal is seeing someone else do the same action, right? So the question was, why do we need this? What is the purpose of this? What we learned was the purpose of this is probably, because we're still very early on, because there are conflicting evidences everywhere, but is probably because it tells us the intention of the action of someone else, meaning that you're picking up a cup to take a drink rather than picking up a cup to throw it at you, right? Because you'd have to take different actions based on the intention. This is how we understand the intention of the action of other people, right? And that's a very fundamental thing. This is also how you live the experience within your brain of something that you're not doing but seeing. Now, how is that important? That is the basis, probably, of why we enjoy seeing, I say football here, but let me say cricket matches. Why? Because you're literally living the experience of the batsman in your brain through mirror neurons. That is why you get so elated when the batsman does well and so disappointed when the batsman does badly, as if you have had that experience, right? Whereas you are actually seeing it from a distance. So the mirror neurons are fascinating. The other thing is, if you were trying to teach someone how to play the violin, right? Or how to play the sitar, how would you teach the person? You can give a theoretical lecture using PowerPoints, which is not going to have much of an impact, or it is much better to actually show the person how to play the sitar, right? Why is that? Because when you show the person how to play the sitar, the mirror neurons are getting activated in the student. So they know how to activate those neurons again and play it themselves. So it may have a profound role in human learning. So this has been a very major discovery. I must say there was a tremendous hype at the beginning, which I think was overblown. But now we are learning that it is still a very, very significant discovery, right? So I'm going to go over the mirror neurons are located in different parts of the brain, in different areas. We're still discovering them. One initial hype was that this is probably responsible for human interaction, because why do you need to understand the intention of another human being if you want to interact with them? And if you do not understand the intention of their action, then you do not interact with them properly. And what is a disorder where people do not interact properly, or kids that do not interact properly? It's autism. So what they found in the initial studies was that there was thinning of the mirror neurons in these autistic children, right? But then later on, when they've done further studies, it's not really a physical change. There may be more of a physiological, functional change that may be contributing to it. It's not the sole description, as you will see later, but it is probably an important one that may have some contribution. So in autism, that is certainly there. The other fascinating part that we have learned, and we don't know what to make of this, half your mirror neurons get activated when you see an action within your reach. So within your reach, if there is something, as half your mirror neurons will get active. If it is much more distant from your reach, the other half of mirror neurons will get active. So just pieces of the information that I'm sharing with you, because the research is still ongoing to see what this is actually meaning, right? And this is not just in monkeys, in human beings. The same kind of epilepsy electrode on the brain studies have been done, and mirror neurons have been found in the human brains too, in similar locations as we found in animals in these areas. One very fascinating study just a few months back that was done was that they found that when you experience pain, the part of your brain that experiences this pain is this purple area right here. I'm going to have to move this out. This purple area right here, which is called the anterior cingulate gyrus. So this anterior cingulate is where you perceive pain. If you see someone else experiencing pain, the same area lights up in your brain, right? So this ACC, the anterior cingulate cortex or gyrus, right? This is the area by which you perceive pain and by which you perceive others perceiving pain, right? So what is the concept here? The concept is also that in these studies, functional brain studies, right, they found that in people who were observing another person experience pain, this region was very active in people who are empathetic, right? Sensitive people who feel the pain of others. Whereas in people with psychopathological states who are not sympathetic or empathetic, this area does not light up as much, right? So the lighting up, the degree of lighting up of this area, activation of this area, is being correlated with empathy or no empathy, right? And mirror neurons have a real role to play here. So it's a very fascinating concept. It may not be the sole explanation of human society as was proposed initially, but it is a very critical area of the brain in many human behavioral attributes and abilities that we have. And so as we know more about mirror neurons, we will understand better human brain, because till the 1990s, we did not even know that this existed, and we found it by accident, as we do a lot of things in neuroscience. Let's see. Now, the next concept that I wanted to touch upon is this resting brain and time travel, right? So, in these times of pandemics, I can't even use the examples that I usually use. So I usually tell people that when you drove to work today, do you remember every person that passed you by, every car or scooter or rickshaw that passed you by? And the answer is usually no, I did not. And why not? Because when your brain is not doing something active in the present, there is a default circuit in your brain that becomes active, right? And that takes you either to reminiscing about something in the past or imagining something in the future. It makes you time travel, right? So if you were to remember, if you were driving someplace today or yesterday, and if you really pay attention, you'll realize that you were remembering something in your past or imagining something in the future. This is what this default circuit in your brain makes you do, right? So what is the default circuit in your brain? The default circuit is that when you're not doing something active in the present, this area, which we refer to as your default mode network, it actually is called DMN, right? This becomes active. So when you're not doing something active in the present, this becomes active and it makes you time travel, reminisce something in the past or imagine something in the future. It's basically mostly in the central part of your brain and in association cortices of your brain. So this is kind of interesting, okay? So this is a part of your brain that becomes active when you're doing nothing active in the present. Okay, that's interesting, but is that really important is the question. So brain at rest has a level of activity and resting, in reading. So at rest has a high level of, and during reading only 5% more activity. So what is happening at rest? This default mode network is active, and that is utilizing energy. And that is what is really making you time travel. So next time you're by yourself and not doing anything active, you're just sitting around, you'll realize that you're either reminiscing about your past or imagining the future, and that's your default mode network that's making you happen, right? So that's kind of fascinating. What is this default mode network important for? It is important for autobiographical memory, meaning your memory, envisioning the future, moral decision-making, social interpretations. So this is a very, very fundamental part. Essentially, what is happening is when you're not doing something active in the present, it actually makes you do these things. Moral decision-making, memory takes you in the future or in the back, makes you do social judgments. Those kinds of things happen with default mode network, right? Now, because we are physicians, once we learn about these things, we try and see what's going on. So in Alzheimer's, the brain areas that are destroyed or when the brain shrinks, some of these default network areas actually shrink. And when we look at our patients, a lot of them complain of this emptiness. They say, I just have this empty feeling. And now we realize part of that emptiness could be because they are not being able to time travel. They're not able to go into the future or reminisce the past. And so they have this sort of empty feeling. Earlier, as an empathetic doctor, we used to just say, oh yeah, we didn't understand why that was, but that was it. And now depression and schizophrenia are being correlated to parts of this network and how it's active. Autism, again, what they found was this this default mode network, this DMN, is hyperactive in autistic children. That means it's just letting you play around in your own brain rather than look outside and do something active in the present with other people. So that's another part that has become active. So default mode network literally is its function, to time travel, right? We also can see, we can predict an error. If we are monitoring your brain, we can predict an error about 30 seconds before it happens. Because what happens is, when you stop paying attention and your default mode network becomes active. If we see the DMN is active, we know a mistake is about to happen in your task, right? And so the DMN can do these kinds of things. And very fascinating is that what we have learned is that during meditation, this default mode network, this DMN, is silenced. So this is actually the activity in the comes down during meditation, right? Kind of interesting that happens. But if you look at some of our Eastern cultures, what do we refer to this, to the mind as? They refer to this as a monkey mind, and you have to calm it down. And one way of calming it down is through meditation. And now we are learning that DMN, the default mode network, which makes you time travel, either go into the past or in the future, that activity is being brought down. And if you look at some of the Eastern texts, they actually say that how do you calm the monkey mind? They say live in the present, do not live in the past, or do not live in the future, right? And with the DMN, that makes a lot of sense. So it's an interesting concept. This is a lot of DMN associated with meditation. So there's a lot of evidence that this is something that does happen. And DMN activity is being now used as to determine as a biomarker and also to determine treatment response in psychiatric disorders. Also, how good the meditation is, because modulation of DMN can be looked at, right? So those are some of the things that are being looked at. But it is somewhat fascinating that this is happening. Now, as we've talked about Alzheimer's dementia in the past, I'm going to introduce this concept to you and then probably move on to something else. In Alzheimer's, if we do not treat Alzheimer's dementia, it will render almost every country on this planet bankrupt. And this will happen within the next 50 years, right? As the population ages, they get Alzheimer's and dementia, they have to be cared for. And so you have to create nursing homes and other facilities for these older people. And so it's going to render, at least the United States, it's predicted if we don't find a cure, the next 50 years, it will render the United States bankrupt. And so it's a similar thing with everyone else. So very recently, they found this very interesting type of concept for Alzheimer's treatment. What was found was that there are these gamma waves. So what are these gamma waves? Since we have a mixed audience, let me explain. When you have brain waves occurring at a frequency of 30 to 100 hertz, right? That's gamma waves. So what was done was they found that in animal models of dementia, for Alzheimer's dementia, there was decrease in gamma waves, right? And they had these amyloid abnormal proteins, plaques in your brain, and that destroyed the cells and led to dementia. So what they did was they said, let's see how we can increase the gamma waves. So they did this 40 hertz LED light, was flickering at 40 hertz, right? Not a very comfortable thing to do, but they did that. And that trained your brain waves and increased the gamma waves, because the 40 hertz is in the gamma wave frequency. And they found that over time, it decreased amyloid plaques. It's published in Nature, one of the most acclaimed scientific journals in the world. So that was kind of fascinating, that gamma wave increase can clean up this bad protein out of your brain and treat dementia, which is kind of fascinating. But why does this happen? So what was found was this gamma waves, they stimulate a kind of brain cell called microglia, which then comes and eats up and cleans up these amyloid from your brain. So gamma waves will clean up the amyloid, the abnormal protein that causes dementia. Interesting. But we can't be doing this flickering lights in everyone's eyes. It's a very uncomfortable thing, and you need to do it for a period of time. The basic thing is we need to increase gamma waves. How do we increase gamma waves in your brain? And so when we looked at this, the one thing that does increase gamma waves that has been studied, and these are all studies from outside of India, you have the Vipassana meditation. Different types of meditation and pranayams will increase gamma waves in the brain. And therefore, that has not been shown that this will clean up the amyloid from the brains in each people developing dementia. But there is a certain value to this, because gamma waves, we have seen in studies that it does clean up the thing. And if it can be done with these meditations, it's an important concept. So this has led to a lot of researchers in the United States to look at meditation, because now we've got default mode network, we've got this activation of clearance of amyloid from dementia brains. So people are very interested in this concept. So let's look at it from a purely neurological perspective. So in meditation, we have learned that it preserves gray matter, right? So essentially, the gray matter volume is preserved better in those who've been meditating for 20 years or more than in those who have not been. This was a study out of California. And then also more peacefulness, right? And happiness with this, because it is actually decreasing activity in the default mode network. So it doesn't allow the monkey brain to go everywhere. And therefore, it's probably contributing to that. So that's another interesting study. All of these have shown cortical and gray matter thickness being preserved better with different kinds of meditations that have been done. So really very interesting kinds of studies that have been done. They've also found that the antidepressant, this is a Hopkins study, which showed that the antidepressant effect of meditation was the same as antidepressant medication, right? And it may lead to volume changes in key areas of the brain. So they found that in hippocampal regions, the volume went up of the gray matter and decreased in the more emotion centers like the amygdala. So which was kind of interesting that it can do that. And this was just by 8 weeks of meditation. So this study just had 8 weeks of meditation in it, and different regions of the brain changed the size. There was a significant change. It improves cognitive function. They did a study and looked at performance in tests, and there was a 16th percentile increase in that. And then it reduces anxiety, clearly because of the activity that we've talked about. I'm not going to go into details of the study. It helps with addiction. Meditation in school. So in San Francisco, they did this twice daily meditation program, and they found that the school performance of the students went much better in these. So clearly what we are learning is that with meditation, we have to understand that a little bit better. It's a state of consciousness. We have very little idea in reality as to what it is, and have to do a lot more work to understand this from a very hard neuroscience perspective. Because if it can have such beneficial effects, if it is done properly and if it can be studied properly, it can be replicated in a very important way. Some of you might have heard of this. This hit the lay press quite a bit. Science publication showing the Sanskrit effect. So essentially, people who've been chanting Sanskrit months for a period of time, their hippocampal gray matter volumes have been larger. And so that's sort of interesting that some kind of chanting could actually make an impact on your brain and the regions where this effect would be there. So it's kind of interesting. All right, I'll just take another 10 minutes and we'll answer questions. In a dish. So this is important. Literally growing a brain in a petri dish. How does that happen? So what happens is that they take some of your skin cells, right? And they treat those skin cells and turn them into pluripotent stem cells, meaning cells that can form your brains, right? So this process won a Nobel Prize. Your skin cells are turned into stem cells, and then the stem cells from your brain. And so they've used this process, skin cells, treated them to make them stem cells. Stem cells are differentiated to form your brain cells, and then it is kept there to grow those brain cells. So kind of fascinating that you're growing a brain, from skin cells. And this, when it's right now, I should say at least till about a year or year and a half ago, they were able to grow a brain in a petri dish that was the size of a 9-week-old fetus, right? So clearly a significant amount. That brain is capable of generating its own electrical activity. So you can measure electrical brain waves from that brain. So it's not only a structure, it also has function in it of a brain, right? And the idea is to see if we can use this to treat brain diseases, or at least understand brain diseases in a better manner, right? And already they're looking at autism, schizophrenia, epilepsy, using these what we call cerebral organoids, which are actually brains being grown in a petri dish. Now, that is more fascinating. Obviously, other organs are being grown in petri dishes for transplanting in people who need those organs, but brain obviously is the final frontier. And so the fascinating part is that they've been able to grow the brain to this extent, where spontaneous electrical activity is coming from brain cells already. Lastly, I'll tell you a little bit about what's new in medicine, particularly of the brain, right? So the first thing is this artificial intelligence that Dr. Kaur talked about too, right? AI or machine learning. So the first, so you have in US hospitals now what's called the artificial intelligence nurse. So it does the intake when the patients come in, it receives them, takes their history, what's wrong with them, and all of that. More fascinating is that the first artificial intelligence medical system that has been approved just a few months ago is for neuroscience. It's for stroke patients. So what does it do? So stroke, what happens in stroke? Your blood to a certain region of your brain gets blocked. That region of the brain then dies off, right? So, and if that region was responsible for, your movement of the right side, you're paralyzed on the right side. So it's a very critical thing. And every minute is a few million brain cells. So you really need to do this very, very quickly. And so earlier, what used to happen here in the United States is that you get a brain scan, and then you process that scan, then you get the radiologist to read it, then the emergency room gets that finding, examines the patient, then refers to the neurologist, and then that happens. This probably took somewhere between 3 hours or so, right? Can you imagine how many brain cells died during it? Now what we do is we have a new artificial intelligence system that is monitoring all our CT scans in our health system, which is about 14 hospitals. So this artificial intelligence system is monitoring these CT scanners. Wherever a CT scan is done of the brain, this artificial intelligence looks at that, and if it finds that there is a stroke, especially a large stroke, it immediately informs us as the neurologist. So what happens? In our smartphone, I get a message with all the brain images of the patient saying this patient in this hospital is having a stroke, and these are the brain images. And what we do is we send a helicopter to get the patient and bring him here, and we treat the patient. We just take out the clot and treat the patient. So this is the first artificial intelligence system approved in medicine in the United States, and it has really made a difference because it is cutting down 3 hours to 6 minutes now, which is a huge, huge change. And this artificial intelligence is not only being utilized in stroke, the potential is it is reading CT scans and MRIs better than radiologists would be reading it, right? It is looking at skin lesions better than skin doctors would read it. And so this is going to have a very major impact on human society. This is how it looks on my phone or on my iPad. All these images are sent to me, and all in under 6 minutes. The blood vessels of supplying blood to the brain is sent to me too. So that's how you look at it. So artificial intelligence is really making a huge difference. So these are just smartphone images that come through, within the 6 minutes of that patient hitting any emergency. What is even more fascinating, which I think all of us need to think about, is that the prediction is that within the next 30, they said 20, but now they're saying between 30 to 50 years, 47% of all employment in the United States will be done by artificial intelligence. So like factory workers, right? Car manufacturing, all of that will be done much better by artificial intelligence systems. So 47% of employees in the United States will become unemployed, not because of any economic downturn, but because the artificial intelligence is able to do those tasks much better than human beings can. So what is the future of the world going to look like? The future of the world is going to be where your governments will get the same amount of money or more, but people will have no jobs, right? So what will the government have to do? It then has to send out this money to people who have no work, but they need that money to be able to eat, move around, have a house, and all of that, right? So society and humans will have to learn to live in an environment where you have no job, but you have the money, right? Our very definition of who we are is now based on is determined by our profession. You're a engineer, you're a doctor, you're a politician, you're a bureaucrat, right? If factory workers. But if you take all that away, it's a very different world that we'll be living in, and it's not too far away. So I would suggest that leaders need to know about this and think about this, because it's going to profoundly change the way we live in times to come. I talked about the CRISPR gene editing technology. And if you remember in September, I had told you that very soon a Nobel Prize will be given. And if you heard, the Nobel Prize for chemistry was given to the people who discovered CRISPR technology, gene editing technology. How is this affecting us? So here's how it's affecting us. So now when a patient comes with a problem, we do a whole genome sequencing, which earlier used to take a lot of money and now can be done in $400 or even cheaper. And then you find out the genetic abnormality that is causing that disorder. You induce that genetic abnormality in an animal model. And on the animal model, which has now the same deficit disorder that you have, that your patient has, right? They try 1,200 medications on that animal model, figure out which are the 4 or 5 that work, and then provide that information to your physician, who then uses that information to treat you, right? This is called personalized medicine based on genetics, right? So this is already being, it's already started here in the United States. The other is the concept of curbing malaria, right? So by CRISPR gene editing technology, you can edit the genes of a mosquito that causes malaria and eliminate that mosquito completely, right? Or you can take the microbe that causes malaria and change that so it does not cause malaria, right? So you and I have now, or human beings have developed this ability to eliminate species off the planet that cause disorders. So again, people need to know this. It has positive roles, but it may have some unintended roles. So everyone needs to consider this as we go. You have senolytics, meaning how do you live forever, right? Everyone wants to live longer and forever. So senolytics is a science by which you get the dead cells out, and so the living cells can live more properly and don't get the inflammation and other things you get from dead cells. So essentially to increase your lifespan, right? Cellular therapies and regenerative therapies. So the only one that I will talk about is this for diabetics. Type 1 diabetics, what do they get? The cells producing insulin die off in the pancreas of that patient, right? Now, Doug Melton and his colleagues, what they have done is they're taking these beta cells that produce insulin and putting it into an immunoprotective device and then implanting it into the patient. And so that the patient now gets the insulin producing cells back and doesn't have to take insulin externally, doesn't have to test his blood sugar all the time. So, it's still in development, but very, very promising. And innovative vaccines. Now, right now, we're only talking about COVID vaccines, but there are all kinds of vaccines that are being produced. So there's a vaccine to lower your cholesterol. There's a vaccine to halt for you to get heart disease because it halts atherosclerosis. So heart disease and stroke, you get vaccines against them. Those are being developed. And you can get these e-tattoos. So you get these special tattoos that are monitoring your heart and sending that information to your physician, right? Just by tattoos, they're able to do that. So really important developments that are happening in this. And then lastly, I'm going to talk about this, which are these little robots. So essentially what they're doing is if you're having some surgery, say in your tummy, right? They make a small cut in your tummy and they put this little robot and they may not even have to do that. You can swallow it as a pill, and it then walks around in your body and does the surgery, and then you can eliminate that, right? So this kind of thing where you take a pill and pill actually goes in and this little robot comes out and actually does the surgery on your body, and then it's eliminated, right? So clearly, because of technology, there is huge changes in medical sciences at this point in time. And these exoskeletons, right, which can make you extremely powerful, but can also make paralyzed individuals start to walk because of these very sophisticated exoskeletons that you have now, right? So what I will do is I will stop here, and I will be happy to take any questions that you might have. So I'll stop sharing my screen.
Yes, sir. I would read out the questions to you and then you could answer them. Is that okay?
Yes. Yes,
Okay.
that should be fine.
Okay. So one of the first questions is, does the brain have the potential to do extraordinary things like healing the body? With an example, if you could help us out.
So the idea there is, I talked about this out-of-body experience that people talk about. That is, in some sense, leaving the body and going out. We have not been able to study it beyond a certain level. Now, one of the things that I teach my students is that you not only have to become an expert in the brain, you also have to become an expert on physical sciences. So a lot of you know that there is the string theory, right? And string theory says there are 11 dimensions to every object. And the only dimensions that we are able to perceive of the brain are 4: length, breadth, height, and age, right? But there are 7 other dimensions of this that we are unaware of. So we are really looking to collaborate with our people in fundamental physics to see if we can understand things better. And then over time, there has been this artificial distinction between the mind and the brain. But they're clearly one and the same things, and that's how these things function. So a long way to go there, but this is the best we have right now.
Okay, thank you so much. The next question, will lack of sleep in the deep stage result in dementia?
So we don't know whether it will or will not, but because this is all studies within the last 2 to 3 years. But we do know that during deep sleep is the best time when they can literally wash your brain of the toxins. And one of the toxins that is washed out is amyloid, which they did in the animals that were injected with it. So we do not have direct evidence in human beings yet, but we do know 2 things: that in animal models, it is certainly washing out the abnormal toxin that causes dementia. And we do know that in people who have dementia, they have abnormal sleep patterns. So between the 2, I would say getting enough sleep and the right kind of sleep is really important for not just dementia, but also for a lot of other types of brain disorders.
Thank you so much. Do we understand why we are paralyzed during sleep?
So we do understand that. We know that the exact neural pathway that I showed that goes through SLD to premotor areas, and from premotor to inhibit the motor neurons, and that's how you're paralyzed. But the pathway is very different from the regular pathways that we have. So we do understand that. We probably have some idea why you need to get paralyzed, otherwise you'll be acting out your dreams. But the more fundamental question is, we don't understand why we dream. And that's what we are all engaged in research on, to really figure out why should someone dream at all.
Okay, there's a related question, so I'm asking that right now. Which circuits would be strengthened during sleep and would wear off? This kind of epigenetic control, is it really a conscious move or something else governs it?
So a very good question. The idea there is we really don't know. We have just learned within the last year or so that this happens. It would be very, very difficult to study it in a human brain to really figure out how this would be, how this could happen. So we are at its infancy at this point in time. What we do know is that synaptic connections will break off or will become less connected during sleep. So essentially, you're getting enlightened forgetting. So the things that we think that your brain does not consider important, you forget, right? And the things that it considers important, you remember. And the brain should normally function in an evolutionary manner. So what is important for your survival, you will remember, and the others will not. But there is certainly something to be said about the fact that maybe can we direct that, which memories are broken off and which memories remain. But we are not there yet in terms of our research.
Okay. Would you say blind sight is similar to the phantom limb phenomena?
So, not particularly. Phantom limb, if you look at Professor Ramachandran's explanation, that's his seminal work of phantom limb. Essentially, what's happening is that the limb is not there, but the brain representation of the limb in the brain is still there. So when that area in the brain, if you touch, it feels like you're touching part of the body. So that's the concept of phantom limb. So blind sight is telling us that there is conscious ability and unconscious ability, conscious vision and unconscious vision, because the evolutionary pathways in the brain still remain. And so we may, unless an ability reaches the cortex, we are not consciously aware of it. So we are not aware of our visual ability, but the visual ability still exists. And what we need to study further is this true of other abilities? So for hearing, we think we are only hearing certain things, but are we hearing more than that? So those kinds of things have not been studied yet in greater detail, and that's what's fascinating.
In sleep paralysis, what is the trade-off between awareness and wakefulness?
So sleep paralysis happens during REM sleep, and it happens essentially so that you don't act out your dreams. And then it goes away. So when you're in non-dream phases of your sleep, or what we call non-REM sleep, you're not paralyzed at that time. That time, your body is completely there. So you are not conscious, obviously during, or conscious as in awake during your REM sleep. And so the conscious awareness part is not there at all. It's, as I talked sometime before, it's fascinating that thousands of years ago in Indian texts, they talk about the concept of Jagrat, which is awake, right? They talk about Swapna, which is your dream sleep, which is REM sleep essentially. And they talk about Sushupti, which is your dreamless sleep, right? And they lay a great deal of importance on Sushupti, which is your dreamless state. And now we are discovering that it is the slow non-REM sleep is when this washing of the brain happens quite a bit. So it's been fascinating to really see those and maybe to study them in greater detail. They may have some information that we may really benefit from.
The next questions are dream related. Why do we remember certain dreams only? And is it good to have dreams? Does it affect a person's consciousness?
So the dreams we remember. So if you wake up during a REM sleep, so when you're dreaming, if you wake up during that because something disturbs your sleep and you wake up, or you wake up very soon after a REM phase or dream phase, you will remember that dream. So if we wake you up during REM sleep, you will vividly remember the dream. And we are doing that in our research studies. They will vividly remember the dream at all. The fascinating question is that the current concepts of neuroscience say that when you're in this dream phase and REM sleep, you're cut off from the environment. We don't think so because, some of you might have experienced this, that when you are, when in the morning you're in a dream and something happens in your environment, like someone comes into the room, you're trying to think whether that person is part of your dream or is this real, right? And then you get up and you see that person. So that has given me the idea that in our research study, we are trying to see if we can influence your dream. So how do we do that? So suppose if you were dreaming, like you're going down while the person is dreaming, and we can tell through brainwaves that they're dreaming, right? We play a sound, right? Like say bells ringing. And when we wake up the person later and the person says, I was walking down the street and suddenly bells started ringing. That would prove that we can influence the dreams and we can change the dreams by external stimuli, right? But the real fundamental question that we are trying to study is, why do you dream in the first place? And that will answer the second part of that question. why do you dream? That would tell us whether things are good or bad and why are these things happening. And despite all this knowledge, we still have no idea why someone is dreaming.
Can empathy develop through activation of the mirror neurons? Are there significant parameters for empathy, for learning empathy?
So as you saw, it's just a few months ago that we learned that mirror neurons may have a role to play in empathy, at least in perception of pain, because of the activation of those regions. So if we are, what I wanted to do is to share with you cutting edge stuff. And so it should give you ideas that, and you may develop some concepts from it that we have not, and we may benefit from you. So clearly, mirror neurons certainly have a role to play in empathy, and we can actually measure in this particular perception of pain through the ACC. We can actually measure the level of empathy by the level of activation. This is something we have never been able to do in the past. Study empathy by level of brain activation in a certain region. So it's fascinating, but we are really starting down this journey. We are not too far off.
Thank you. Do the terms IQ and common sense have something to do with the brain, or are they only dependent on how we are trained and how our experiences in life are?
So it's a very interesting concept of nature versus nurture, right? Are we born with certain things, or are we only trained with certain things? The concept is it is likely that both play a role. The genetic constitution and the formation of your brain certainly play a role in that, but the nurture or the experiences that you have also play a role in that. So the whole birth to 3 programs in Western societies is that you enrich the environment of a child and make them listen to the good kind of music, and that will lead to right connections in the brain cells, and the wrong connections will break off. And so we don't have any hard evidence of that, but we do know that when a baby is born, they have many more connections than an adult brain. And then over the next few years, some connections will break off and an adult brain will develop, right? So the concept here is that if you enrich the environment, the right connections will remain and the wrong connections will break off, right? But there is no hard neuroscience to prove that, even though there are government programs to do it. I'm sure there's no harm in enriching the environment of a baby to do this kind of concept. One thing that I do want to tell you is, so when babies are born, they have many more connections in their brains, right? Over time, some connections will break off, others will remain, and that will be an adult brain. We think that breakage, what we call pruning of synapses, or the breakage of those brain connections, develop unique brains in every individual. And that is why you have unique personalities in every human being on the planet, because the way those synapses break off or those connections break off could be different in different people.
Yeah, thank you. The concept of FOX2 genes that you had mentioned, is it possible that we can add the 2 amino acids in the chimpanzees and enable language or communication in them?
So as I said, just last year, they put these, just a few years back, they put this human FOXP2 in mice. And those mice became much more intelligent. They could go through the maze much quicker, much more easily than the others could. And so a chimpanzee has not been tried yet, but I'm sure it is not just language, but the intellectual abilities that we develop because of language, they come through this. So can it be done? Yes. Has it been done? Not that I'm aware of.
Neuroscience have prescription for
Could you repeat that, please?
Does neuroscience have a prescription for improved REM sleep?
So neuroscience does not have a prescription for doing that. It is sort of, but we've been looking because it's an important part of sleep. So we do what's called sleep hygiene, and that should improve sleep. We don't have a way of increasing your non-REM sleep yet. We do know that if you give them antidepressants like Prozac and Zoloft, they do decrease the REM part of your sleep, so the non-REM would go up. But we don't have that. what is fascinating is that if you read some of the ancient yogic texts, they will tell you techniques on how the sushupti part can increase. And so the concepts like yoga nidra and all are profoundly based on some of those kinds of concepts.
Does brainwashing take place in people who suffer from insomnia?
So that's what we are looking at, because insomnia, this is a huge problem, because if in insomnia, they just over time, insomniacs, if they don't sleep much at all, they develop other kinds of problems. And so now we might have an answer as to why they develop other kinds of problems, because these toxins are not being washed off in these individuals. So, but now, fortunately, we have much better ways of treating insomnia. Because earlier it was just giving you a sedative, right? That was the general treatment of insomnia. Now we know why people go to sleep, how people go to sleep, and how they wake up. And so we know the three essential chemicals of sleep, which is melatonin, adenosine, and orexin, right? Or hypocretin. So now that we know how your brain goes to sleep, how your body and brain go to sleep, we know the role of these three chemicals. New therapeutics are being developed based on this knowledge. So now there is a new drug that actually prevents orexin action. Orexin is what keeps you awake. So if that is being blocked, and that those kinds of drugs, based on our fundamental understanding of the neurophysiology of sleep, they will have a profound impact. One is already available. That will have a profound impact in treating insomniacs, because I think that can now be treated now that we understand the basic physiology of sleep and wakefulness.
Ma'am, there's no audio.
So the next question is on synaptic pruning. What synaptic in development, even progression of adult onset?
So we don't know if synaptic pruning plays any role in schizophrenia. There's been no correlation there. What has been is in brain development has been related, has been found to have some relationship to schizophrenia. So what happens is when your cortex of your brain is developing, you get a bunch of cells in the middle of the brain, and then they migrate outwards like this, right? And one of the chemicals that controls this migration is called renin. And that renin protein has been connected with schizophrenia. So that is there. The pruning itself has not been really definitively linked with schizophrenia.
According to you,
Miss Roshni, please mute yourself.
So will meditators have less chances of getting dementia, in your opinion?
We'll have to do a study to prove it. So there are a couple of problems, in studying this. There is a exhaustive document on meditation that is being brought out by the United States Health and Human Services Department of Ministry. And so they've looked at different types of meditation and how they affect it. The problem is that these studies would have to be done on master meditators. We are trying to do those studies here in the United States. What we lack is the master meditators. So I would suggest that these studies probably can be done better in India, where you might find master meditators. You just have to have this right kind of equipment, and then those studies can be done better there than here. But yes, the thought is that there should be decreased incidence. The issue also is that in some of the preliminary studies coming out of India, there is some decreased incidence of dementia. It doesn't mean that people are meditating. What it also means is that dietary factors are being looked at. So, like curcumin, which is in haldi, that has been shown to prevent brain cell damage from oxidative stress. And so We did a study over the last 7 years or so with my collaborators where we have found that there's a particular substance in the brain called peroxydodoxin that is upregulated by curcumin, and that prevents brain cells from dying from oxidative stress, which can be found in dementia. So meditation studies have yet to be done, but because there is circumstantial evidence, I will not deny it, but there is no direct evidence yet, and that has to be developed. But yes, dietary factors can also help in prevention of dementia.
Question is, as per neurodevelopmental hypothesis, it is schizophrenia at the developmental stages, and often aberrant synaptic pruning early onset. What are your thoughts on this?
So this aberrant synaptic pruning would have to be proven, and we have no way of proving that in humans yet, because you'd have to literally look at the brain and its connectivity in real time at a cellular level. And, with 100 billion brain cells and 10,000 potential connections of each brain cell, that's a highly complex thing to do. So there are some animal studies that have been done, and there are conflicting evidence in that animal studies. So is there a developmental aspect to it? Could be, definitely. But is it pruning or is it cortical development through this reelin protein? Because the reelin abnormalities have been found in human beings. That reelin protein, which determines this radial migration of neurons, that abnormality has been linked to schizophrenia. The others, there is not a very good direct evidence on that. So a lot of work is being done.
Can meditation protect against cognitive decline?
Yeah, yoga and meditation, we don't have any direct evidence yet that it can, but there is a lot of circumstantial evidence, because if meditation can increase gray matter and prevent gray matter loss in people who do this regularly, one can certainly extrapolate to say that it can be beneficial.
Next, ma'am.
What is your view on using CRISPR on COVID? Will it be successful?
So CRISPR on COVID, probably in the long term, it may have a role in it, but in the short term, we need to really find a vaccine. That's our only hope in the short term. So I'm glad that we have a few candidates that are in phase 3 trials. And that's what we need to really concentrate on. In the long term, it could have some impact, but not in the short term.
Next, ma'am.
Has any more questions? Because I've, I think, exhausted on the chat.
Please type in your questions. So there's one more.
Okay, another question. Could you describe about your interest in artificial intelligence and EEG?
So yes, we are developing an artificial intelligence system on EEG with our partners in the School of Engineering in Lincoln, and we've had some publications on that. The idea is that we use artificial intelligence paradigms to read your brain waves and detect when seizures are happening, and predict that. So what happens is that you have seizures occur even in the patients who have really bad seizures. This happens only 1% of your lifetime. But the problem is you don't know when it's going to happen. And so that is why it becomes a problem. So what we are trying to do is predict about a minute or 5 minutes before a seizure is about to happen, based on your brainwave patterns, to determine that this is about to happen. And so then it can, sort of text you on your smartphone that seizure about to happen in 5 minutes. Then you could take some therapy, and that will, some medication that will stop it, or you could at least go to a safe area and have your seizure and then move on from there. So we are developing that. We've published a bit on this, and it's a very active area of research, artificial intelligence and EEGs.
That our question now,
right,
for a vote of thanks to you, sir. Thank you, Dr. Singh, for the amazing amount of knowledge that you shared with us. It panned a whole lot of. It was interesting to learn that just like the brain, we do not. There are lots of things we are still not familiar about, especially the genome, the human genome. It was also amazing to learn about the concept of visual agnosia, the relation between the jerky eye movements as well as sleep was also very interesting for all of us. We probably do not give as much of importance to sleep, and I'm hoping with your lecture, particularly the youngsters will start focusing on the relevance and importance of sleep, particularly its memory. The concept of mirror neurons and what they can do to us, I call it to me about empathy and mirror. If by activation we could really increase empathy, I think that would help so many of us. Concept of time travel, really, again, very fascinating. One is time travel from the perspective of neuroscience. Idea about the diseases that are developed through the default mode in the brain and how is coming to the rescue of all those may probably be the area of the future, and we are grateful to you for having taken time out at such short notice and having given us an extensive understanding about the brain, because truly whatever you shared with us, for a lot of us it was something that we were not really looked at. Like you said, you shared with us cutting research. So we are extremely grateful to Dr. Singh for having even taken all those questions. Heard you the first time round, we thought that was amazing, but really given us a keynote address, and we are all from the team at Gargi College, organizers of this, are extremely grateful to you for having taken time out for us. Thank you so very much.
Thank you.
Hello, everyone. As we are left with some time till our next talk begins, so as we had requested and we have taken permission from Professor Ramachandran, so we would play a short video of his on TED Talks. So please be hooked.
Chris pointed out, I study the human brain, the functions and structure of the human brain, and I just want you to think for a minute about what this entails. Here is this mass of jelly, 3-pound mass of jelly, you can hold in the palm of your hand, and it can contemplate the vastness of interstellar space, it can contemplate the meaning of infinity, and it can contemplate itself contemplating on the meaning of infinity. And there is this peculiar recursive quality that we call self-awareness, which I think is the holy grail of neuroscience, of neurology, and hopefully someday we'll understand how that happens. Okay, so how do you study this mysterious organ? I mean, you have 100 billion nerve cells, little wisps of protoplasm, interacting with each other. And from this activity emerges the whole spectrum of abilities that we call human nature and human consciousness. How does this happen? Well, there are many ways of approaching the functions of the human brain. One approach, the one we use mainly, is to look at patients who have sustained damage to a small region of the brain, or there's been a genetic change in a small region of the brain. What then happens is not an across-the-board reduction in all your mental capacities, a sort of blunting of your cognitive ability. What you get is a highly selective loss of one function with other functions being preserved intact. And this gives you some confidence in asserting that that part of the brain is somehow involved in mediating that function. So you can then map function onto structure and then find out what the circuitry is doing to generate that particular function. So that's what we're trying to do. So let me give you a few striking examples of this. In fact, I'm giving you 3 examples, 6 minutes each during this talk. The first example is an extraordinary syndrome called Capgras syndrome. If you look at the first slide there, that's the temporal lobes, frontal lobes, parietal lobes, okay, the lobes that constitute the brain. And if you look tucked away inside the inner surface of the temporal lobes, you can't see it there, is a little structure called the fusiform gyrus. And that's been called the face area in the brain because when it's damaged, you can no longer recognize people's faces. You can still recognize them from their voice, say, oh yeah, that's Joe. But you can't look at their face and know who it is. Right? You can't even recognize yourself in the mirror. I mean, you know it's you because when you wink, it winks and you know it's a mirror. But you don't really recognize yourself as yourself. Okay. Now that syndrome is well known, it's caused by damage to the fusiform gyrus, but there's another rare syndrome, so rare in fact that very few physicians have heard about it, not even neurologists. This is called the Capgras delusion. And that is a patient who's otherwise completely normal, who's had a head injury, comes out of coma, otherwise completely normal. He looks at his mother and says, this looks exactly like my mother, this woman, but she's an imposter. She's some other woman pretending to be my mother. Now, why does this happen? Why would somebody, and this person is perfectly lucid and intelligent in all other respects, but when he sees his mother, this delusion kicks in and says, not mother. Now, the most common interpretation of this, which you find in older psychiatry textbooks, is a Freudian view. And that is that this chap, and the same argument applies to women, by the way, but I'll just talk about guys. When you're a little baby, when a young baby, you had a strong sexual attraction to your mother. This is the so-called Oedipus complex of Freud. I'm not saying I believe this, but this is the standard Freudian view. And then as you grow up, the cortex develops and inhibits these latent sexual urges towards your mother. Thank God, otherwise we'd all be sexually aroused when you saw your mother. And then what happens is there is a blow to your head, damaging the cortex, allowing these latent sexual urges to emerge flaming to the surface, and suddenly and inexplicably you find yourself being sexually aroused by your mother. And you say, my God, if this is my mom, how come I'm being sexually turned on? She's some other woman, she's an imposter. It's the only interpretation that makes sense to your damaged brain. This never made much sense to me, this argument. It's very ingenious, as all Freudian arguments are. Didn't make much sense because I've seen the same delusion, a patient having the same delusion about his pet poodle. He'll say, doctor, this is not Fifi. It looks exactly like Fifi, but it's some other dog, right? Now you try using the Freudian explanation there. You have to start talking about the latent bestiality in all humans or some such thing, which is quite absurd, of course. Now, what's really going on? So to explain this curious disorder, we look at the structure and functions of the normal visual pathways in the brain. Normally, visual signals come into the eyeballs, go to the visual areas in the brain. There are, in fact, 30 areas in the back of your brain concerned with just vision. And after processing all that, the message goes to a small structure called the fusiform gyrus, where you perceive faces. There are neurons there that are sensitive to faces. You can call it the face area of the brain, right? I talked about that earlier. Now, when that area is damaged, you lose the ability to see faces, right? But from that area, the message cascades into a structure called the amygdala in the limbic system, the emotional core of the brain. And that structure called the amygdala gauges the emotional significance of what you're looking at. Is it prey? Is it predator? Is it mate? or is it something utterly trivial like a piece of lint or a piece of chalk or I don't want to point to that, but or a shoe or something like that, okay, which you can completely ignore. So if the amygdala is excited, and this is something important, the messages then cascade into the autonomic nervous system. Your heart starts beating faster, you start sweating to dissipate the heat that you're going to create from exerting muscular exertion. And that's fortunate because we can put 2 electrodes on your palm and measure the change in skin resistance produced by sweating. So I can determine when you're looking at something whether you're excited or whether you're aroused or not, okay? And I'll get to that in a minute. So my idea was when this chap looks at an object, when he looks at any object for that matter, it goes to the visual areas, and however, and it's processed in the fusiform gyrus, and you recognize it. A plant or a table or your mother for that matter, okay? And then the message cascades into the amygdala and then goes down the autonomic nervous system. But maybe in this chap, that wire that goes from the amygdala to the limbic system, the emotional core of the brain, is cut by the accident. So because the fusiform is intact, the chap can still recognize his mother and says, oh, yeah, this looks like my mother. But because the wire is cut to the emotional centers, he said, but how come if it's my mother, I don't experience a warmth or terror, as the case may be, right? And therefore, he says, that, how do I account for this inexplicable lack of emotions? This can't be my mother. It's some strange woman pretending to be my mother. How do you test this? Well, what you do is if you take any one of you here and put you in front of a screen and measure your galvanic skin response and show pictures on the screen. I can measure how you sweat when you see an object like a table or an umbrella. Of course, you don't sweat. If I show you a picture of a lion or a tiger or a pinup, you start sweating, right? And believe it or not, if I show you a picture of your mother, I'm talking about normal people, you start sweating. You don't even have to be Jewish. Yeah? Now, what happens if you show this patient? You take the patient and show him pictures on the screen and measure his galvanic skin response. Tables and chairs and lint, nothing happens, as in normal people. But when you show him a picture of his mother, the galvanic skin response is flat. There's no emotional reaction to his mother, because that wire going from the visual areas to the emotional centers is cut. So his vision is normal because the visual areas are normal. His emotions are normal. He'll laugh, he'll cry, so on and so forth. But the wire from vision to emotions is cut, and therefore he has this delusion that his mother is an imposter. It's a lovely example of what the sort of thing we do. Take a bizarre, seemingly incomprehensible neuropsychiatric syndrome and say that the standard Freudian view is wrong, that in fact you can come up with a explanation in terms of the known neuroanatomy of the brain. By the way, if this patient then goes and mother phones from an adjacent room, phones him, and he picks up the phone, he says, wow, Mom, how are you? Where are you? There's no delusion through the phone. Then she approaches him after an hour, he says, who are you? You look just like my mother. Okay? The reason is there's a separate pathway going from the hearing centers in the brain to the emotional centers, and that's not being cut by the accident. So this explains why with a phone, he recognizes his mother, no problem. When he sees it in person, he says it's an imposter. Okay. Now, how is all this complex circuitry set up in the brain? Is it nature, genes, or is it nurture? And we approach this problem by considering another curious syndrome called phantom limb. And you all know what a phantom limb is. When an arm is amputated or a leg is amputated for gangrene, or you lose it in war, for example, in the Iraq war, it's now a serious problem. You continue to vividly feel the presence of that missing arm, and that's called a phantom arm or a phantom leg. In fact, you can get a phantom with almost any part of the body. Believe it or not, even with internal viscera. I've had patients with the uterus removed, hysterectomy, who have a phantom uterus, including phantom menstrual cramps at the appropriate time of the month. And in fact, one student asked me the other day, do they get phantom PMS? A subject ripe for scientific inquiry, but we haven't pursued that. Okay, now the next question is, what can you learn about phantom limbs by doing experiments? One of the things we found was about half the patients with phantom limbs claim that they can move the phantom. It'll pat his brother on the shoulder, it'll answer the phone when it rings, it'll wave goodbye. These are very compelling, vivid sensations. The patient's not delusional, he knows that the arm is not there, but nevertheless it's a compelling sensory experience for the patient. But however, about half the patients, this doesn't happen. The phantom limb, they'll say, doctor, the phantom limb is clenched spasm and it's excruciatingly painful. If only I could move it, maybe the pain will be relieved. Now, why would a phantom limb be paralyzed? It sounds like an oxymoron. When we looked at the case sheets, what we found was these people with the paralyzed phantom limbs, the original arm was paralyzed because of a peripheral nerve injury. The actual nerve supplying the arm was severed, was cut by, say, a motorcycle accident. So the patient had an actual arm, which is painful, in a sling for a few months or a year, and then in a misguided attempt to get rid of the pain in the arm, the surgeon amputates the arm, and then you get a phantom arm with the same pains, right? And this is a serious clinical problem. Patients become depressed, some of them are driven to suicide, okay? So how do you treat this syndrome? Now, why do you get a paralyzed phantom limb? When I looked at the case sheet, I found that they had an actual arm, and the nerve supplying the arm had been cut, and the actual arm had been paralyzed and lying in a sling for several months before the amputation, and this pain then gets carried over into the phantom itself. Why does this happen? When the arm was intact but paralyzed, the brain sends commands to the arm, the front of the brain, saying, move. But it's getting visual feedback saying, no, move, no, move, no, move, no. And this gets wired into the circuitry of the brain, and we call this learned paralysis. Okay, the brain learns, because of this Hebbian associative link, that the mere command to move the arm creates a sensation of a paralyzed arm. And then when you amputate the arm, this learned paralysis carries over into your body image and into your phantom, okay? Now, how do you help these patients? How do you unlearn the learned paralysis so you can relieve him of this excruciating clenching spasm of the phantom arm? Well, we said, what if you now send the command to obeying his command, right? Maybe you can relieve the phantom pain, the phantom cramp. How do you do that? Well, virtual reality, but that costs millions of dollars. So I hit on a way of doing this for $3, but don't tell my funding agencies. Okay? What you do is you create what I call a mirror box. You have a cardboard box with a mirror in the middle, and then you put the phantom. So my first patient, Derek, came in. He had his arm amputated 10 years ago. He had a brachial avulsion, so the nerves were cut. The arm was paralyzed, lying in a sling for a year, and then the arm was amputated. He had a phantom arm, excruciatingly painful, and he couldn't move it. It was a paralyzed phantom arm. So he came there, and I gave him a mirror like that in a box, okay, which I call a mirror box, right? And the patient puts his phantom left arm, which is clenched and in spasm on the left side of the mirror, and the normal hand on the right side of the mirror, and makes the same posture, the clenched posture, and looks inside the mirror. And what does he experience? He looks at the phantom being resurrected, because he's looking at the reflection of the normal arm in the mirror, and it looks like this phantom has been resurrected. Now I said, now look, wiggle your phantom, your real fingers, or move your real fingers while looking in the mirror. He's going to get the visual impression that the phantom is moving, right? That's obvious. But the astonishing thing is the patient then says, oh my God, my phantom is moving again, and the pain, the clenching spasm is relieved. I remember my first patient who came in, my first patient came in and he looked in the mirror, and I said, look at your reflection of your phantom. And he started giggling so that I can see my phantom. But he's not stupid. He knows it's not real. He knows it's a mirror reflection, but it's a vivid sensory experience. Now I said, move your normal hand and phantom. He said, oh, I can't move my phantom. You know that. It's painful. I said, move your normal hand. He says, oh my God, my phantom is moving again. I don't believe this. And my pain is being relieved, okay? And then I said, oh, nothing, it's clenched again. Okay, open your eyes. Oh my God, it's moving again. So he's like a kid in a candy store. So I said, okay, this proves my theory about learned paralysis and the critical role of visual input. But I'm not going to get a Nobel Prize for getting somebody to move his phantom limb. It's a completely useless ability if you think about it. But then I started realizing maybe other kinds of paralysis that you see in neurology, like stroke, focal dystonias, there may be a learned component to this which you can overcome with the simple device of using a mirror. So I said, look, Derek, well, first of all, the guy can't just go around carrying a mirror to alleviate his pain. I said, look, Derek, take it home and practice with it for a week or two. Maybe after repeated practice, you can dispense with the mirror, unlearn the paralysis, and start moving your paralyzed arm, and then relieve yourself of pain. So he said, okay, and he took it home. I said, look, it's after all $2, take it home. So he took it home, and after 2 weeks, he phones me, and he said, doctor, you're not going to believe this. I said, what? He said, it's gone. I said, what's gone? I thought maybe the mirror box was gone. He said, no, you know, this phantom I've had for the last 10 years, it's disappeared. And I said, I got worried. I said, my God, I've changed this guy's body image. What about human subjects, ethics, and all of that? And I said, Derek, does this bother you? He said, no. Last 3 days, I've not had a phantom arm, and therefore no phantom elbow pain, no clenching, no phantom forearm pain. All those pains are gone away. But the problem is I still have my phantom fingers dangling from the shoulder, and your box doesn't reach. So can you change the design and put it on my forehead so I can, do this and eliminate my phantom fingers? He thought I was some kind of magician. Now, why does this happen? It's because the brain is faced with tremendous sensory conflict. It's getting messages from vision saying the phantom is back. On the other hand, there's no proprioception, muscle signals saying that and your motor command saying there is an arm. And because of this conflict, the brain says, to hell with it, there is no phantom. There is no arm, right? It goes into a sort of denial. It gates the signals. And when the arm disappears, the bonus is the pain disappears, because you can't have disembodied pain floating out there in space. So that's the bonus. Now, this technique has been tried on dozens of patients by other groups in Helsinki, so it may prove to be valuable as a treatment for phantom pain. And indeed, people have tried it for stroke rehabilitation. Stroke you normally think of as damage to the fibers, nothing you can do about it, but it turns out some component of stroke paralysis is also learned paralysis, and maybe that component can be overcome using mirrors. This has also gone through clinical trials, helping lots and lots of patients. Okay, let me switch gears now to the third part of my talk, which is about another curious phenomenon called synesthesia.
Thank you everyone for listening in. We have our next lecture now. So I hand over the mic to Dr. Neera.
Thank you, Jaswinder. I would now like to welcome our guest of honor, Professor Pravat Mandal. Just an important announcement for the participants who've just joined. All questions for Professor Mandal should be posted in the chat section, and they'll be taken up towards the end of the session. Thank you. Professor Mandal graduated from IIT Madras and was a Tata Innovation Fellow. He taught at the Department of Psychiatry, University of Pittsburgh Medical School, and later in the Radiology Department, Johns Hopkins Medicine, USA. Currently, Dr. Mandal is a senior professor at the National Brain Research Center, India, where he heads a multidisciplinary research laboratory. He is also an honorary professor at the Flores Institute of Neuroscience and Mental Health, Australia. Dr. Mandal's work has been highlighted in Scientific American Mind and the New Scientist for his out-of-the-box ideas and implementation. He has discovered the early diagnostic biomarkers of Alzheimer's disease and is the creator of the Indian brain template Brahma, international database Ansh, and novel brain signaling processing package Kalpana. Welcome, Professor Mandal. We are all eager to listen to your talk.
Okay.
Yeah, I can see that also.
Sir, please let me know whenever you want me to just play on the next slide. So I'll put it on the theater mode.
Okay. So yeah, that is fine. Good morning, everyone. It's my honor to be giving a talk, and I was listening very patiently for other speaker, Dr. Singh, and what a nice talk and with a high rich content. This is very enriching. Here we are going to talk about the Brahma, the development of population-specific MRI template for the Indian population. The reason being that we are, and I was told that the main purpose of this great college, Gargi, to enrich or the students so that at some point some of you can come to the neuroscience research and enrich this area. So this is why the purpose is that to show that there are many aspects that are available in brain research. So here, and the reason I'm going to, next slide please. Next one please. Next slide please.
Okay. It's not working,
Yeah,
sir.
next slide please. Okay.
Okay.
Yeah. So the reason I am putting this one is all are meaningful way that brain research is actually not possible to do in a single institute or single laboratory. It need different types of people and also different environment, because we are human, we are understanding the human brain behavior, we want to predict, we are living in the age of artificial intelligence. So here we have to have a various types of information available. So a collaborative research, until unless is in full place, the outcome of the research will not be possible, meaningful way outcome. So this is, that's what we want to show them, that the various scientists from doctors, All Indian Institute of Medical Science, and in other continent, in also in Flore. So these places people contribute and they share the ideas, and that's why good things are happening. Now, next slide, please. Yes. So here I will spend some time because you can see that I'm not talking about Brahma yet. Brain is an entity. Everybody knows that in MRI. MRI is actually a structural aspect of it. Suppose somebody has any tumor in the brain, you can do MRI, you can find it out correctly that how much of the brain has the tumor, whether tumor is a benign or it has any other aspect of it or is a cancerous, that can be looked at by MRI. But in the case of if you look at a depressed person, suppose somebody having a depression, if you look at their MRI images, you will find it out there is no abnormality whatsoever in his brain, because the brain MRI structure will be as usual like any other normal person. So this MRI doesn't help to for that type of person, depression or any psychiatric problem. Similarly, there are other issues will be there, like a functional MRI. That part the earlier speaker was raising about. Functional MRI means brain is a dynamic and living entity. Suppose you activate particular regions, that particular activity monitoring parts in the brain, it will be activated, and that you can monitor by functional MRI. This functional MRI is very, very critical. Let me give you some example. Suppose somebody comes to us, then he said he will have a surgery after some time. So he wants to see whether it is motor cortex area there is a tumor. So whether after surgery the neurosurgeon wants to know that whether after removal of this tumor, whether it will have any impact or not, or how much it has impacted. So they do pre-surgery or functional MRI activities and see the activity of this person, and after surgery again the same person after a 1 month or so, when the person goes for the functional MRI, you can see that one, although tumor is removed but the functional aspect is remain intact, so it was a correct surgical procedure. So this is also there. Now another aspect I want to highlight is called MRS. Brain is full of neurochemicals. Brain, this point I raised to you that any psychiatric problem or any Alzheimer or neurodegenerative problem, the neurochemicals actually are having a serious implication because these are the things that changes in the case of psychiatric problem or in the case of Alzheimer related problem. So these three components, one is called MRI, one is called functional MRI, one is called MRS. They are independent and they are very helpful to each other. They give a supportive information which you cannot get in other technique. So this is why I want to introduce you the three different aspects. But you please know that this you don't have to go to the different MRI machine. Same machine can do this one. It need to have a specific hardware. So same machine can do MRI, same machine can do fMRI, same machine can do MRS. But MRS to do you need a very expensive parts like a coils, head coils you need. So that's what you get all this information from your imaging output. Okay, so these need to be analyzed case to case basis with a earlier, with a healthy control and with the disease condition and how the disease is progressing. So these are can be monitored very well. So these are wide aspect of this MRI is I'm going to explain to you. Now next slide please. Yes. So this is another very nice representation that from the brain you can get various types of neurochemical. This first one you get, you can see that from the same parts, frontal cortex, and you get a glutathione that is antioxidant. You also get the ATP levels, and you also get the N-acetyl aspartate level, inositol level. These are all very important, and this is taken from a normal person, so there are not many abnormality is observed. But in clinical cases that we do regular basis on Alzheimer patient, Parkinson patient, we do see a significant differences, and it is very easy for us to help the patient. All patients come from AIMS or Medanta or Paros Hospital. So here we are, you can see this is very important. This is your brain and a particular area of the brain. You can get all this information and you will be able to quantify. You will never tell that you have this much amount. You will say, this is your clinical condition, this is your amount of chemicals, and this is what was there for about 2 years ago. So this is so very useful for us. Now, next, so this is very, very important that from the brain you can get different types of information, and that is directly clinically relevant for us. Next slide, please. Here, so now coming back to our topic, how this idea that the Brahma came about, we have been Indian, there is only one template available in the Montreal Neurological MNI temple. So everybody follow their template. So we actually thought of that we should not depend on the American or Canadian brain because they are different. So we should have our own, and that's what that idea came, and DST gave us some money to start with and make it Indian brain template. And that's what a Hindu likes. The product came here to discuss, and he likes it, and he put it in a, he was fascinated by all this. So this came here, and it gives a moral booster. So we actually pursue the dream, and ultimately we got it done. And it is successfully launched, and it is available free for any academic use. in India and anywhere else in the world. Next one, please. Yes. So, as earlier speaker was saying, in our research, we may have ideas, but until unless it is published in a form of paper, peer-reviewed paper, journals, it is not any valid thing. We cannot claim anything. So in this case, you can see, first we thought of having the Indian brain template, and we need to do this one. That came in May 2012. In Journal of Alzheimer's Disease, we published that one article that what should be done, and they took us 8 years to ultimately a complete form of paper or with all information, that is Brahma. It took us 8 years because there are many factors. We have to collect the data all over the India, so it should represent a true pan-Indian template. So this is why it came. Now, next slide, please. Yes. I was trying all the other, right now it is, as I was telling, that AI is the main part is taking, but AI is depend on the data. How much data, how quality is the data, how validated is the data. That is the main issue. That is the main yardstick of success in that case. Now, frontier in neuroinformatics, we realize that we are actually putting a huge amount of neurochemical data, which is not available for Alzheimer's. We are actually putting them through so that anybody, anywhere in the world, they can use it. Now, what is the novelty? Till date, there is no such data like neurochemical glutathione, GABA, glutamate. This data is not there anywhere in the world. So at National Brain Research Center, we have developed it, and now we have published it in Frontier anyway. This is being expanded. So maybe a major program is expected on data development and nursing. So this will be expanded. So this is another aspect that we are continuously evolving to reach to the next level. Now, next slide, please. Next slide. I think the video will be coming. The reason I'm saying that one, there is a special program where Dr. Tandon and myself was invited, and that is very much required. So it will be very helpful for the students to see how top neurosurgeon look at it and a scientist look at it. So please listen. This is about 18 minutes video that is on the same topic. So I'll stop now. After that, I'll rejoin.
Now I would request one of my colleagues to play the video. Anyone, Udita or Dr. Madhu? Can you please?
ये कैसा लगता है?
Yeah, it's working now.
देखिए, मेरी उम्र की जो आपसे बात करी, उसके लिए शायद ये जरूरी है.
It's working.
देखिए, मेरी उम्र की जो आपने बात करी, उसके लिए शायद ये जरूरी है कि ऐसा मैप हो. लेकिन कोई नई चीज नहीं है. सबसे पहला मैप बना था कोई 1950 के करीब, 45 थिंक लाइक दैट. ओके, उस वक्त उसका दूसरा पर्पस था.
जी जी.
उसके बाद कई मैप बने दुनिया में डिफरेंट. स्पीगल एंड वाइसेस ने बनाया, शाल्टन ब्रांड ने बनाया, नारा बायोशी ने जापान में बनाया.
जी जी.
उस वक्त इसका फंक्शन एक था कि जब हम पार्किंसंस डिजीज के पेशेंट को ट्रीट करते थे तो ब्रेन के खास हिस्से को डिस्ट्रॉय करना होता था. तो बिना ब्रेन को खोले हुए उस जगह पहुंचने के लिए वो मैप हेल्प करता था कि कैसे वहां पहुंचा जाएगा.
जी जी जी.
तो ये उस वक्त फंक्शन था. वो हम लोगों ने इस्तेमाल किया न्यूरो सर्जरी में. उसके बाद एक एरा आया जब वो डिस्ट्रॉय करने की जगह स्टिमुलेट करने लगे. इलेक्ट्रोड डाल देते थे वहां पर ठीक उसी जगह पे पहुंचे जो कंट्रोल करता था उस बॉडी के हिस्से को, और उसको ऑन करने से जो मूवमेंट होती थी पार्किंसन डिजीज की वो बंद हो जाती थी। तो ये पर्पस था
जी, उस तरह।
अब एक दूसरा पर्पस है जो आजकल जो काम हो रहा है वो इस वजह से हो रहा है कि अब हम डायग्नोसिस करने के लिए यूज करते हैं एमआरआई
जी,
और उसमें भी वो पार्ट यूज करते हैं जिसको कहते हैं हम लोग फंक्शनल एमआरआई।
ओके।
जिसमें हम देखते हैं कि ब्रेन जिस वक्त कोई फंक्शन हो रही है उसका कौन सा हिस्सा स्टिमुलेट हो रहा है।
ओके।
तो वो हिस्सा ब्रेन का कौन सा है वो हर एक आदमी में अलग अलग है। तो जब तक एक मैप न बन जाए स्टैंडर्ड तो पता लगाना कि ये दरअसल में कौन से ब्रेन के हिस्से को इन्वॉल्व कर रहा है वो पता नहीं लगेगा। ये दूसरी चीज है। एक तीसरी चीज और अब हमारे यहाँ खासतौर पे हिंदुस्तान में और बहुत डेवलपिंग कंट्री में और जगह भी इतना माल न्यूट्रिशन है
जी,
उससे ब्रेन डेवलप हो रहा है कि नहीं हो रहा,
ये भी चेक।
तो उसका स्टैंडर्ड क्या है। तो जब तक हमें पहले स्टैंडर्ड ना मालूम हो, स्टैंडर्ड मैप क्या हो है, तब तक हम उसे कंपेयर कैसे करेंगे कि वो ठीक है उससे कम है उससे अलग है। तो इसलिए इसकी एक तीसरी फंक्शन
है। जी जी जी। नेशनल ब्रेन रिसर्च सेंटर में आप तो हमेशा ही ब्रेन पर रिसर्च करते रहते हैं। आपने ब्रेन एटलस पर भी काम किया है। ये ब्रेन एटलस जो है ये एक्चुअली बनाया कैसे जाता है, क्या करते हैं इसके लिए?
इसके लिए आप जो सर बोला जो पहले बार आपको एमआरआई हाई क्वालिटी एमआरआई इमेज आपको बनाना कलेक्ट करना चाहिए।
अच्छा।
तो ये अभी तक इन 2-3 तरीका एमआरआई इमेज आपको कलेक्ट करना है। पहले वाला जो इमेज है जो हाई क्वालिटी इमेज होगा तो T1 वेटेड इमेज बोलता है। तो उसकी तरफ से आपको सुपर आउटर सरफेस को अच्छा से आपको इमेज मिल जाएगा।
जी जी जी।
तो दूसरा वाला चीज है T2 वेटेज इमेज है, वो ब्रेन का अंदर में जो डिटेल पिक्चर है सर जो बोला, तो डिटेल पिक्चर वहां पे आ जाएगा। तीसरा है फ्लेयर वाला एक इमेज आएगा, जो वेंट्रिकल्स है ब्रेन में उसको साइड में क्या क्या स्मॉल चीज है उसका डिटेल आ जाएगा। तो आपके ब्रेन टेम्पलेट जो और एटलस जो बोल रहे हैं आप, तो इसीलिए आपको 3 चीज का डेटा आपको होना चाहिए। उसके बाद उसको टेम्पलेट बनाना चाहिए, उसके बाद उसको वैलिडेट करना पड़ेगा। ये बहुत इंपॉर्टेंट है। तो ब्रेन साइज और बड़ा है छोटा है इसके लिए कुछ
फर्क नहीं
पड़ता है।
जी जी। आप सर ब्रेन साइज आपके हिसाब से कितना इम्पोर्टेंस है ब्रेन साइज का? आप हमें बता रहे थे कि ब्रेन के साइज को लेकर आपकी क्या राय?
मैंने जैसे बताया आपको
जी,
ब्रेन का साइज किसी हद तक इम्पोर्टेंट है
जी,
लेकिन यही एक चीज नहीं है जो ब्रेन की फंक्शन डिटरमिन करती है। इसलिए इसके ऊपर हमें ज्यादा अपना ध्यान देने की जरूरत नहीं है। तो अगर ये नहीं है तो क्या है इम्पोर्टेंट? इम्पोर्टेंट है पहले ये जाने आपके ब्रेन में है क्या।
जी।
तो अगर आप कहें तो मैं इसके बारे में
जरूर सर, जरूर।
तो एक एडल्ट ह्यूमन बीइंग का ब्रेन जो है वो तकरीबन 3 पाउंड का होता है।
जी सर।
और उसमें जो ब्रेन है उसमें सेल्स होते हैं, लाइक जैसे लिवर में लिवर सेल्स होते हैं, ब्रेन में ब्रेन सेल्स होते हैं, उसको हम लोग न्यूरॉन कहते हैं ब्रेन में। तो आपको मालूम है कि ब्रेन में कितने न्यूरॉन्स हैं?
कितने हैं सर?
30 बिलियन cells. Okay, that is important. The number of cells, neurons, that's important. और जो अभी हमने बात करी थी कॉर्टेक्स की, जो प्रभात जी ने अभी कहा था, उसी में 20 मिलियन है।
ओके।
अच्छा, अब हर न्यूरॉन में एक इसको कहते हैं डेंड्राइट्स एंड एक्सॉन्स निकलते हैं
जी,
जो उसको दूसरे सेल से कनेक्ट करते हैं।
बिल्कुल।
तो ये इसमें दिखाया है कि ये न्यूरॉन से निकल के एक्सॉन अगर उसे मसल तक जाना है तो कहाँ कहाँ उसमें, तो बीच बीच में उसमें जो एक सेल से दूसरे सेल जाने में एक जगह होती है जहाँ वो टच करता है दूसरे सेल को, उसको हम लोग कहते हैं साइनेप्स।
हाँ जी।
अगर आप साइनेप्स का जिक्र करें जो सबसे इम्पोर्टेंट है, तो आपको ताज्जुब होगा कि कितनी साइनेप्स हैं।
कितनी है सर?
मिलियन बिलियन मिलियन बिलियन।
माय गॉड।
और वो हमेशा एक नहीं रहती,
जी जी जी,
बदलती रहती है एक्शन के हिसाब से। आप काम कर रहे हैं उसके हिसाब से बनती है, बिगड़ती है। तो ये हाल है कि एक सेकंड से दूसरे सेकंड तक हमारा ब्रेन एक जैसा नहीं होता,
डायनामिक।
ओके।
और दो आइडेंटिकल ट्विन का ब्रेन एक जैसा नहीं होता।
जी जी जी।
तो इसके मतलब ये हुए कि साइज ही पे ज्यादा ध्यान न दिया जाए
जी,
जो असली काम हो रहा है वो उस पे जरा ध्यान दिया जाए।
सर जैसा आप कह रहे हैं कि ब्रेन जो है वो एक आदमी से दूसरे आदमी का, इवन ट्विन्स का भी डिफरेंट होता है, तो इस पर रिसर्च करना तो बहुत ही मुश्किल होता होगा। आप लोग कैसे ये फाइंडिंग्स निकालते हैं, कैसे रिसर्च करते हैं फिर इतने सारे डिफरेंसेस के बावजूद?
नहीं, डिफरेंस है दैट अंडरस्टैंडिंग सेम इंफॉर्मेशन आपके पास है, उसके ट्विन ब्रदर के पास है, बट एक इंटरप्रेट डिफरेंट तरफ से करता है। आप दो सिस्टर है, बट साइज के लिए मेरा है कि स्टैटिस्टिक अमेरिकन साइंटिफिक अमेरिका में एक रिसर्च हुआ, वहां पे देखा तो ब्रेन साइज के साथ उसका जो फंक्शनलिटी है जो उसका कोरिलेशन 0.3। सो दैट मीन्स only 9% brain size and the functionality. The brain आपका छोटा है doesn't mean incompetent at all.
Okay.
How आप कैसे information process कर रहे हैं,
जी,
यही important है। तो पहली बार इसीलिए पहले डिबेट हुआ that brain size matter है। It kind of in conclusive way they settle down that इसका साथ इसी तरफ से ध्यान नहीं, सर जो बोला वही कंक्लूजन है यार।
जी जी जी। जैसे ये जो ब्रेन का साइज का हम बात कर रहे हैं, जो इंटेलिजेंस है उससे भी कोई कोरिलेशन होता है?
विद इन लिमिट्स कोई रिलेशनशिप नहीं है।
अच्छा।
आइंस्टाइन का ब्रेन जब एग्जामिन किया गया,
जी जी जी,
और कॉमन लोगों का ब्रेन से एग्जामिन किया गया, तो कोई मेजर डिफरेंस नहीं था साइज में,
नहीं मिला।
लेकिन एक पार्ट था उनके ब्रेन का जिसका साइज एवरेज साइज से बड़ा था। ये इंपॉर्टेंस है मैप की। अगर आपके पास मैप होता ही नहीं, तो कैसे पता चलता कि बड़ा एक छोटा है?
जी बिल्कुल।
दूसरी चीज, एक बड़ा अच्छा इंटरेस्टिंग चीज है, जो टैक्सी ड्राइवर होता है, अब तो लगा लिए मैप वैप लगा लेते हैं, लेकिन टैक्सी ड्राइवर तो पहले अपने याददाश्त से चलते थे। तो जो टैक्सी ड्राइवर 20 साल से काम कर रहा था, उसका ब्रेन कंपेयर किया गया ऐसे लोगों का कंपेयर टू एवरेज ब्रेन। तो देयर वाज अ पार्ट ऑफ द ब्रेन व्हिच वाज नॉट द होल ब्रेन बट ओनली पार्ट ऑफ द व्हिच कंसर्न्स विद स्पेशल ओरिएंटेशन। वो पढ़ा निकला उनमें। तो हमें ये लगा कि अगर आप किसी पार्ट को यूज ज्यादा करते हैं तो वो पार्ट बढ़ जाएगा। सारा ब्रेन नहीं बढ़ा, सारा ब्रेन छोटा नहीं हुआ, लेकिन वो बढ़ गया। इसलिए मैप की जरूरत पड़ती है।
जी जी जी। बड़ी बीमारियां और उनके डायग्नोसिस पर ये जो सारा हम ब्रेन एटलस डेवलप कर रहे हैं, रिसर्च हो रहा है, इसका कितना असर होता है, क्या फायदा है?
पहले मैं क्लियर करना चाहता हूँ, जो अभी तो ब्रेन टेम्पलेट बोल रहा है, द एमएनआई टेम्पलेट बोला, वो यंग लोग को ब्रेन टेम्पलेट बनाया।
अच्छा। वहाँ
पे एवरेज एज है 32।
ओके,
ठीक है। तो इसीलिए नहीं है, इंडियन नेशनल ब्रेन रिसर्च सेंटर NBRC में एक हमारा देखना है दैट और सीनियर सिटीजन को रिलेटेड जो डिजीज है, अल्जाइमर है, पार्किंसन है, दिस डिजीज आर यंग पीपल को नहीं होता है, तो इसीलिए टेम्पलेट चाहिए, उसका एज मैच करना जरूरी है, अदरवाइज इसका कुछ यूज नहीं होता सकता। तो हमारा नेशनल ब्रेन रिसर्च सेंटर से टेम्पलेट बना दिया एक नॉर्मल एज के लिए, वो कंपेयर कर सकते हेल्दी लोग के लिए।
अच्छा,
बट दूसरा वाला चीज यही है दैट एक सीनियर सिटीजन के ब्रेन को अच्छा से इमेज करके वो टेम्पलेट काम चल रहा है, वो एक महीने में कंप्लीट हो जाएगा।
जी जी जी।
सो ये आप अल्जाइमर के लिए, पार्किंसन के लिए आप यूज कर सकते हैं।
आप डॉक्टर टंडन कैसे देखते हैं? आपने तो इतने सालों इलाज किया है इस तरह की बीमारियों का, दिमाग से जुड़ी हुई परेशानियों का, आप इस ब्रेन मैपिंग को इसमें कितना मददगार देखते हैं?
जब कोई सिम्टम होता है ब्रेन डैमेज से, तो छोटे मोटे उससे नहीं होता है, उसमें काफी डैमेज हो जाता है, तब सिम्टम आता है। तो वो वैसे ही ऑर्डिनरी MRI पे, CT स्कैन पे वो दिख जाता है।
अच्छा।
अब जैसे मैं ये आपको बताऊं, ये एक मैप है। यू कैन हैव सिमिलर मैप ऑन MRI, ओके, इमेजिंग पे, नॉट फंक्शनल MRI। मान लीजिए कि उसे स्ट्रोक हो जाता है पेशेंट को, तो ये हिस्सा जो है ब्रेन का, यहाँ पे आपको डैमेज दिख जाएगा, तो आपको पता जाएगा कितना साइज है, कितना बड़ा है कि नहीं। पेशेंट आया, उसको पैरालिसिस है, आपने मैप देखा, ये MRI किया, उसमें आपने देखा कि ये हिस्सा अफेक्टेड है कि नहीं है। अगर अफेक्टेड है तो कितना अफेक्टेड है, उसके लिए तो जरूरी है। लेकिन अगर आप स्टडी करना चाह रहे हैं कि कोई ड्रग दे रहे हैं या कोई और ट्रीटमेंट दे रहे हैं, देखना चाहते हैं कि उसके देने से ये जो डैमेज्ड एरिया वो छोटा होता है कि नहीं होता है, तो मैप की जरूरत होती है। ये हिस्सा है ब्रेन का जिससे मेमोरी डेफिसिट का।
अच्छा।
तो अगर मेमोरी आपकी खराब है, आपने MR स्कैन लिया और उसमें आपको दिखा कि ये नॉर्मल है, तो आप कहेंगे तो उम्र के हिसाब से थोड़ा बहुत कम है। और अगर ये डैमेज दिखा तो आपको मालूम है कि ये डिजीज है, बीमारी है। उसके आगे ये और भी काम करते हैं इसमें, ये तो उस मैप से कोई इन सम वे इट इज इम्पोर्टेंट, ये इसमें और उसके केमिकल्स देखते हैं अपने टेस्ट करके, ये बताएंगे
कैसे।
एक नया चीज यहाँ पे आविष्कार किया नेशनल ब्रेन रिसर्च सेंटर से, वो सारी भी इंट्रोड्यूस किया इंस्टीट्यूट के लिए, इसीलिए तो मैं चांस मिला काम करने के लिए। तो वहाँ पे एक एंटीऑक्सीडेंट है ब्रेन, वही हिपोकैम्पस बोलता है जाएगा।
ये तो दिखाया मैंने,
सर दिखाया वो हिपोकैम्पस में एंटीऑक्सीडेंट आपका ज्यादा होगा। तो वो हिपोकैम्पस का लेवल जब कम हो जाएगा तो आपका थोड़ा सा दिमाग का प्रॉब्लम शुरू हो जाएगा। बात यही है, जो दिमाग आपका प्रॉब्लम आने के पहले ही ये चेंज हो सकता है। तो हमारा यहाँ पे इंस्ट्रूमेंट है, वो वहाँ पे टेक्नोलॉजी है, आपको पता चल जाएगा, आपको बोल देंगे जो आपका इतना अमाउंट है, थोड़ा सा लाइफस्टाइल चेंज करना है, कुछ करना है, तो ये हमारा तरफ से।
लाइफस्टाइल चेंज करने से फर्क पड़ता है सर उसमें इलाज करने से? बहुत
फर्क पड़ता है। और सबसे जरूरी है एक्टिविटी करना। और हमारा एक स्लोगन है, वो ये है ब्रेन के लिए, यूज इट और लूज इट। ओके
जी, समझ गए।
तो अगर आप इसे जितना यूज करेंगे
उतना ये चलता है। बिल्कुल सही। सर, जो आम आदमी है वो ब्रेन के बारे में ज्यादा कुछ नहीं भी जानता होगा, वो एक चीज समझता है इंटेलिजेंस कोशेंट, वो उससे रिलेट करता है जनरली इंटेलिजेंस को या ब्रेन की एक्टिविटी को। टंडन सर, आप कैसे देखते हैं इंटेलिजेंस कोशेंट, IQ का?
इट्स अगेन ए इंडेक्स ऑफ हाउ within quote intelligent you are, right? But the intelligence tests themselves are biased. जिसने पढ़ा लिखा नहीं है वो भी इंटेलिजेंट होता है, लेकिन वो टेस्ट में फेल हो जाएगा।
बिल्कुल।
ओके, सो हर चीज की एक रिलेटिव इम्पोर्टेंस होती है, ये ध्यान रखना जरूरी है। नाउ इसमें कोई शक नहीं कि जिनकी IQ 130 के ऊपर है वो जरूरत से
ज्यादा।
और जिनकी 70 से नीचे है वो कमजोर है।
जी जी।
और जिसकी 30 से नीचे वो इम्बेसाइल है, वो तो बिल्कुल बेकार है, राइट। तो अगर हमें कोई स्टडी करनी है, लेट अस से एक पॉपुलेशन स्टडी करनी है, या हमें कोई किसी को एम्प्लॉय करना है आर्मी में या किसी काम में, तो उसमें देखना जरूरी है कि ये इसकी इतनी इंटेलिजेंस है कि जो काम हम इससे कराना चाहते हैं वो कर पाएगा कि नहीं कर पाएगा। उतनी इम्पोर्टेंस से ज्यादा इसको इम्पोर्टेंस नहीं देनी चाहिए।
मेरे दिमाग में एक और चीज आ रही थी, बस क्लोजिंग का ही टाइम हो रहा है। ब्रेन का एटलस, एक्चुअली जैसे हम एटलस से जो समझते हैं वो ज्योग्राफी का एटलस, ब्रेन का एटलस कैसा दिखता है? किसी ऐप के जरिए या किसी कंप्यूटर स्क्रीन पे हम उसको देख सकते हैं एग्जैक्टली कि कैसा दिखता है?
ब्रेन का एटलस कंप्यूटर में आपको एक इमेज है, उसके साथ और जगह का एक नोमेनक्लेचर है ना। आपको एक छोटा सा एग्जांपल दे, जीपीएस का एक कोऑर्डिनेट है जी। तो एक XYZ आप डोकलाम जाइए, कश्मीर जाइए, एक कोऑर्डिनेट है। हाँ। तो ब्रेन का भी और जगह का XYZ कोऑर्डिनेट है, उसका अलग अलग नाम होता है। जी जी जी। ठीक है ना। हाँ। तो यही है एटलस। अच्छा होगा तो इसको वैलिडेशन नहीं होगा तो इसका कुछ काम नहीं है। तो वैलिडेशन फंक्शनल एमआरआई के तरफ से करना है। एक छोटा सा एग्जांपल देके आपको बोल रहा है ये डोकलाम का और कश्मीर का बारमूला का जाएगा का कोऑर्डिनेट है, आप सीधा उसको फॉलो करके वहां पे पहुंच सकते हैं। ब्रेन का इसी तरफ से हो सकता है। आपको अच्छा से टेम्पलेट किया, टेस्ट किया, वैलिडेट किया तो कोऑर्डिनेट बोल देंगे, तो एक रिस्पेक्ट सर्जन जो ब्रेन का असली काम कर रहा है तो उसको भी पता चल जाए कहाँ तक जाना है, वहां पे वो जहाँ के जाए रीच करके बोले यही जाएगा है। इतना कॉन्फिडेंस होना चाहिए।
बिना ब्रेन को खोले हुए,
बिना ब्रेन को खोले हम उस जगह तक पहुंच सकते हैं जहाँ कुछ
वहां पे आप इलेक्ट्रोड डाल सकते हैं। उससे चाहे वहां पे स्टिमुलेट करें, चाहे उसे डिस्ट्रॉय करें, चाहे उसे रिकॉर्ड करें।
ओके, ये चीज की जाए।
ये हमारा नेशनल ब्रेन रिसर्च से ब्रह्मा एक टेम्पलेट बना दिया। तो यहाँ के एक चीज है, आपको डीप ब्रेन सर्च जो बोला अंदर में वो जगह को सब्सटेंसिया नायग्रा जो पार्किंसन डिजीज में बहुत इन्फॉर्म होता है, वो आपको एग्जैक्टली पता चल जाएगा। इसीलिए उसको मैं उस सबको फ्री में एकेडमिक इंस्टीट्यूट में दे सकते हैं, उसको देने की कोशिश कर रहा हूँ।
बहुत अच्छा, इस तरह के रिसर्चेस आगे आप करते रहें और इससे बहुत सारी और जानकारी हमें मिलते रहे, इसके लिए आपको शुभकामनाएं। आप दोनों का ही बहुत बहुत शुक्रिया हमारे स्टूडियो में आने के लिए और हमें इतनी सारी अच्छी अच्छी जानकारियां देने के लिए। आपको हमारा कार्यक्रम कैसा लगा? आप अपनी राय और सुझाव हमें नीचे दिए गए ईमेल आईडी पर भेज सकते हैं। तब तक के लिए हमें दें इजाजत। नमस्कार।
Over to you, Jaswinder.
All right. So can you see now? Professor Mandal, can you see the slide?
He's not audible.
Okay, let me just call him.
Can you hear me now?
Yes, sir.
Yes, sir, we can hear you. Yes, sir, we can hear you.
Okay. So can I start giving a remaining portion of my talk?
Yes, sir, sure.
Okay. So that brain is a very certain place of the brain is fixed, like ACC, PCC, that main central line. So that's what when somebody is making the brain template, this is the two focal points are always considered. I have some example because Jaswinder was telling that it will go out of the way, like in a Connaught Place there is a post office and there is a medical college. That's the, you can correlate this one that this is the two places are close by, so you can orient. They are not changing and they are prime areas and they are always there in Delhi. Similarly, in the brain, everybody's brain, these two ACC and these two regions are all fixed. So you can orient or you can say put a central point based on this points. So everybody's brain, these points are fixed. So that was taken as a leveling purpose, as a standardization purpose. So in entire cases, brain template is being developed based on these particular points. Next slide, please. I'm talking about some technicalities so that you can see that one. You take a brain template of same places, then you have to do some twisting, particular angulation, shifting, and then it will become particular a angle, you shift it and it will be in a correct shape. So this is being done in the computer programs, and you don't have to do anything. What you need to do, you need to collect very high quality T1 image. I was speaking that time, the T1 weighted and T2 weighted and flare. This has to be there, but already everything is programmed, and you need to get that data to, so the program can work it out. Thank you. Next one, please. Yes. So you can see that one, that whenever many challenges come still, any MRI images need to be very much quality control, because you are making a template for the clinical use. So the MRI quality of the images will be very high quality, and that's because it is surgery is being done. So it has to be very correct. So that's what many images, there are it gone through huge amount of quality control. So this we generally take it very seriously. So although we may have a 200 images, but we may not get the 200 images for template preparation. We may be 150 or 160 images can be done, because they have some distortion, some movement. So that's what it has to be. The reason I'm saying is that it is very serious matter. We lot of precaution has to be taken into consideration. Next one, please. Yes. So this is one to show you that how the different types of templates are developed throughout the world. But I have this because this is the way the students can see that although MNI template 305, that one, this is 1992 and then ultimately 2001, this is being popular throughout the world. The rest of the template are never validated except Chinese, that is also not clearly written what they have validated. They claim validation, because any template you prepare, you have to have a functional MRI data. That's what I was telling you, that these functional MRI data, until unless you validated it, it has no use. So IBT actually has accomplished that Indian brain template. Brahma has accomplished that part. Next slide, please. Yes. So what we did is that it is a pan-India subject. People from all over the world, India, they came and participated from Nagaland, Assam, Kashmir, Gujarat, Bengal, Andhra, everywhere. So that's what we got a very nice response also, because the Hindu coverage, many people volunteered and they said we want to come and join in this program. So that's what this we could be achieving this much success. Okay, next one, please. Yes. So these are all different parameters. We got it. And so these you can see that what are the characteristics features. These may not be very relevant to you, but these are all quality marker for the MRI. The T1 weighted images we collect, T2 weighted images we collect, pair images we collect. These are all marker. This is actually quality control. Next one, please. Yes. See, this is the main contribution of ours. We have made this construction pipeline. So what we have is, this is, I want to spend this on. This is, we try to make it such a way that a common person doesn't have to depend on anything. They have to give the correct images, and then automatically the program will check the quality control of the images, and they also go for the ultimately the entire the template will also be collected. It will take about in a high-speed computer we need it, but it will be done in 100 hours of computing time. So this is be required once you need to have all this information in place. Once you do it, and then you don't need to get anything involved at all. So this is our creation, that not only we created our MRI images, very high quality. We also have our programs, the Brahma pipeline, that is actually generate the template, and that's what we could do. Next one, please. Yes. So you can see that one. This is the template T1 it looks like in 3 different orientation. How they looks like. They are different images, so they are picture color are differences. T1, T2, and player. Next one, please. Yeah. So this is just a tissue probability map. This is also, you get it from there. It is very important because your brain has a lot of gray matter, white matter, and CSF. CSF takes in the ventricles, but these are all and deep green matter. So these can be actually, when you are making the template, the tissue probability map is also very critical, because this is why you understand the content of gray matter, white matter of these persons. Next one, please. Yeah. So here we are similarly, we are talking all these cases that you have to label them. That is very important. Every parts of the template of Indian brain template Brahma has to be labeled. And that labeled, suppose somebody else is doing fMRI study or any other study from other places, they will take our images, and when they fuse in their images, they will find out what is the labels. So we are providing the labels of entire brain. It's a big project, so maybe not be in the beginning all parts of the brain cannot be labeled, but lot of places we have labeled them, so that will be useful for a normal use, for our research purpose or clinical use. So next one, please. Yeah. So this is a tissue probability. This is a niche piece of work that you can see that one. There is a atlases we have generated compatible with Brahma. So we are actually telling people that look, this is why these are the atlases available, and our Brahma is actually compatible with them. You can use it. So basically, this has opened up a instead of depending on American brain, now we can use our structures. We can do a comparative analysis from different continent, and we can see the differences. So this is why it is very useful. Next one, please. Yeah. Now you can go to the next one. I want to show the validation of the yes. Next one. Next one. Yes. So this is suppose you do a motor cortex experiment, and now that means you move your hand when you are inside the finger tapping, or move your hand, press your hand, then motor cortex area will be activated. Now if we do a left hand, the right side will be activated. If you right hand, then left will be activated. If you do the both side will be activated. So we compare our work with existing Brahma template and also MNI template, which is there in the case of in other software packages they have it. So we want to see that what is the difference between our template and if an MNI template available. So there are some activation pattern wise, there are some differences, but it may not be visible in one group. So next slide, if you go, then you will see that one. Yes. So we took a 10 subject, and there are some differences that when you use the Indian template, then you get a better activation, because you are comparing Indian brain with the Indian study with the Indian brain template, and that is actually very good. So that is logical, but previously it was not known. So this is a big confidence and satisfaction that we have achieved something that. So this is there. Now next, this is very important because without this validation, this has no value of any mentor. Among these, there are many people every country has made a India template, but they did not do any validation on that one. So, and that's what they are not. So template construction, we made it completely a GUI, graphical user interface wise. It is very easy to use, and that is generally a scheme is available. So we are actually planning to have some training program also later on, so that people can make use of this facility. Okay, so next one, please. Yeah, so we have made a comparative analysis. Our in the case of Indian template, whether IBM, MNI, and you can see there is a comparative analysis has been made. What is the real differences between this one? There is some amount of changes, but the problem is that when you talk about the activation pattern in functional MRI, they are actually changing. So this is very, very important. So this is, we need to observe it, and that is coming because Indian brain template, we are able to use it. Next one, please. Yes, this is a most important part of the talk because you need people and their contribution and their motivation. Like so many of bright students from Gargi is listening to us, and so these are the people, they actually motivate and they contribute, and then we make some new discovery. And this is why I want to always highlight the co-workers who are actually a very important part of this research program. And I thank Dr. Jasvinder for giving me the opportunity, and this is a fantastic program you did. And with this, I'll stop here. Thank you very much.
Thank you, sir. We'll take up a few questions, sir.
Yeah, sure.
So the first question is regarding the comparison of the Indian brain template with the American and Canadian brain templates. What is the major difference?
There is the size. American brains are generally bigger in. I have a transparency on that last slide. It is there, comparative analysis. I made it on that one. So that slide, I cannot see it here. So there is
in general, how will you explain it?
Generally, the ACPC size, I was telling in the long run that axis, that is little bit bigger, and American brains are generally much bigger in volume, generally 1400 to 1400 cc or 1350 cc. Other cases, we do not have. Yeah, so the volume is much higher in American brain than the Indian brain. Actually, that is not a Canadian template. That is a Canadian template, not American template. There's a Canadian. Actually, Montreal Neurological Institute. They developed that template.
Thank you, sir. So this is
okay.
There the role of nutrition was discussed, right? That it impacts the brain. So how does functional MRI help you to understand that impact of malnutrition on brain?
Can you repeat your question, please?
Yes, sir. This is regarding the video that was shared by you. It was discussed that malnutrition has an impact on brain, and it was studied through, I think, fMRI. So can you just expand a little and explain to us how?
Okay. Suppose fMRI is a technique that suppose your malnutrition means your brain did not develop correctly, right? So, and then there are different types of task is available. That task actually, when a baby is born, maybe baby is 8 months, 8-year-old or 12-year-old. So there are certain amount of task you are supposed to do by normal means. So when this, due to malnutrition, there is a deficiency in the brain. So when you perform that fMRI activities on this kid, so they will not be able to respond correctly, and you will not see any activation in the particular brain areas of that person. So this is why functional MRI is actually a technique where if you are active, you will be asked to do particular questions, or you ask me to answer that. You suppose you are deficient, you will not be able to answer. So in that case, your particular brain areas will be not activated. So it will be like a dark spot. So by functional MRI, you can find it out that baby has a problem of because of malnutrition. So these different places, depending on the extent of malnutrition, this could be very complex. I hope I am able to answer this question, because it is not a very simple question, but it is the way it can be addressed like that. There are a specific task can be there, but but
Next
yeah.
question. How do you see the applications of neuroscience to
Excuse me. Can I please interrupt? Excuse me. Professor Mandal, can you please check the audio on your end? Is it on speaker? There is an echo actually.
Okay.
So you can please unmute yourself and use a microphone if possible. You're not audible now. Can you please use a microphone? Hello?
Can you hear me now?
Yes, sir.
So this question is related to explaining to non-neuroscience people the role of how the application of brain mapping works.
How the role of
brain mapping
brain
works?
map. Okay. Brain mapping, I don't know. Who is this? When brain mapping that is not for brain mapping is required for your two things. First of all, either in a healthy conditions or in a clinical condition. So you need to know some information. Suppose without that, how can it, brain is there within and it is going all right, but we should know that what is inside in our body, inside on our head. So the mapping is mean I need to know the but please note that that all the mapping MRI, fMRI, and MRAs are non-invasive. They don't need any radiation or any blood test, so that you can get in and you will get a nice picture of your brain. So, but without the pictures, I don't know how we should have everybody's brain to be mapped, because at some point or other you need it in your lifespan, because that time a comparative analysis has to be done. It is not aware because of the expenses or awareness, but I think many people are doing nowadays that some mapping of the MRI images in different form should be there. I hope that clarify that one.
Yes, sir. So the question is how can I
Can you repeat? I can't.
How can I access this template, the brain template?
Mail, you can send an email and then you can access it. We have website also, Brahma, the Indian brain template. We have a website and you can send a drop a mail and we will respond to you.
You have said that Americans have a larger brain capacity. Why do you think that? Is it
What is that?
Is it evolutionary that the Americans have a larger brain size capacity?
This is about a complicated thing. I will not comment because evolutionary things also there. I cannot comment on that one. But they are generally in Germans also has a very huge brain size. Germans also, and they are Aryan. Our civilization they consider that they are Aryan. We are also Aryan in some cases of thing. But I am not very competent enough to comment more than that at this point.
Okay, we'll take the last question. Does neuroplasticity vary according to the size of brain?
Neuroplasticity?
Vary according to the size of the brain?
No. Neuroplasticity is not very according to the size of the brain, no. This is an intrinsic property. It doesn't matter of the your brain size. The answer is no.
Okay. Thank you so much, sir. I would like to vote of thanks. Thank you so much for that captivating talk with details about the brain template. Your focus on this new Indian brain template called Brahma, which you say has been generated using 3D MRI scanners, taking a lot of intense brain mapping and studies across India, is truly fascinating, particularly since it is the Indian brain template. You're sharing that the striking feature of the template is that it is validated from functional MRI experiments using finger tapping and working memory and is completely India centric is really a proud moment for all of us. We would like to acknowledge our gratitude to you for taking time out from your busy schedule and sharing all your expertise with us, for patiently answering questions in spite of lots of technological glitches, for which we do apologize. We are indeed indebted to you for spending time with us. Thank you so much to your answering of the questions, sir. I would like to thank the participants as well for participating interactively in asking all these questions. I would now like to hand over to my colleague, Dr. Udita Mukherjee. Thank you so much.
Hello, everyone. I am sorry to inform you that Professor Roop Lal, who was scheduled to speak to us next, wouldn't be able to join us today due to some unforeseen emergency that needs his undivided attention. We'll, however, take an interesting turn, and it is time for the brain teasers. We'll be presenting 2 brain teasers, and the correct answer for which will have a cash prize each. The rule is simple. The first person to email us the correct answer wins. Please do not forget to mention your names, your affiliation, and your mobile number. Please send in your entries through email at neuroeunoia2020@gmail.com. As already mentioned, we have prizes for the first correct entry that we receive. The prizes are sponsored by the Indian Academy of Neurosciences. So good luck to all the participants, and here we go. I request the participants to kindly take a screenshot and go through the instructions. Here we go on to the first brain teaser. Good luck. We'll now move on to the second brain teaser. Good luck to the participants. So that will be it. I hope all the participants have seen both the brain teasers. Thank you for your responses, dear participants. We'll announce the winners of the brain teasers tomorrow during the valedictory session. As we all are aware, we invited poster presentations for this virtual conference. We would like to thank all of you who showed such keen interest and submitted their entries. It is imperative that we let you know that we asked our eminent speakers and associates to evaluate the entries for poster presentation, and we take this opportunity for thanking them wholeheartedly for their immense contribution. I thank Professor Suman Jain, Neurophysiology Laboratory, Department of Physiology, AIIMS, New Delhi. Dr. Arun Sasidharan, Center for Consciousness Studies, Neurophysiology, NIMHANS, Bangalore. Dr. Neera Pant, Department of Psychology, Gargi College, University of Delhi. Dr. Elavar C.A., Department of Neurology, AIIMS, New Delhi. We thank you wholeheartedly for your contribution. However, due to paucity of time and unprecedented numbers of entries we received, we will not be able to show you all the posters and would only be able to display the winning posters and present the winners' videos. To all the best poster awardees, we'll be sending you the best poster certificates soon after the conference. The winners of the best poster awards are, it is in no particular order, I must mention that. So the winners are Rahul Kumar, Department of Zoology, Shiodini Sao College, Bihar. His topic was prevention of optic nerve atrophy by treating early glaucoma using nanoformulation with promising therapeutic potential. Next in line is Manisha Singh from Department of Biotechnology, JP Institute of Information Technology, Uttar Pradesh. Her poster was on the topic, development of aposinin-loaded polymeric nanoparticles to target oxidative stress at ROS modulator for neurodegenerative disorders. Next in line is Vinayak Agarwal from Department of Biotechnology, JP Institute of Information Technology, Uttar Pradesh. His topic was in silico validation and development of chlorogenic acid-loaded polymeric nanoparticle for targeting neurodegenerative disorders. Next, we have Ms. Gauri Jadhav, Department of Biotechnology from Savitribhai Phule, Pune University. Her topic was gut dysbiosis after repeated mild traumatic brain injury cause anxiety-like behavior and HDAC2 mediated BDNF expression in the amygdala of Vista rats. Next, we have Priya Mishra from Amity University, from the Amity Institute of Neuropsychology, Amity University, Uttar Pradesh. Her topic was anti-epileptogenic cicuta virosa prevents cognition, memory, and motor coordination impairment through downregulation of oxidative stress load and upregulation of SIRT1 PGCA alpha pathway in PTZ-induced kindling mice model of epilepsy. Next, we have Nabila Majid from Interdisciplinary Biotechnology Unit, Aligarh Muslim University, Uttar Pradesh. Her topic was biophysical elucidation of amyloid fibrillation inhibition and prevention of secondary nucleation by cholic acid and unexplored function of cholic acid. Next, we have Nagalakshmi Balasubramanian from Department of Biotechnology, Savitribai Phule, Pune University. Her topic was repeated mild traumatic brain injury perturbs the mitochondrial biogenesis via nuclear and mitochondrial DNA methylation. Next, we have Ms. Ishi Tandon from Amity University, Rajasthan. Her topic was depleted grey matter volume in REM sleep behavior disorder in Parkinson's, a voxel-based morphometry study. Next, we have Pranshi Agarwal from Gargi College, University of Delhi. Her topic was integrative genomic analysis of schizophrenia in the basal ganglia and the frontal lobe. Next in line, we have Dr. Ashlesh Patil from the Department of Physiology, All India Institute of Medical Sciences, Nagpur. His topic was Sudoku puzzle differentially activates prefrontal cortex, an FNIRS study. Next, we have Dr. M.G. Ramesh Babu from Melaka Manipal Medical College, Manipal. His topic is Raj Yoga meditation induces grey matter volume changes in regions that process reward and happiness. Next, we have Mr. Amit Bhatt from the Department of Zoology, Sri Venkateswara College, University of Delhi. His topic was CG dinucleotide and their role in pathogenicity of coronaviruses. Next, we have Pratibha Maria D. Almeida from Department of Physiology from Melaka Manipal Medical College, Manipal Academy of Higher Education, Manipal. Her topic was effect of Clitora ternatia aqueous root extract and choline with DHA supplementation on brain oxidative stress in perinatal maternal separated rats at different time points of aging, a comparative study. And last but not the least, we have Lydia Shobha Andrade from Department of Anatomy, Kasturba Medical College, Manipal. Her topic was a study on the variants of phrenic nerve roots with histological correlation. Congratulations to all the winners. We will now be presenting the winners of the posters. The next poster we have is from Dr. Ashlesh Patil. Next, we have the poster from Mr. Rahul Kumar. Next, we have the poster from Ms. Gauri Jadhav. Next is from Ms. Priya Mishra from Amity University. Next, we have the poster from Ms. Nabila Majid. Next is this poster from Ms. Manisha Singh. Next in line, we have the poster from Vinayak Agarwal. I will be displaying the posters again for those whose name I could not take due to some connectivity issues. So here we go. This is the poster from Miss Lydia Shobha Andrade. Next, we have the poster from Mr. Amit Bhatt. Now we have the poster from Ms. Pratibha Maria D. Almeida. Next is this poster from Ms. Nagalakshmi Balasubramanian. Next, we have this poster from Ms. Pranshi Agrawal. Next, we have the poster from Ms. Ishi Tandon. Next, we have this poster from Ramesh Babu MG. I'll stop here because from here we already know the names of the winners. Thank you for your patience, all the participants, and congratulations once again to all the Best Poster Award winners. Now, out of all these 14 Best Poster winners, 4 posters have been selected for the cash prize by the judges. Before we announce the names, I must once again thank the Indian Academy of Neurosciences for being so generous and sponsoring all the prizes. We truly cannot thank you enough for your benevolence. We have cash prizes under 4 categories: neural disorders, treatment of neural disorders, health sciences and neurobiology, and neuropsychology. From the category of health sciences and neurobiology, the cash prize goes to Ms. Lydia Shobha Andade from Department of Anatomy, Kasturba Medical College, Manipal, Manipal Academy of Higher Education. The next prize is from the category of treatment of neural disorders, and the prize goes to Mr. Rahul Kumar from the Department of Zoology, Shiodeni Sao College, Magad University, Bihar. Next in line, we have the prize from the category neural disorders, and it goes to Ms. Nagalakshmi Balasubramanian from Savitri Bhai Phule Pune University. And last but not the least, the final cash prize.
The chat box, and we shall be taking the questions once the talk is over. So it's my pleasure to introduce to all of you to our next speaker, Dr. Arun Sasidharan, who is a neuroscientist trained in modern medicine and neurophysiology at the New Center for Consciousness Studies, NIMHANS, Bangalore. He has over 10 years of expertise in terms of acquisition across various EEG-based techniques. He has been involved in the research and development of novel EEG devices, neuromodulation techniques, EEG-based signal processing algorithms. He has contributed to more than 200 sleep recordings and has contributed immensely to development of novel sleep analysis parameters, innovative techniques to assess sleep stability, robust sleep oscillation dynamics, etc. He has published around 18 papers in international journals and has made over 45 conference presentations. I welcome you, Dr. Arun. Over to you, sir.
Dr. Arun, please unmute yourself.
At the outset, I would like to thank the organizers for giving me this opportunity. I think it's slightly rare for some of us scientists to speak to audiences from various backgrounds. So I think one of my primary aim is to reduce the amount of jargons in this particular topic. So I think without delay, I'll start my screen. Let me know if the screen is visible.
Yes, sir.
Is the screen visible?
Yes, we can see that. Yes.
Yeah. Am I audible now?
Yes, sir.
Yeah. Okay. So thank you. I hope everyone had a good lunch. And my topic is sleep, cognition, and consciousness after a lunch period. It's a topic that many people would like to explore more, but it's an extremely vast area. Several people work around this, so it's primarily an interdisciplinary area. You can't have a person from a single background to cover various aspects of sleep. So from zoology, from wildlife to insect, from medicine to engineering, almost every field contributes to sleep. So my presentation will also be oriented in that line. I would like to inspire people from varying backgrounds to explore sleep as part of their research or exploration. So before I start the core part of my talk, I would like to give you some inputs before you all go to sleep. Some awareness tips related to sleep. So this is one aspect that many people ask, especially when they try to contact me or anyone related to sleep. Are we all sleep deprived? If you ask people, that means through surveys. There have been surveys by health device makers, etc. So Philips had done a survey, first of its kind in 2015. They found that 93% of the Indians tend to say that they are sleep deprived. And what's the criteria? Duration. So less than 8 hours of sleep. And the thing is, though they know this is a problem, or they feel that this is a problem, only 2% of the people tend to visit a physician for this problem. Because we know we go to doctors for many other ailments, but if we find that we have a problem with respect to sleep, we don't go. The second part is there is another fitness company, at least Fitbit you would have heard of. They conducted a survey across 18 countries across globe, and they tried to rank different countries based on the average sleep duration. So here you can clearly see some of the Western countries tend to have average sleep more than 7 hours, and many of the Asian countries, including India, tend to show a very low average sleep duration pattern. Now, this is one thing we have to keep in mind, because in the previous slide, in this 93% case, we are putting a hard threshold for the sleep duration, whereas across globe, there tend to be differences in average sleep duration. So according to this study, Indians sleep less or poor, but that probably need to be revisited. And I hope during my talk, I would try to convince that. Now, another survey, slightly more recent, they looked into Indian population alone across multiple metro cities. So they tend to find that 53% of the people say anyway they have sleep problems, and they attribute it to gadget use and social media at odd times, and because of which the sleep onset is delayed. So there is kind of awareness among people. At the same time, there is some amount of information which is either wrongly placed or inappropriately shared. Now, if you ask experts, so they also, there are multiple experts, as I said, multiple fields. So one group called American Academy of Sleep Medicine, they are the body which since 2014, they have been giving the guidelines on all sleep-related studies and clinics. So they try to combine multiple sleep experts together, and their primary aim was to find out what is the best sleep duration where you have good health. So looking at general health, cardiovascular health, and many medical parameters, they try to find out which would be the ideal duration. So if you look at this red trace, you can't sleep more. Most of the people can't sleep more than a particular hour of sleep, whereas several people can sleep short. So this red trace just indicates what is the probability of us going into sleep. And this green one is with respect to this medical part. So they found that if you sleep less, poor medical health. If you sleep too much, poor medical health. If you're somewhere in the middle, you have good health. And this part of the color graph shows you what is the consensus among all these experts. And they found 7 to 9 hours would be an ideal period. And there are other groups who don't go with this concept. They actually studied multiple people. And this is claimed to be the largest sleep study because they collected over 40,000 people. And what they primarily look for is they were doing a cognitive test on all these 40,000 people. And they found that people who reported to have slept 7 to 8 hours for weeks together, they tend to perform better compared to the other time period. So the consensus is again back to the 7 to 8 or 7 to 9 hour sleep pattern. Now, what the aspect of sleep that I mentioned now is all to do with sleep, with the duration of sleep. So whether if 7 to 8 hours is the right criteria, you sleep less, you have a problem. And there is actually a booklet, which is the guideline for all the sleep disorders. And they found, I don't know if you know, if many of you might not even know, there are about 81 documented major sleep disorders for which there is a clear-cut diagnosis. And these disorders are actually grouped into 6 major categories, of which the first one we described, which is the duration. So you sleep less than what is recommended, then you are insomnia, with other features like you can't perform well during daytime. Even if you have the time and leisure to sleep, you can't get sleep. Those things. Another disorder under this categorization is called sleep-related breathing disorders. This is now the major sleep issue which has been propagated across the globe, which is called sleep-related breathing disorders. A common thing is called obstructive sleep apnea. Means you snore. So you snore and someone else get disturbed. That is not a disorder. But if you snore and because of that snoring, you have to put in that extra effort to breathe, which means you will not be able to stay in deep sleep. That will affect your sleep badly. You'll have to wake up multiple times unknowingly. And that is a problem. And that comes under this second category. Third category is too much of sleep. And usually too much of sleep is related to some problem, some inflammation in your brain. And that is termed as central disorders of hypersomnolence. Another category is circadian rhythm. That means we have a rhythm of when to sleep and when to wake. Now, if that gets altered, either because of our habit, our job, shift working, etc., or you travel from one part of the world to another part, you have jet lag. So they all come under this category. And in fact, especially in this adolescent period, in the college group, you tend to push your sleep towards early morning, like 2 a.m., etc., and try to wake up quite late. That in the initial part, it doesn't cause a problem. But 2-3 years down the line, you may end up having a circadian rhythm disorder, which might require some medical help. Fifth category is called parasomnia. Parasomnia basically means you walk, you do abnormal behaviors which are not expected during sleep. For example, you walk during sleep, not expected. You kick someone else during sleep, not expected. You talk and babble during sleep, not very much expected. So those all come under parasomnia. And it's considered disorder only if it affects your day-to-day activity. Now, instead of a complex behavior like walking, if it's only tiny muscle movements like moving your or chin, clenching your teeth during sleep, etc., they are called sleep-related movement disorder. And again, if it affects your sleep and comes under this disorder. Now, any other thing which doesn't affect your day-to-day activities, whether it is snoring, whether you sleep too much, or talk during sleep, they are all considered as isolated symptoms and apparently normal variants. at least for now, because this book is currently 3rd edition, and every edition disorder number has been going up. So I'm sure that in the next edition, some of the isolated symptoms might get categorized into one category. So I didn't mean to frighten everyone with a lot of sleep disorders that are there. I just wanted to send this message that sleep is a large thing and there are several issues which are not well known. So with this, let me try to describe what I would like to convey. I have 2 things in my mind. The first part is to look at the sleep, its fundamental aspect from a neuroscience point of view. And the second part is to show you how sleep and wake are related and need not be separated out as 2 separate entities. Whatever you learn about wake can sometimes partially at least be applied to sleep and vice versa. So let's start with the sleep fundamentals. What is sleep? So this is a question which has been haunting many. But there is one definition over the last 10 years. I've come across multiple definitions. I found this to be a wonderful one because one, it addresses the nature, it addresses all species, all type of sleep, not just our culturally relevant sleep. So the definition goes like this: sleep is a rapidly reversible. So that means you should be able to go into sleep and come out of sleep. So if it is not reversible, it's like coma. It's a rapidly reversible state of reduced responsiveness, reduced motor activity, and reduced metabolism. So you are awake, you will be responding to sound, you will be responding to somebody's call. But when you are asleep, that responsiveness comes down. You may not be speaking. Motor activity, you anyway have a tendency to move around when you are awake, but that will come down during sleep. And your metabolic activity, your bodily activities, many of them come down. So these features, because for a human, you can go and ask whether you got to sleep or not. But if you want to study the same thing in an infant, if you want to study the same thing in someone who can't speak, in case of animals, etc., this is a definition which fits all of them. Now, accordingly, for us currently, whenever we say good sleep, this is a picture that comes into our mind: an isolated, cozy environment, relatively safe. We tend to say that we need to sleep from say 10 p.m. to 7 a.m., something like that, as a single bout of sleep. But if you look across society, there are various varieties of sleep. There are people who sleep on very minimalistic conditions where we can't sleep probably. In Indian railways and all, you can see them sleeping in much worse conditions than this. In PGs and in many camps, etc., people sleep in cramped together. Some people, especially in Mediterranean countries, they tend to sleep during noon. And there, probably the people on the street can even sleep during the noon sun. So these are things which we typically don't expect from a good sleep, but they are all doing their day-to-day activities well. So we need to look at the society, culture, etc., while defining sleep. Because in all these conditions, irrespective of where they are, their state is rapidly reversible. They have reduced responsiveness. Their motor activity is low. Their bodily metabolism will also be lower than waking sleep. So they are actually in sleep, and in fact, proper sleep. Now, if you look across species, also you tend to gain insight. For example, dogs and cats curl up and then sleep, not like us in bed position. There are animals like horses and large birds like cranes who stand and sleep. There are birds, seals, dolphins, etc., who can continue their swimming activity despite sleeping. Bats, which can turn upside down and sleep. Leopards in extremely odd position on tree branches, maybe monkeys also. And we tend to sleep in multiple postures, but yet we sometimes can't mimic some of these animal sleep habits. Now, to understand sleep, we understood the definition and the fact that just lying on a bed doesn't mean sleep. We also need to know what happens during sleep. What are its major functions? Because that will also help us determine whether sleep is good. So one core aspect is balance. Sleep allows the whole system, the whole body to go into a proper balance. If you look at blood parameters, etc., you will see one of the hormones related to stress called cortisol tend to reduce and become almost low in the first half of the sleep and start increasing in the second half. And once it reaches the peak, that is when we normally wake up. Whereas another hormone called melatonin is otherwise called sleep hormone, and they are extremely low when we are awake, and immediately when we start planning to sleep, around that period when sun goes down and lights are off, this hormone continues to rise, and your deeper sleep tends to happen when the hormone is at its peak. And besides the hormone also influences your entire metabolism and body temperature, called core body temperature, that tends to dip from the time you go into bed, and it kind of reaches the lowest part in the early morning period when you are in your dreaming period. Okay, so they have to get aligned, and the fact that this rise and drop always happen during sleep clearly means that there is something specifically happening during sleep which requires this change to happen. And in fact, any of these changes, if they are tilted, they seriously affect our health. And it's not just few hormones. In fact, the entire organ system have a cycle and rhythm, and they tend to do differing activities during wake versus during sleep. For example, the fat tissue during wake, it tries to accumulate fat, and during sleep, its primary role is to break down fat. And that explains, if you don't get proper sleep, you tend to become obese. And besides the other organs, you can also look at heart. Heart is severely impacted by sleep. If you look at BP, for example, blood pressure, usually we have somewhere around 120, 140 systolic and diastolic around 60 to 90. So these red and blue traces represent them. And usually from awake, it starts dipping during your sleep. It will be very low. So your normal, when you say your normal BP, that normal is different for wake and different for sleep. This is one thing we have to keep in mind. And if it doesn't reach that low level and it stays in the same level as the wake, it's actually a problem. It's a sign of heart disease. And similarly, your heart rate, if you look at during sleep, it tends to come down. And in fact, it has a trend of this dip, which is called hammock. And this is a sign that is required for proper health. So if it doesn't have this dip or this trend, that means your heart is not healthy. And sleep, multiple hormonal changes and other things that happen during sleep helps in this regard. And because of this, people have found that healthy people who sleep 6 hours or more have no major impact on cardiac arrest and death later. But if you already have some amount of hypertension or cardiac disease or stroke, and then you tend to sleep, you tend to have much higher risk compared to a person who sleeps more. So if you're already having a vulnerable heart, it will further aggravate the condition. So sleep is definitely required as part of a cardiac health treatment. Immunity is something that many people do not know is attributable to sleep. In fact, many of the immune system, because immune system also needs some memory and recouping each time it gets exposed to a virus or infection. It does some recouping during the deep sleep. So deep sleep amount corresponds to the immune system's activity. So it will be calm if you have the right amount of deep sleep. Otherwise, it will be in a slightly hyper responsive state. Because of this, many studies have found that people who tend to sleep more tend to catch viral infections like flu much less compared to those who sleep less. So this might be of interest to people who now that the COVID pandemic is around, sleep has a protective effect. Now, the last function that I would like to describe is deep cleaning part. So this is a function that has been recently found. Most people might not be aware of that in brain, we have a special kind of blood supply. The blood directly do not touch the brain tissue. Instead, we have another fluid, a filtered fluid called CSF, cerebrospinal fluid. And these fluids tend to be much more spread. For example, in this MRI image, you can see this red patches. So these patches represent that the fluid is going into the surrounding of the brain tissue. So during your deep sleep, this happens very prominently, whereas during wake, this kind of rush of fluid doesn't happen. And what is the function? People have found that during this rush of fluid, during deep sleep, it can clear off many of the debris which are found. So these debris, because of nerves being active, their byproducts get accumulated in the surrounding. They will not get cleared off during the waking period fluid flow. They get properly washed off only during your deep sleep. So you tend to have less deep sleep over a period of several years. These debris get accumulated, and now it has been one of the cause for conditions like dementia, where these accumulated proteins cause problem to nerves. Okay, now that you have understood that sleep duration has to be looked into from multiple angles, we also need to look at sleep function. We should also look at other factors and factors which affect sleep. So one major factor is our body clock, and this is called circadian rhythm. So we tend to be awake during wake when lights are on. We tend to be asleep when it is night. Upon that, your activities during daytime will also increase the sleep pressure. You work heavily during wake, you will get much more sleep. You have minimal physical activity during wake, you get less sleep. Now there can be misalignment. You have your clock at a particular time, but your physical activities are, say, during night time. That will affect your sleep. Similarly, if you travel to different countries, your work schedules are different, your body rhythm or the circadian rhythm gets misaligned to your day-to-day activities. That can also cause a problem. Now, besides these two factors, there are other factors too. For example, if you're erect, if you're standing, it's very tough to sleep unless like you have a stand like this lady has. And in extreme environment, it is very tough to sleep. Whereas if it's a calm environment, you have a bed to lie down, you'll always get better sleep. So that is over and above these two factors. Now, another important factor is health. If you have fever, you would have noticed that you tend to sleep more, especially in your childhood period. Your diet, if you take fruits, vegetables, less cooked fruits and vegetables, you tend to get sleep. If you tend to take fatty foods, cooked foods, etc., more often hot food, you tend to sleep much more. So diet can have an impact on the sleep duration. There is a lot of studies on individual differences, especially with respect to genetics. So people now discriminate sleepers into normal sleepers, long sleepers, and short sleepers. So the short sleepers will be anyone who sleeps less than 5 to 6 hours, and long sleepers will be anyone who sleeps more than 9 to 10 hours. So it is found that across generation, a person who is already a short sleeper, his next generation, they will all be short sleepers mostly, because they pass on a particular gene, and it is now they have identified several genes which are specifically related to short sleeping and long sleeping habits. So we need to identify them because they will be normal. It is part of their genetic makeup that they sleep less. Similarly, long sleepers have a genetic makeup and they can be normal. So we have to look at their history, family history, to know what would be their ideal sleep duration. And of course, with age, your sleep duration heavily changes. For example, during your newborn period, 15, 16 hours, you sleep continuously or in bouts. But once you become child, by 3 years, a school-going period, you tend to be closer to the adulthood. And once you're older, you start decreasing your sleep duration heavily. So there are several factors which affect sleep. We need to look at them. And of course, you can also look at the other species to know. For example, animals who live inside burrows tend to sleep longer compared to animals who are in the open. If the animal is a prey, it's a predator, it tends to sleep longer. If it's a prey, it tends to sleep shorter. A large animal like elephant, giraffe, etc., tend to sleep shorter sometimes, though they have a huge body. Small animals like rodents and opossums, they tend to sleep the longest. So these are some interesting facts which will help us look into sleep from a different perspective. Now, another very, very important part is we have evolved over a period, our cultures have evolved, and they have severely impacted our sleep. So when we were hunter-gatherers in the forest, we tend to sleep more. We didn't have light sources. We had a completely different sleeping pattern. Once we shifted to homes, apartments, closed rooms and bed with light, we tend to have changed it. In fact, there was a study where people were, the participants were put inside a cave without any light and time cues. And they try to emulate the environment that a person would undergo in a hunter-gatherer forest kind of situation. So there what happens is the daytime is short, say around 10 hours, and nighttime is 14 hours, like a winter in the ancient period. So what happens is, here each horizontal line represents one particular day. And if it is blank, that means the person is awake. And if it is dark, that means the person is asleep. So using a actigraphy fitness band kind of thing. So you can see, though the lights were off, say at a particular period, after a period of time, the person took several hours, 1 to 2 hours before going into sleep, even though lights were off and the person was lying in the bed. And then when the person gets sleep, he continuously gets sleep for a few hours, and then he tends to get up in the middle. And this doesn't happen in one day. It actually happens in several days. In fact, you can see a gap. That means the person getting up in the middle of the night. And later, to get up. So the sleep is spread, but in the middle, you can see a gap, which is a break in sleep. And you can also see that there is a latency to go into sleep. Same person, after several days, you shift the regimen. You switch off the light much later, after 16 hours of daylight, and then allow them to sleep. Now you see the person gets instant sleep. Within 15 minutes, the person goes into sleep, and very rarely does this person get up in the middle of the night. And this is what our modern lifestyle is teaching us. We have lights which push our sleep time further, and we have fixed schedule, isolated environment. So we have kind of lost that getting up in the middle of the night. But people have found that some genes still retain in some of the individuals. So anybody who has what is known as biphasic sleep, that means getting up in the middle of the night and getting sleep only after, say, half an hour to 1 hour or 2 hours, that's actually normal. It's built into our genes. And it is normal to take some time before going into sleep if you have a peaceful environment. It doesn't mean that you should get instant sleep within 15 minutes. Okay. So, as I said, sleep is a complex phenomenon. When you go to sleep, it is not that you go into a particular state instantly. You actually go from wake to many stages of sleep and come back. There are multiple stages involved. So in this graph, for example, when you're awake, we put a line here, and then your awareness decreases. So that dozing off period is called NREM stage 1. I hope none of you are in that state now. So some people call it light sleep, but as you can see here, it's colored light green. So that's more like awake than asleep. And after that, the proper sleep starts, which is called NREM stage 2. Now, what is NREM? NREM is non-rapid eye movement. So non-rapid eye movement just means that if you observe the eye doesn't move during that part of the sleep. Whereas after some time, you get a stage called rapid eye movement sleep when your eye starts moving. So this period where your eye is not randomly moving too much, that is divided into stage 1, 2, 3 based on the depth. So first will be stage 1, which is a light period, which even during wake time, you can actually, when you doze off, when I say you doze off, I would actually mean that you went into NREM stage 1. But in a proper sleep quota, you will never just go to NREM 1. You will always go to NREM 2, which means that you actually slept. And that is a light sleep because you can be awakened easily. Then you go to deep sleep, which is called NREM stage 3. This is the deepest sleep where I mentioned about immune recouping. I mentioned about deep cleaning in the brain, etc. happening. So it's an important region. And after that, there is one more deep sleep, which is called rapid eye movement sleep. And this is the period where your brain is active. At the same time, it is deep sleep. And you can't wake the person up very easily when the person is in dreaming state. You would have realized that. So these two are two different deep sleep that one goes through. And we tend to come back to wake or light sleep again, and then again go back to deep sleep. And this repeats multiple times. And this is called sleep cycle. And in fact, how do you study this? People study this by looking at brain activity. So brain activity shows traces like this. I'll not explain too much, but you would have heard, at least some of you would have heard about this beta, alpha, theta, delta. So beta means the waveform is going up and down faster. Then the brain rhythm becomes slightly slower called alpha. Then it becomes much slower called theta and much slower called delta. So it's kind of from fast to slow. Now, if you look at the sleep stages, you will find that the faster one, beta and alpha, occurs when you are in light sleep. The theta rhythm occurs during your light sleep, N2. And once you enter deep sleep, you get delta. But the paradox is that in the other deep sleep, which is called rapid eye movement, instead of delta, you see fast activity, which is beta, alpha, and theta. So all these things in mixed form happens during rapid eye movement sleep, which is also another deep sleep. So you can, from the brain rhythm, you can make out the difference between these two deep sleep states. And the method used, the technique which is done to do this estimation is called polysomnography. Somnography means sleep study, and poly means multiple measurements. So not only brain activity, which will give you these waveforms, it will also look at your respiration from your chest movements, muscle movements from your leg and your arms, ECG, breathing around the nose, snoring. Many measurements can be done together. Now, you don't need a sophisticated sleep lab to understand sleep. Most of you might have your phone app or fitness bands, which can also do this reasonably well. And you get graphs like this, which cycle that I mentioned. So in your initial part, you will have a lot of deep sleep. Later part, you have more of REM sleep. And you get scores nowadays in some of these apps. So using scientific studies, people have found this trace of how the sleep should be. And you can count the amount of stages you pass through and create graphs. So people have made normative graphs like your BP, height, weight chart. You can have a sleep chart also and compare whether for that appropriate age you have the appropriate duration. Now, we don't tend to sleep at the same time and get up at the same time every day. We tend to have some amount of variability. But the problem, and this variability is linked to our hormone. So I mentioned about melatonin peaking up in the middle of your night. So if you look at the melatonin peak, it will almost correspond to the time when you go to sleep and wake. So that amount of variability will be there, say 1 hour here and there. But there are disorders, mental disorders like schizophrenia, where this variability is much larger. In fact, there are groups where the variability is extremely large. They might be severe mental disorder groups. So too much variability is actually bad. So 1 or 2 days, few days, it's fine. But if it's continuous and even your hormone has that variation, then you are in trouble. Now, in schizophrenia itself, we have done a study and we found that even within the sleep, the cycle pattern itself, if it shows huge amount of variability, it's a sign of disorder. Okay, so with that, I think I try to cover some of the fundamental aspects of sleep. Now, I would like to convey another important aspect of sleep, that is the discretion or the division between sleep and wake. So how is the brain different in sleep and wake? So don't worry about this graph. Just concentrate on this fact that this is about waking, and everyone knows about cortex. Everyone knows about cortex, and everyone knows about thalamus. So whenever we have an information coming from outside, when you hear a sound, etc., it actually passes through thalamus and finally reaches our cortex. And some of the other brainstem regions, they tend to act together and help in making our sleep state or wake state possible. So this is what normally happens in wake. And during sleep, what happens is, you can see cortex is slightly light colored and thalamus is light colored, and this whole region is not active. So it's primarily cortex and thalamus communicating with each other, and this external information can come, but it is very low. So you can see that here cortex and thalamus, it's more like a one-way traffic with the influence from other part of the brain. Now other part of the brain is disconnected and cortex and thalamus goes into a continuous loop. This is what happens in our deep sleep, NREM deep sleep. Whereas now if you look at dreaming state called REM sleep, now thalamus do not come into play much. So no more external information comes. Now the cortex has to deal with the internal information. So whatever you have in memory, with that, the cortex will be playing and you tend to get dreams and other activities. So this is in nutshell the different brain activities which allows different sleep stages. So for waking, every region is connected to each other. During NREM stage, it's cortex and thalamus alone connected, but much lower than the waking state. Whereas in REM stage, the cortex is almost as active as the wake stage, but it is not connected to the thalamus much. And it is taking all information from within to create all these dream and other states. Okay. Okay. Yeah. So the other question is brain disconnected as such? When you are in deep sleep, I said that it's disconnected from sleep, from many other parts of the brain. But is it completely true? If you look at dolphins, which actually dolphins and whales have to travel several miles to reach their location for breeding. So they tend to have something called half brains. I don't know if anyone knew about this. So when people have studied their brain, they find that if you look at their right brain and left brain, one brain will be having this low waves, which are showing sleep. The other brain is showing fast activity, which means awake. After some time, after several hours, the right brain will go into wake and left brain will go to sleep. So this transition happening, so that one of the part of the brain, one half of the brain will always be awake and the other part can sleep. This is very much useful for dolphins because they will be able to use the part of the brain which is awake to do the swimming part and navigate, whereas the other half can get sleep because you have to travel for several days before it can reach its destination. Now, this is also true for many migratory birds also who need to travel several kilometers. In fact, recently, people have found that humans also have some genes and some networks related to this. So when you sleep, especially when you go to a new place, new environment, one half of your brain will always be showing less sleep compared to the other. Because unlike, say, dolphin, you can't have a complete waking in one hemisphere and complete sleep in the other hemisphere. But there is a tiny bit because of which you can see the wave is slightly larger on the right side and it is slightly smaller on the left side. So, which means that your brain is actually, this part of the brain which is showing smaller activity is actually more awake than this other part of the brain. And this has been linked to something called first night effect. That means more this discrepancy, more this size difference between left and right, more time it takes for you to sleep. Again, something we learn from nature. Now, that tells you that brain is not fully disconnected. Some parts can connect with each other and do the waking part. Some parts can connect with each other and do the sleep part. Even if your entire brain is sleeping, you can see from wake to that light sleep when you enter. Normally, we would expect that the brain is disconnected because you're losing your awareness partly. But what is observed in many of the neuroimaging studies is, in fact, from wake to NREM1, when you shift, many brain regions are much more connected than wake. If you see this red, that means this red regions are connections which are more in NREM1 than in wake. In fact, many regions are much more connected during NREM1 than in wake, which is again not something that we would otherwise expect. But once you shift, then compared to wake, many more regions become less connected. And once you are in deep sleep, most of the regions are much more disconnected than the wake. So this is one thing we have to remember. So connection actually increases from wake into light sleep and gradually decreases. So it's not like a progressive decrease or progressive increase. It's an inverted U kind of. And one core aspect is one among the network, very common, which helps in mind wandering, thinking, etc. Random thoughts that comes into your mind is because of a network called default mode network. Whenever you are not doing a task, this is what comes. So some part of the front region communicates with the back region. This is how it happens in wake. Now just see what happens when you are in deep sleep. The same network, but the posterior part, the parietal, occipital, that region are much stronger connected than wake, and it almost lost the connection with the frontal region. So this is something we have to look into. I'll come back to this later. So this shows that this specific disconnection from front is what helps in deep sleep and is required for deep sleep. Whereas if you have to be awake, you have to connect to your front part. Now, people have tried to study this.
Interrupt, sir.
Yeah.
So you have 10 minutes to wind up, then we'll go in for the questions.
Yeah, sure.
Thank you.
So one innovative study to look into is to do something called ZAP study. That means you give strong magnetic pulses when somebody is asleep. So this particular person is asleep in a chair, and this gadget which is sitting on his head gives strong magnetic pulses to stimulate the brain, a particular region of the brain. So here, if you see, this yellow spot is a place where the stimulation is done, but this blue and red regions are the regions which got stimulated. Though stimulation happened in one tiny region, the signal spread to many other regions. So this is what happens in wake. You get this giggled waveform. Now, same thing, if you give when the person is in deep sleep, NREM, you see that the information doesn't spread anywhere else. It remains in that place and you get a smooth wave. Whereas now, if the person is in dream phase, again, the information spreads to many regions and it's almost similar to wake. So this is one test to identify how conscious the brain is. Is the brain spreading information or is it localizing it to one or two regions? In fact, it can also be used to differentiate sleep from coma, like stage, locked-in syndrome, etc., where the patterns are quite different. You can also give sound and see the response in the brain. So we feel that during sleep, your brain will be disconnected. In fact, it produces some waveform. In fact, it's called ERP. When you give the same stimulus during awake, you get lots of giggles like this. Whereas the same stimulus, if you give during NREM sleep, it goes in another direction, and it goes in another direction if it is REM sleep. So by giving auditory stimuli, you can check which stage the person is in, and we can actually know whether the person is processing these audio. In fact, if you give the person's own name, the response is larger. And we have worked on this in our lab too, found huge amount of accuracy. Another aspect is, can we be asleep while awake? So when we are awake, I said most of your brain will be active, but some part, one tiny region in the brain can be asleep, whereas the rest of the part can be awake, which is called local sleep. And when you do EEG, you can find only some sites showing that pattern, others showing wake activity. And when you look at the neuron, you can see that those neurons will be silent during that period. In fact, this local sleep itself, depending on where the local sleep happens, whether it happens in the front, middle, or posterior, you can have different mentation. You may see a dream or so if the local sleep happens during this period, whereas if it happens, the local sleep happens in the posterior part, you will be blank. And during actual dream during night, many brain regions become less active, like this blue region in the front, and regions like in the posterior regions become much more active during dream. So if you look at the brain, you see there are differences between dreaming and waking, but you have an intermediate stage called lucid dreaming, where some part of the front region are almost as active as wake. Here you can control, and nowadays people have come up with gadgets which can be used to go into this state. And sleep can also be used for memory improvement. So you learn something and have some cue like a bell. If the same bell is played during sleep, subsequently, the morning when you get up, you will be able to recollect much more, because during the sleep, when you play the sound, the same memory which was formed during wake get replayed and get strengthened. This is an area that many people work on. We have recently tried to look into meditation. We found a huge response by meditators when they are in stable sleep, and the DMN network that I mentioned are much more stronger in these meditators and helps them to become aware of external environment and still remain asleep. So this is one interesting area that sleep studies provide, looking into all these extremely mentally trained individuals. I suggest these two books if anyone is interested in understanding sleep more. And this is the lab. It's a new Center for Consciousness Studies, and my team here. Thank you. Any questions? I would be happy to answer.
Yes, sir. We would now take up a few questions from participants. Smiling during sleep seen in infants. Does that fall under parasomnia?
Yeah, good question. So I said all those disorders that I mentioned are only called disorders when they impact the person's day-to-day activities. For an infant, if it disturbs the sleep, if it disturbs the feeding habits, if it disturbs any other thing, then it's a parasomnia. But otherwise, it's not a parasomnia. And in fact, in children and infants, these habits, with they jerking their legs, they smiling, they moving, are normal, extremely normal.
Okay, the next one is, what is the role of sleep peptide in the genesis of sleep?
Sorry, can you repeat that question?
You want me to repeat, sir?
Yeah, I didn't get that.
What is the role of sleep peptide in the genesis of sleep?
Okay, yeah, good question. So I didn't mention about sleep peptides specifically. So when, for example, when you take coffee, you know that your sleep is decreased. You become much more alert. So what actually happens is, one of the key sleep peptides is getting blocked. So sleep peptide, the key sleep peptide is adenosine. When you do day-to-day activities, this peptide gets built up because it's a byproduct of nerve activity. So they get built up into one part of the brain region around the hypothalamus, etc. And this build-up actually causes sleepiness in you. When you take coffee, what happens? It blocks this peptide's activity in that brain region. So even though the peptide is built up, it doesn't evoke sleep. So similarly, many peptides, including the regular hormones, they all have they all either increase or decrease.
Okay. What is the cause of loss of muscle tone during narcolepsy? Any neurotransmitters involved?
Good question. So I spoke mostly about what happens in the brain during sleep. I also mentioned that from the brainstem and spinal cord, etc., information comes to the brain, especially the cortex and thalamus, to make sure you are awake, you stay awake, or you stay in a particular sleep state. So for this muscle tone to be low during sleep stages, especially REM, you have connection with spinal cord, and that connection, if it is affected, so there can be degeneration in that region, especially dopamine and dopamine related circuitry. That circuitry, if it is affected, it will prevent the spinal cord from shutting down the muscle. And that is what happens during narcolepsy.
Can rest and active phases duration be taken as sleep wake durations? If yes, how approximate will that be? Can that be interpolate?
Yeah. So when we say rest, we have to decide more. For example, I mentioned there is something called local sleep or micro sleep. So which means when we call sleep, according to current guidelines, if your activity, if your brain activity is low, your physical activity is low, very similar to sleep, for 15 seconds or more continuously, then we call it sleep. If it happens for 5 to 15 seconds, then we call it micro sleep, which means behaviorally you may not be able to make out whether the person is asleep. But if you can quantify this for more than 15 seconds continuously, that motion can be considered as sleep.
Okay. What happens if we sleep too much? Is it bad? I think you talked about it.
Yeah. So if you saw that part, yes. There are many studies which have found increased sleep actually harms. So that's why it's critical to understand what is the ideal sleep duration. That's why I spent that first half specifying that we need to understand sleep from a larger perspective. And for each individual, you'll have to figure out what is the ideal sleep. Only then you can figure out whether the sleep is more or less.
When you say CSF washes the debris, what toxins are you referring to?
So some of you would be knowing when you say dementia, Alzheimer's, you know there are neurofibrillary tangles, beta plaques, etc. These are supposed to be toxic materials. They are actually materials which are released by day-to-day neuronal activity. But when they accumulate to a larger extent, they tend to harm the neurons. They tend to evoke immune responses and cause neuronal death. So any of these moieties are called the toxins. And some of these toxins, once they become too large, they cannot be cleared by the cells around. We have cells called microglia, etc. They usually take care of them. They engulf and take them off. But when they become large in size, they do not get removed by cells. They have to get removed by the flesh.
Why is that some people sleep in single position throughout and some people keep twisting and turning all night?
I do not have an answer to that. I can only say that there are lots of physiological variation. In fact, without disturbing our sleep itself, our body has a mechanism to move our limbs. So you can always turn during the right cycle, which will not disturb your sleep. So if your sleep is good, you will always be twisting and turning around during the end of each cycle, because of which it doesn't harm your sleep. In fact, it helps because if you stay in one posture, that part of your muscle will be pressing against, there will be less blood supply to that region. And turning around actually benefits your body. You will not have much muscle pain when you get done. But it only happens if your movements and your sleep cycles are in proper alignment.
We have another question. What about people who always fall into sleep just few minutes after getting into bed, even when they aren't fatigued? Are there any biological differences in this case?
Yeah, that's another important question. So a common phenomenon and normal variant which I mentioned is called hypnagogic hallucinations or hypnic jerks, etc. Where when you start entering into sleep, your muscles become flaccid, your balance system becomes much more alert, and you kind of lose consciousness. So the idea is you lose consciousness, by that time your muscles should be already flaccid. Now, if there is a mismatch here, then you will experience hypnic jerk. And the problem is cases like narcolepsy, Parkinson's also have this problem. Say 10 years before your actual florid Parkinson's disease, you can have these kind of hypnic jerks where even if you have a calm environment, you had a normal day activity, if you still get these hypnic jerks, this possibility is there. But in which case you will need to check on those. But otherwise, majority of the cases, it's a normal variant.
There is another question. Why do some people sleep with their mouth open?
Okay. Yeah. So that's not directly my answer. So our jaw tend to have different shapes. So some of us have something called high raised palate, because from the childhood itself we put our hand inside and other things. Our palate is large, so our teeth get crowded in a particular manner, because of which our mouth tend to be open. Some people have jaw slightly inside than the others. So these are variants in our anatomy which lead to having our mouth open. So that's the commonest form.
Is sleeping in segmented form, for example, sleeping in two shifts worse than sleeping in a straight stretch, given that the amount of sleep remains the same?
Yeah, very good question, because I forgot to mention some communities, like I mentioned, Egypt and many Mediterranean regions, they tend to follow something called siesta, means 2.5, 3 hours afternoon nap, and at night they sleep only 3 to 4 hours. So if you add them together, they get 7 to 8 hours sleep, but they have a huge gap in between. Similarly, I mentioned that waking up in the middle of the night is normal. So if you sleep very early, say 9:30, 10, you wake up say 2, 2:30 and wake up for 1 hour and then go to sleep. So ideally that's 2 separate sleep during night. They are all normal.
Another question, sir, is there any particular sleep pattern in coma state also?
Yes. So one discretion is coma is different from sleep. So coma patients also, if they are in a normal coma state, means say for example, a coma patient having a sleep pattern is a good sign, because in extreme coma you may not even be able to differentiate the sleep and wake, and they may not even get a proper sleep. So coma patients can have sleep patterns, and there can be coma stage where sleep pattern is not there.
What is the phenomenon when we feel like we are falling in our sleep or in our dream?
In our dream, we falling in sleep.
Like we are falling in our sleep or in our dream, both, like.
Sorry, I didn't get that question properly. Sorry.
Okay, so we'll go on to the last one. How does an energy drink affect our sleep?
Good. So like coffee, many energy drinks have caffeine. And as I mentioned, many of the sleep peptides, including adenosine, its function is severely affected and it will prevent sleep. So if it prevents sleep, subsequently you will not get, because if your sleep pressure builds up towards the early part of the night, you take energy drinks, it will prevent that build up to happening, and you will only get the proper sleep in the next night.
Okay, sir, that was the last question. So thank you, Dr. Arun Sasidharan, sir, for introducing us to different sleep stages and for sharing interesting facts about sleep. We learned how sleep can be viewed as window to examine the state of a mental functionality, and also how content and duration of dreaming could be examined using sleeping brain patterns. Your talk also gave us an insight into neuromodulation techniques that can be used to not only improve sleep, but also affect waking mental activities. We are indeed thankful to you, sir, for inspiring inquisitive minds to explore the vast potential of sleep and for sparing your precious time. Thank you once again, sir.
Thank you.
Okay. Our next speaker for the day is, hello.
You're audible, ma'am. You're audible.
Okay. Our next speaker for the day is Professor Madhuri Behari, Director and Senior Consultant, Fortis Hospital, Delhi. Professor Madhuri was also former head of Neurology Department, AIIMS, New Delhi, and President of Indian Academy of Neurology. Professor Madhuri has been awarded with many prestigious fellowships, including Commonwealth Medical Fellowship, Fellowship of International Federation of Society for Electroencephalography and Clinical Neurophysiology, and many more. Professor Madhuri is Fellow of Indian Academy of Neurology and National Academy of Medical Sciences. Professor Madhuri is member of various professional bodies and has published more than 300 research articles on Parkinson's disease and neuromuscular disorders. Before I hand over the mic to Professor Madhuri, participants are requested to type in their questions in the chat box. Thank you. Welcome, ma'am. Over to you.
I hope everybody has woken up from this beautiful talk on sleep, and after having a very good lunch and the postprandial state is over. At the very outset, I'd like to thank the Indian Academy of Neurosciences and the team NeuroEunoia and the faculty of Gargi College, especially Dr. Jaswinder Kaur, for inviting me to this very prestigious multidisciplinary neurosciences conference. My talk will be on dementia, a malady which threatens to reach a pandemic proportions in the coming decades, so that it will be there all over the country, all over the world. As the world is longevity is increasing over the world due to better medical sciences, it has resulted in problem of the elderly becoming more available, and so we have a large number of elderly with dementia. So dementia is proportional to the age of the population, and so we find because of elderly population increasing, the number of dementia is also increasing. I'd like to share my presentation now. Fine. It doesn't come here.
Ma'am, it will come. The network might be a little slow.
Okay.
Just share the screen
I have shared it.
with the optimize for text and images option.
Optimize for text, huh? I have no videos, so it should be working.
Okay, so ma'am, did you select the screen?
I haven't got the screen here.
Okay.
I think I have to go down and get my screen out.
Okay.
Yeah?
Okay, ma'am. All right.
How to get this thing back?
Ma'am, what's missing?
See, I've got my open my presentation, but I'm not able to get the.
Okay. So, ma'am, are you on the Cisco WebEx platform right now, the application?
Yeah, yeah.
Okay. So, in the application, in the bottom row, can you see the share button?
Got it, got it. So you will see it now.
Okay.
देखिए, आया?
Yes, ma'am. आ गया. Very, very nice.
Great. So my talk today is on dementia, and this is the pandemic which stares in our eyes, and it's likely to become throughout the world as the population grows. And we come to what is dementia. As the word shares, it means it's a Latin word, and it means devoid of mind. So when somebody has dementia, the person has impairment of memory, and any of the following cognitive functions are impaired. And this is apraxia, inability to do any learned complex tasks, or aphasia, which is difficulty in speech and language function, agnosia, which means inability to recognize common known articles, impairment of executive functions like judgment, abstract thinking, etc. There may be other domains which may be affected, like personality, behavior, and social misconduct. This may result in decline from a previous level of cognitive and social functioning, and also it has to be a progressive decline. We cannot call a person demented if the person is in abnormal conscious state or is in severe depression. So we have to make sure that the patient is conscious when we make this diagnosis. The WHO estimated that the number of people living with dementia will double every 20 years from 42 million in 2020 to about 81 million in 2040. The rate of growth is going to be highest in countries like India, China, South Asia, West Pacific region, and it is going to be around 300% or more. According to Basel 2001 population, approximately 24 million people had dementia, and every year about 4.5 million new cases are added every year. This contributes to a disability adjusted life years to 4.1%. It is thought that if we live long enough, that is about 120 to 140 years, most of us will develop dementia. In Western literature, it has been shown that in people above the age of 65 to 70 years, almost 5 to 10% have dementia, and people between the age of 75 and 80, about 15 to 20% have dementia, and over the age of 80 years, almost half the people have dementia. So it is a very huge numbers. We see that among the dementias, Alzheimer's disease forms almost more than 50%, and dementia with Lewy body forms about 15 to 35%, and vascular dementia forms only 5 to 20%. So maximum number of cases of dementia are formed by Alzheimer's disease. In India, a study is done by two authors in in the southern part of India has shown that above the age of 60, in 1951, the graying population had dementia of 5.6%, and in 2001, above the age of 60, 7 to 8% had dementia, and in people above the age of 60 in 2011, it was expected that about 10% will have dementia, and in the 2021, almost it is going to reach about 15%. So which is fairly large number. Why does it not progress? When we look at the prevalence of dementia, it is a major clinical public health problem. It is a burden on the society as well. And we find that with the increase in care and survival, increase in people above the age of 65 is observed, and number of people having dementia is also increasing, especially in the developing worlds, also in the developed worlds. The maximum number will be in the developing countries. Coming to the Indian scenario, studies in southern India, North India, and western India have shown that the dementia occurrence prevalence is close to 1.5%, whereas in eastern part of India is close to 1%. And it ranges from 0.4% to 3.4% above the age of 60%, and the Alzheimer's disease forms about 0.2% to 1.3%. What happens every time?
Ma'am, the right arrow doesn't work here.
A study from a community study by Ganguly in 1996, which is also known as the movie study, followed up about 1,300 non-demented subjects over years, and the crude incidence rate was about 25 per 1,000 person for all per year for all dementias. However, this rate Alzheimer's disease was 93 per 1,000 per year, and for according to age adjustment between 70 to 74 years, it was 8 per 1,000 person per year, and above the age of 90 years, it was 70 per 1,000 person years. So that is how it is in the US. Now we go to the next slide, which shows that our Indian studies are not too many. Can you go to the next slide, please? Dr. Jaswinder. Yeah. So there are not too many Indian studies in India, and the burden of dementia is very time consuming and cost intensive. It also affects the quality of life of the caregivers. The estimated cost has been shown to be about $600 billion a year, which is based on the insurance claims. We have no such studies from Indian workers. The time spent by the informal caregiver of a demented person is about 10 times higher than the formal care. And generally what happens is, because of the stigma attached to demented persons, many people do not say that their family has a demented person, and they take over the roles and responsibility of the dependent person, thereby decreasing the number that we see. Next. So coming to what are the risk factors, it has been seen that the women are great risk for developing Alzheimer's. Men are higher risk for developing vascular dementia. However, this gender difference vanishes after the age of 90 years, and women also get affected by vascular dementia and men by Alzheimer's disease. Next, please. Age is one of the strongest correlates of dementia, but it is not an invariable consequence of aging. We have seen young persons less than 65 years of having dementia, and it accounts for about 9% of cases. Next, please. Race also is shown to be one of the risk factors for dementia. And it has been, earlier it was thought that black people do not have dementia. But recent studies have shown that the dementia is higher in black population versus non-black people, and it is considered as a silent epidemic among African American community. And Alzheimer's disease is found to be about 14 to 100 times higher among African Americans as compared to non-black people. Next, please. Education is being found to be correlated with dementia, especially in China and India, and people with low education seem to have higher prevalence of dementia. In higher prevalence of dementia in African Americans are also is also thought to be because of low education in this group. Next.
Madhuri ma'am, have you lost connection?
I can see her. She's
Madhuri
there.
ma'am, have you lost connection? No, I think she has lost connection again.
But I can see her. How can I?
There is an icon coming on the top right, top left. Ma'am is joining back again. She lost her connection. Hello. Please bear with us for some time. Ma'am is having severe connection issues at her end.
Hello? Hello, I'm there.
Yes, ma'am, we can hear you. You may continue.
Okay, so next slide दिखाइए. Next slide, ma'am. ये तो बहुत स्लो है.
Okay, ma'am, you're there. Don't worry. Just go with it. Our next speaker is here. We welcome Gurcharan ma'am, and she has very graciously accepted to actually postpone her talk by half an hour. So we have some time, ma'am. You can go on.
Okay, so
No
can
problem.
you proceed with the slide?
Yes, ma'am. Head trauma you had already explained.
Yeah.
Okay.
Untreated hypertension. When hypertension is poorly controlled, there are several ischemic damages which occur in the brain and multiple small infarcts which occur into the various parts of the brain. Also, if there is a single large infarct involving the hippocampal area, that can also lead to dementia. And we have seen that the untreated hypertension can lead to dementia in which is known as vascular dementia. Next slide, please. We come to the causes of dementia. So we know that Alzheimer's disease, which is a degenerative and other degenerative dementias are dementia with Lewy body, dementia with Parkinson's disease, and so on. Then vascular dementia also I mentioned, which is because of untreated hypertension. Untreated hypertension and because of small vessel vasculitis or large infarcts, everything can cause vascular dementia. Infections like syphilis, tuberculosis, CJD can also lead to dementias. Among the nutritional factors, most important are deficiency of B1 and B6. So we always insist that one should be very well fed and nutritional things are given. Other causes are metabolic, traumatic, and neoplastic causes. So next, please. In dementia, as we said, that apart from memory, other faculties can be involved. So what are these other faculties? One of them is apraxia, when is difficulty in doing familiar tasks. The tasks that we have learned and we have done over years, we are not able to do. For example, a housewife can no longer make tea. She doesn't know how to. She will look at it. She'll hold the cup. She'll hold the cap of the teapot. Will not know what to do for. And a person who won't all his life will not be able to tie a knot, and person can not wear a shirt, will not know how to button the shirts, and so on. So next, visuospatial disorientation can also occur in these people, and people are not able to go from their workplace to their home, or when they go out for walks, they do not return home, they keep roaming around. and such things can happen. And people inside the house also lose their way, and sometimes when they get up at night or even during the day, they don't know where the bathroom is, and they keep roaming around here and there, and they may even pass urine at inappropriate places. Next, please. They also get disoriented in time and place. They don't know whether it's daytime or nighttime. They may get up from a sleep in the afternoon and then think it is early morning, go to bathroom and brush their teeth and ask for breakfast. So they can't tell the date also, what is the date and what are the new things happening in the next few days. For example, they forget that they have had the Shara or Diwali in the past few days. Next, please. They also have problems reading and writing. This is also known as alexia, in not able to read and agraphia, write. So this is also a major. And they are not able to find retrace their steps to find a place which they have misplaced. They can keep their keys and specs somewhere, and when asked to say, okay, you retrace your steps and find it, they are not able to do that. Whereas a normal person, elderly person also does that. But when they are asked to retrace their path, they can say, okay, I came home and I kept the key here, and then I went to wash my hands. So they are able to retrace their steps and find the lost thing. Whereas a person with dementia will not be able to do that. Next, please. The judgment is very poor and they do not know. Suppose somebody does a job for them like a mali, they don't know how much money to give. And somebody, they go out to a wedding, they don't know how much money to give to the as shagun, and they do not know how much, for a housewife, how much salt to put in the dal that she makes every day. All these judgments are lost. Next, please. They cannot even do simple calculation, addition, subtraction, simple things like go to market and they do not know. They buy a thing for 10 rupees and they give a 50 rupee note, how much to get back from the shopkeeper. So simple calculation also they can't do. Next, please. There may be behavioral abnormality also. They may have become withdrawn socially. For example, in the house or even in the office place, when people are chatting, they remain separate, or sometimes they become aggressive, and for no reason they lose their temper, throw things at people, shout and do such things. So abnormal social behavior is also one of the features. Next, please. So these are some of the faculties that may be lost early on, and we should be able to identify when one of our relatives has this. So what are the stages of dementia? The very early stage is known as mild cognitive impairment, and this may lead in a large number of cases to early dementia, and that further leads to advanced dementia. Next, please. There are different types of mild cognitive impairment, and this is a nosological entity, and not in this the patient himself or herself come to the doctor to say, I'm losing memory, that I tend to forget, and it's not the family members generally. And when we do cognitive evaluation, we find that the cognitively the scales, the scores are not so low. For example, if we do MMSE, the score out of 30 may be about 25 or even more. And when this cognitive impairment, mild cognitive impairment occurs, it does not impair the activities of daily living. There's no effect on that, and people work normally in their daily activity. Next, please. I was coming to subtypes of MCI, and if we can divide into two major types, the amnestic type and the non-amnestic type. In amnestic type, only single domain, that is memory, is affected. Whereas in non-amnestic type, memory is retained, but there may be a single domain, non-amnestic MCI, or multiple domain MCI. In single domain or multiple domains, one or many of these faculties may be affected, like aphasia, agraphia, alexia, poor judgment, poor visual spatial orientation, and so on. as we have discussed earlier. Next, please. So how many people convert from MCI to full-blown dementia? A study in Canada has shown that when people were followed up for 19 months, the conversion rate, about 44 people converted to 44% converted to Alzheimer's disease, which is corresponding to annualized conversion rate of 28%. A large meta-analysis which included 141 studies found that that was in Mayo City, found that the conversion of MCI to dementia was as below. Dementia was 9.6% and Alzheimer's disease was 8% and vascular dementia was about 2%. Next, please. What are the risk factors which make MCI get converted into dementia? One of the major risk factors is if the faculty or the domain involved is beyond memory. So the person has memory deficit and other involvement as well. Or there is non-memory, non-amnestic type, 2 types. So that is more likely to be converted into dementia. If only memory is affected, it may not. Also, it is seen that the people who have high levels of serum homocysteine develop dementia. They convert into dementia. So how to recognize early dementia? As said earlier, these are language disturbances. People find to find the right word while speaking. They cannot read, they cannot write. Hand tools, tools that they use regularly like kerchi, like scissors, like hammer, they can't use properly. There's change in behavior. For example, they may be withdrawn or aggressive. There may be visual spatial disorientation, not able to find words or not able to write. They may be not able to read the clock, which they have been doing earlier. There is clumsiness in dressing, not able to plan, and poor judgment, poor solving. Problem solving is also poor, and they are confused in time and space. Next. So how we diagnose dementia? Most important is our clinical examination and the history that the patient gives and whether it affects the daily activities of life or not. And we do these 2 scales are generally used, the mini mental state examination and the Montreal cognitive assessment. These are the 2 tests which are routinely done to find out what is the state of cognitive functioning. Apart from that, certain tests can be done. Next slide. MRI is the first basic test that we the times do. And we find that this is the normal MRI of a normal person. We found that the ventricles are small and the sulci are very narrow. And when we go to the next slide, next slide will show a patient with Alzheimer's disease. The sulci are so wide and deep, and the ventricles are huge, and the hippocampus shows very significant atrophy. So this is what we see in patients with Alzheimer's disease. We can see the next slide now. This will show a frontotemporal dementia where we find the majority atrophy occurring in the frontal and in the temporal regions. Next one. A vascular dementia will show ischemic changes around the ventricles, like you see these white areas. Next one. Amyloid imaging is one of the imaging modalities, and it is done using a Pittsburgh compound B, which is a ligand which is used to which binds with the amyloid in the brain. Whether it is amyloid associated angiopathy or neuritic plaque, it get joined to that, and then it can be seen on the PET scan. As we see a normal subject and MCI, there is hardly any red area. In another MCI, there is very small red area. So shows that there is plaque amyloid accumulation here. In another MCI patient, you see large number of amyloid deposition. And similarly, in a patient of Alzheimer's disease, we see this amyloid deposition. So this is a very good test which shows that there is accumulation of amyloid in the brain. Next, please. SPECT is also a similar test which is single photon emission computed tomography, and it shows what is the perfusion in the brain. So we see the perfusion here is much low in the occipital area and patchy areas in the brain, like here and here and here. So these areas are not perfused and showing that these areas are non-functional, so to say. So this is in vascular dementia case. Hypoperfusion is also seen in patients who have other types of dementias like neurodegeneration. So let's go to next slide. Biomarkers. Now, before understanding the biomarkers, we have to go to the pathophysiology of Alzheimer's disease. We have tau proteins in the, when it gets phosphorylated, it becomes non-insoluble, and it forms tangles within the neurons. So it becomes tangles in the neurons. So in the extracellular space, the tau is much less phosphorylated as well as the total tau. Its level in the CSF becomes less. And we have the amyloid beta, which is 42 and 40, which forms clumps in the CSF space, in the extracellular space, and we find that the amyloid is increased. For example, the amyloid beta 42 is increased and the beta 40 is also increased. But 40 is not so important, but the ratio of amyloid beta 42 over 40 is very suggestive and helpful. Next slide. Now we see the synapse. What happens with the synapse? This is the synapse. We see that there is lot of proteins like synapse, SNAP25, synaptobremin, and VAM, vesicular associated protein. And these proteins help in the extrusion of the neurotransmitter in the synaptic cleft. And in Alzheimer's disease, these synapses are lost and they are degenerated, and so these synaptobremin and SNAP are also decreased in patients with Alzheimer's disease. Next slide. Now we come to dendritic spine. The axons have a lot of dendrites, as we see here, and these have a protein called neurogranin. When this is very important, the function of this is very important in memory, and these are in high number in amygdala and hippocampus. So when they degenerate, there is a neurogranin gets into the extracellular space and the CSF level rises. Next slide. So when we do series of biomarkers, we find that the amyloid beta lowered, as peptide gets, which is in cell prolog, they are fine. In the extracellular space is low. There is high concordance between CSF beta 42 and amyloid PET scan. It is seen that the CSF beta amyloid shows a higher positivity as compared to amyloid PET scanning. It occurs in the early stage, whereas PET scan may show abnormality much later. The ratio of amyloid beta 42 over 40 is much more important, a stronger accuracy as compared to only 42. Next. Next slide. Phosphorylated tau proteins. This degree associates with degree of atrophy on the brain and severity of cognitive impairment. CSF total tau and p-tau are highly important in preclinical Alzheimer's disease or in MCI. The tau PET scan may be normal, but this may become an early marker. And PET scan occurs later. Next slide. The core CSF biomarkers for Alzheimer's disease are volumetric MRI, amyloid PET, and tau PET. These are more important at the later stage of disease, whereas CSF biomarker of low beta amyloid 42 and the ratio beta amyloid 42 over 40 and tau, high tau, total tau and phosphorylated tau, these are important in early dementias. Next. The presynaptic membrane, as I told earlier, is also important for cognitive functions, and they can be elevated before we can see any atrophy on the MRI. Next slide. Next slide. Dendritic protein, which is I have said is neurogranin, is also elevated, and it's a good biomarker. So now we move from the CSF to blood. Most of the clinicians prefer to do blood tests rather than CSF. So in this we find that the shows that the data shows that the plasma tau level has longitudinal studies have shown that there is significant correlation between plasma tau levels and the future cognitive decline, and it is a biomarker in the early stage of the disease. Neurofilament L is high in Alzheimer's disease patient in the CSF and also in the blood, but it's not so important in the blood. So measurement of amyloid beta 42 in blood is not very important. So we can go to next slide. Next please, next. So what are the prevention do to get dementia? The WHO has chalked out 3 levels of prevention. First is primary prevention, the secondary prevention, and the tertiary prevention. In the primary prevention, we address at the modifying risk factors. These risk factors are cardiovascular risk factor, diabetes, type 2 diabetes mellitus, hypertension, obesity, and hypercholesterolemia. So one should check these and keep it under regular values. Lifestyle factors, for example, alcohol, smoking, unhealthy diet, sedentary lifestyle, all these should be taken care of. One should be more active. And depression and head disease are also targeted intervention. One should prevent having depression and it should be treated as soon as possible. Next, please. So it has been shown by several studies that you can reduce your risk of dementia by getting regular exercises, by not sewing, avoiding harmful use of chemicals, controlling weight, eating healthy diet, maintaining healthy blood pressure, cholesterol, sugar levels, controlling depression, educating yourself to prevent low educational status, social isolation should be prevented, cognitive activity should be done all the time. Even in later stages of life, one should keep on learning new hobbies, new languages, and new puzzles. And it is the slogan that if you don't use your brain, you will lose it. Next, please. The secondary prevention is twofold. First is to prepare the family for the consequences and the roles that the family member may have to play in the eventually, and this will enhance the quality of life of the patient as well as the caregiver. The next prong approach is to identify MCI and control it by doing screening tests as mentioned earlier in the CSF blood and very PET scan, fluorodeoxyplate and volumetric plus MRI and other tests. And the major challenges in second the diagnosis of clinical changes because all these tests are very expensive and not available in many areas of the country. Next, please. Coming to tertiary prevention, it focuses on timely diagnosis and treatment. As we know, the treatment that we have at the moment cannot prevent or regress the previous one cannot prevent or we cannot go back into the disease, and we should take care of the abnormal behavior, depression and aggressiveness of the patient. There are other drugs which are available, which we actually need strategies which can reduce the deposition of amyloid beta into the brain and also the reduce phosphorylation of tau. So I'd like to conclude here. Next, please. We know that dementia is a pandemic and the rate at which it is rising, especially in the developing countries, is very high. The number of graying population is increasing worldwide and hence dementia will become a great drain on our resources. Prevention is more important and to avoid it by adopting healthy lifestyle, keep on learning new skills to prevent it. Early detection and timely treatment is most important. Biomarkers can help in detection of the early disease. And again, as I said, use it or lose it. So you must keep on learning new hobbies, new languages, and new puzzles, etc., to prevent dementia happening. So thank you very much. Next slide. Thank you very much for your patience with me. I'm sorry for the problem that has happened over the presentation.
Okay, that's all right, ma'am. It was not under your control. That's all right.
Okay, it's still connecting. You know, the goal checker is going on still.
No problem, ma'am. Actually, that might be because of low bandwidth. So the moderator can take up the questions now.
Sure, sure.
Thank you, Professor Madhuri.
Thank you.
Thank you, Professor Madhuri, for such an intriguing lecture on dementia. So we shall now take up questions from the participants. So our first question is, what is the role of BDNF in hippocampal and prefrontal cortex mediated memory?
See, as we know, brain-derived factors, they are very important and they play a major role in the repair of the dendrites and the neurons. And we know that the dendrites and the synapses play a very important role in the cognitive functions, so they help in the repair.
Yes, ma'am. So our next question is, are shorter telomeres related to senile dementia?
Yes, it has been seen that those with shorter telomeres have more chances of getting dementia. Also, there is one ApoE4. Those individuals who have ApoE4 are also likely to develop dementia, Alzheimer's basically. As I say, work has been done on Alzheimer's disease. So whatever we say, even more research has been done on Alzheimer's disease. So most of the data is on Alzheimer's disease.
Yes, ma'am. And there's this last question. If identified or detected early, can dementia be arrested?
See, at the moment, there is nothing like arresting. I said in the last slide also, it cannot be arrested. We are still waiting for some drug or some molecule which can prevent deposition of amyloid beta in the brain and which can prevent phosphorylation of tau, because these are the two factors which are mainly responsible for Alzheimer's disease. Otherwise, if a person has vascular dementia, then we should control it by, and sometimes we can also make it regress by controlling factors like diabetes, hypertension, and so on.
Supriya, it would be all
Yes,
right to
ma'am.
take a few more questions.
So, ma'am, we have a few more questions there.
Okay.
So, how low education level affects dementia?
See, as we know, when we get educated, there are better synapse formation, there are better network formation, there is more dendritic outgrowth. And as we know, these are very important in cognitive functions. So we can correlate that when the dendritic budding is not so high and the synaptic function is not so good, the cognitive functions is not so good also.
Yes, ma'am. So what about the role of NMDA and AMPA receptors in AD?
NMDA receptors are the receptors which are towards the causing very high degeneration of the cells, especially neurons. AMPA also is important in that. Yeah. Okay.
Yes, ma'am. So, on behalf of Gargi College, the entire team of NeuroEunoia 2020 and all the participants, I extend a very hearty vote of thanks to Professor Madhuri for her extremely informative and interesting talk on dementia in spite of all the technical issues. Thank you, ma'am, for sharing with us how an early identification may be done, as well as the biomarkers which can be utilized during the pre-symptomatic phase. Thank you so much, ma'am, for sharing with us your findings.
Thank you very much.
Thank you, ma'am. Thank you so much.
Thank you very much.
And now I would now hand over the mic to Smitha, ma'am. Thank you so much, everyone.
Thank you, Dr. Supriya. Good afternoon, everyone. It is indeed a privilege for me to introduce our next speaker for the day, Professor Gurcharan Kaur. If one takes a closer look at the alchemy of an achieving person, two distinct virtues pop up, beside perseverance and hard work. These are pioneering spirit and daring attitude to venture into the unknown, and Professor Gurcharan bio reflects the same. Professor Gurcharan Kaur is currently working as University Honorary Professor in Department of Biotechnology, Guru Nanak Dev University, Amritsar. Professor Kaur obtained her PhD from Jawaharlal Nehru University and postdoctoral training from All India Institute of Medical Sciences. With her research experience of more than 40 years in the area of neurosciences, she has played a very active role in human resource development in biological sciences as the chairperson of DBT GOI sponsored MSc Biotechnology program at Guru Nanak Dev University, Amritsar. She has major research contribution in the field of biomedical neuroscience, encompassing area of healthy brain, aging, scientific validation of Indian traditional practices of intermittent fasting, dietary restriction, IFDR, natural products from medicinal plant serving as promising interventional approach for various lifestyle and neurological disorder. Another active area of research in her lab is retasking of FDA-approved drugs as a potential glycomimetic candidates for the treatment of spinal cord injury. Professor Kaur has many publications in peer-reviewed international journals, and she has delivered n number of talks in international as well as national conferences. She is the recipient of UGC's Career Award for Young Teachers, Fellowship of Indian Academy of Sciences, and B.K. Jawat Lifetime Achievement Award, INA. Professor Kaur has served as Vice President of Indian Academy of Neurosciences and is also a member of many reputed academics, societies, including Transnational Neuroscience Task Force of ICMR, Member Task Force, DBT Star College Scheme, DST Expert Committee for Women Scientists Scheme, WOSA, IUS, and so on. She has also served as the Vice President and Executive Member of Society of Neurochemistry India, Association of Gerontology India.
I will send a mail.
Now, I would like to invite Professor Gurcharan Kaur to enlighten us with her much awaited talk. Before I hand over the mic to Professor Gurcharan Kaur, participants are requested to type in their questions in the chat box, which will remain open for the next 30 minutes of the talk and will be closed sharp 50 minutes before the talk finishes. Thank you. Professor Kaur, kindly proceed. Thank you.
Thank you, Dr. Samita. Am I audible?
Yes, ma'am, you are.
Okay. So thank you very much, Samita, for kind words of introduction. I don't know really I deserve this or not, but let's see. And at the onset, I also thank the organizers of this conference, Dr. Jaswinder in particular. And I know during these hard times of pandemic, arranging the meetings online is again a very, very difficult task. Because we are not used to this, and these are new experiences for all of us, and I think our brain is getting more and more sharper. So I'll start with my talk, and like to share my slides. Yes. So Samita, you can see the slides, okay?
Yes, ma'am, they're visible.
Okay. So the topic I am going to talk today is on the one of the important medicinal plant, Tinospora cordifolia. So this we all know in Ayurvedic practices, this particular plant is well known for its many medicinal properties, and so I thought because in our lab we have done quite a bit of work in neurotherapeutics on this particular plant. So I thought let me share with you all some of the work. And as Jaswinder apprised me that the audience is going to be a mixed group, so I'll try to keep my presentation very simple and talk in more general rather than concentrating on the hardcore science on this particular plant. Okay, so the main focus of the lab work which we have been doing in my research career, that is on adult brain plasticity, which means that the brain function is not only about the interplay of neurochemicals and neurotransmitters. Brain also undergoes structural changes, new synapse formation, neurogenesis, new neurons and glial cells formation and their migration. So as you have just listened to Dr. Madhuri Behari, who is a very renowned neurologist and has a lot of experience, and she has given an excellent talk on the dementia and what are the causes, what are the how we can diagnose it, and how we can best prevent it. Okay, so adult brain plasticity again suggests that yes, although our brain is dependent on neurotransmitters for nerve conduction, but it also undergoes structural changes, and those structural changes become the basis for lifelong learning and prevention or interventions for various disease conditions. So the major focus of my lab work in adult brain plasticity or healthy brain aging has been to test various interventions like intermittent fasting, dietary restriction. We have also worked with the high-fat diet-induced obesity and how high-fat diet-induced obesity leads to cognitive impairments. And then we have worked with the natural products from two of these important medicinal plants, ashwagandha, Vidhania somnifera, and giloy or Tinospora cordifolia. And another area we are working with is retasking of some FDA-approved small molecules and testing them as mimetics of polysialic acid, which is one of the very important neuronal plasticity marker protein, which helps in synapse formation, exogenesis, and exon pathfinding, and new neuron neurogenesis or neuron formation. So we are testing these mimetics for repair of spinal cord injuries. Okay. So brain, as we all know, brain is one of the most intricate and one of the most complicated organ in the human body, and the brain disorders, which include neurodevelopmental disorders, psychiatric diseases, and neurodegenerative disorders or diseases. And all these brain disorders, they present a enormous disease burden on the healthcare system globally in terms of human suffering as well as the economic cost, because a lot of funding is spent annually for taking care of the patients suffering from brain diseases. And Dr. Madhuri's lecture has apprised you already about those diseases which are afflicting our brain. And you can see these lines below. It's really, really alarming. WHO estimates by 2040, neurodegenerative diseases will surpass cancer as principal cause of death in industrialized nations. So it's a sort of alarm for us that we must be very, very careful with our lifestyle and our mental as well as physical health. So if we talk about lifestyle disorders, the root cause for these lifestyle diseases, be it depression, anxiety, heart diseases, cancers, diabetes, or thyroid or high blood pressure, all these diseases we group under the heading of lifestyle diseases. And the root cause for all these diseases is, you can see, inflammation, stress, poor diet, toxins, then lack of sleep, poor relationships, nutrient deficiencies, lack of physical activity, trauma, poor digestion. So all these factors play a very, very important role in the onset of these diseases. And as we will go further talking about these role of lifestyle in the healthy aging or healthy brain aging, we'll be looking into how these various factors are causing great damage to both our physical as well as our mental health. So how WHO, World Health Organization, defines mental health? It defines that mental health is a state of well-being in which every individual realizes his or her own full potential and cope up with the daily stresses of life, can work productively and fruitfully, and is able to contribute to his or her community. So mental health is not just the absence of mental illness. Mental health is much more than just the absence of mental illness, and is also much more than just feeling happy, is again not in true sense mental health. Okay, so it is very true that mental health and the physical health, they have a bidirectional and a very complex relationship. And often people with serious mental health conditions, they are at a high risk of experiencing chronic physical diseases. So when a question is asked, are you depressed because you have diabetes, or diabetes is causing you depression? So the answer to both the questions is yes. The reason being, people with chronic physical conditions are at a very high risk of developing poor mental health and vice versa. So we all know whenever we are under some stress, our first instinct is, let me eat something, and we always eat unhealthy, overeat and unhealthy. And this is a real problem with our youngsters, especially children, adolescents, and the young people, that they are indulging into unhealthy eating practices, overeating, eating high salt, high sugar, high fat, processed or junk food, and taking less of water, consuming unhealthy diets, and trying those fat diets, keto diets and all. So that is again is one of the very, very important factors, as Dr. Behari has also pointed out, that our diet, our balanced diet, is very, very important for our brain or mental health. And also, we know in our society, be it our government or our local administration, they talk a lot about drug addiction, alcohol addiction. But what about food addiction? And we know that the malnutrition, which is one of the major problem our country is facing for the malnutrition in the children and their growth is affected. But this is another area which we need to take into account, because very soon the malnutrition, which is the eating the junk food, and childhood overweight and obesity and adolescent obesity, it's going to surpass the cases we are having of malnutrition. So this is one very, very important area to keep in mind. So what are the various causes of obesity? One is it could be genetic also, but now we know epigenetics, that is how our environment is interacting with our genes. So even if we have the genes which are leading to obesity, or our metabolism is so directed that it leads to gain of the body weight, but then we can always intervene and modify our lifestyle, and to some extent even the genetic cause of obesity can be prevented. There can also be some hormonal or physiological factors, but this is where the most important aspect we need to keep in mind is most of us are, like especially our youngsters, they are spending a lot of time with the electronic appliances, eating junk food, so they're becoming couch potatoes. So these are psychosocial issues which need attention of our healthcare system, our general public, and our youngsters in particular. Okay. So before I go further into the general discussion about the mental health, I would like to share with you some of our work which we have been doing with this wonder herb, or this is called heavenly elixir or amrita, commonly known as giloy or goduchi, Tinospora, cordifolia. So this plant, which is a creeper, it has lot of medicinal properties. It has good antioxidant properties, anti-diabetic, anti-inflammatory, anti-cancer, antimicrobial, immunomodulatory, and protecting the liver from the onset of diseases. So a lot of these potential medicinal properties this plant has. So what our lab has been working on is to look for its anti-brain cancer activity and its potential to treat the anxiety-like behavior, which could be the outcome of obesity, and this could also be the outcome of sleep deprivation. So we have been testing those specific animal model systems like high-fat diet-induced obesity and sleep-deprived animals, looking for neuroinflammatory, anti-neuroinflammatory activity, anti-anxiety, cognitive impairment corrections, and anti-brain cancers activity. So this is one very important observation we made, was that in high-fat diet-induced obesity, we took 3 groups of animals. One was low-fat diet, that's normal chow feed was given to these animals. Another group was given high-fat diet. So the diet, instead of about 4 to 5% fat, there was 30% fat by content. And then the third group was given high-fat diet, but this high-fat diet was supplemented with the stem powder of this medicinal plant. And what we could observe was, and we performed these experiments, this regime was done for 12 weeks, for 3 months. And right from the 2nd week onwards, we could clearly see the animals which were on high-fat diet, but supplemented with the extract, their body weight was much less than the high-fat diet alone. And of course, it was higher than the normal feed, but it was definitely significantly lower than the high-fat diet group. So this observation shows that the fact is that they were consuming the same amount of calories. So in the face of consuming the same amount of calories, these animals were maintaining their body weight compared to the high-fat diet group, and the adiposity index also, you can see there was down-regulated, and here's the percent change you can see in this particular histogram. And along with that, we also looked at because we saw this very important change in the body weight of these animals. So we immediately thought of looking at the lipid profile and blood glucose levels, because lipids and glucose, excessive glucose in the blood becomes the source for fat deposition, and that leads to adiposity and weight gain. So we looked at blood glucose. There was, although very small increase in the blood glucose in the high fat diet, which was normalized by the extract. But the main observation was that there was a steep rise in the cholesterol in the high fat diet group, which was suppressed by the extract. Same was triglycerides. And you all know the cholesterol and triglyceride levels in the lipid profile, which is basically we call dyslipidemia, where we have high cholesterol and high triglycerides. So both these components of our lipid profile, they were corrected, and high HDL, which is a good fat, was reduced, although in the high fat, but it was again increased in the group which was given the this stem powder supplement. So this is a very complicated slide. I will not go into the details. I just want to share with the audience that when you in the scientific manner of working with a particular problem, you always have to go step by step. Okay, so like in this case, when we fed the animals with the high fat diet, what we observed was there was maintenance of the body weight, because we were taking body weight and the feed intake every week. So we could see that yes, there is management of the body weight by this plant. So then we looked at the lipid profile. We got good indices for the lipid profile. Dyslipidemia was corrected. So next step was, let us see underlying mechanism where exactly this plant extract may be acting. So we worked on this very important pathway, which is responsible for the energy homeostasis and fat metabolism. So AMPK alpha is the main regulator of this pathway. So basically what we looked at was whether the lipid synthesis or lipid degradation is being targeted, because when we gain weight, either there is reduction in the utilization or degradation of the lipids or beta oxidation, or there is increase in the synthesis. So we looked at these pathways and we got the Western blot data, which shows that yes, the hepatic lipid metabolism is affected, and the correction by the in the high fat diet and correction by the extract is to regulate or down regulate those enzymes which are involved in synthesis, and upregulation of those enzymes which are involved in the degradation. So both the synthesis as well as the degradation, they become the targets of this plant extract. And one very important observation we made was that this is HMG-CoA reductase is the enzyme, you know, these lipid cholesterol lowering medicine, we use the statins. So this is one of the targets for the statins. So we could see there was an increase in the HMG-CoA reductase. That means more of cholesterol synthesis in the liver, but it was suppressed by the extract. Okay. So another lifestyle problem we have is not sleeping at the right time. So, you know, our body has a biological clock. So that biological clock goes with the day and night cycle. We call circadian rhythms. And our body metabolism, our body temperature, our pulse rate, our heart rate, they're all affected by the biological clock. And our brain is the master regulator, or the clock in the brain. So when we are not sleeping as per the body clock, or we sleep late and get up late, that also plays a very, very significant role in the onset of these lifestyle diseases. So sleep loss or insomnia or sleep fragmentation are the other problems which are associated, of course, with the medications. There are people suffering from psychiatric conditions, depression, anxiety, and use of alcohol or drugs or smoking, and then professional commitments and the lifestyle. They are the common causes for having inappropriate or sleep deprivation. And we all must have, you all must have experienced one or the other time that if you're not able to sleep properly just one night, next day next morning, you can very well see what condition we are in as far as our attention, our daily work is concerned. So we feel lost even after losing one night's sleep. But when it is a regular feature for those professionals working late hours, or our children spending more time with the screen, with these electronic appliances, so they are losing sleep every day, and it leads to sleep debt. Sleep debt ultimately leads to the cognitive impairments. So this was another area which we studied with this plant, and we did a lot of fractionation and tried to find out the active ingredients also from this plant, which are responsible for the neuroprotective activities. So we identified one of the ingredient, Tinospora, and with the this particular Tinospora, we named it C1. We could see that this was very high, this is having a very high potential for regeneration of the neurons. Again, Dr. Behari has apprised you about the neurodegeneration and how this neurodegeneration leads to major neurodegenerative diseases. So again, this plant, we could clearly see supplementation of the alcoholic, 50% alcoholic extract of this plant, feeding them orally. It suppressed the anxiety-like behavior, which we see in the sleep-deprived animals, which we have shown by these parameters like grooming behavior, average bouts of the number of those grooming behavior bouts, number of rearings. So all these behavioral patterns, they showed that the supplementation or oral feeding of the extract to these sleep deprived animals, it helped in suppressing the anxiety, and also it helped in maintaining the motor coordination, as a rotarod test is when the animal is made to move on a rotating rod, and those animals which have good grip strength, they can hold on to the rod and they keep moving with the rotating rod. But the anxious animals or even the obese animals, they are not able to keep sticking or moving on the rod, and they start falling. So number of falls and the time spent on the rotating rod gives us an estimate of the motor coordination behavior of these animals. And further, this again, I would like to bring to the notice of the youngsters who aspire to become a scientist in their further academic pursuits. So when you design a particular study, you need to look at a particular, say, treatment or aspect you are studying or regime you are using from multiple angles. So since we saw in the previous slide, I've shown that these animals, the anxiety-like behavior was suppressed by the plant extract. So we looked at these glial cells and the microglial cells expression in the brain, because these glia and microglia are the indicators of inflammation. And also there's interleukin 1 beta, TNF alpha, and IL-6. These are all inflammatory cytokines, which are the indicators or biomarkers to the neuroinflammation in the brain. So we could clearly see upregulation of these proteins in the brain of the sleep-deprived animals, whereas in the Tinospora supplemented animals, there was downregulation, and also there was downregulation of these peripheral inflammatory proteins. Now, this is what I was referring to, the circadian rhythms or the biological clock. So you can see from this slide, the circadian rhythm disruptions. Circadian rhythms are regulating your day-night changes in the body temperature, your respiratory rate, your hormonal production, the menstrual cycles in the females, urinary excretion, cell division. These are all under the regulation of the master clock or the biological clock, and these changes are shown with the circadian rhythms with the day and night. So when there is a circadian rhythm disruption, when we are not following the circadian rhythms or the biological clock of our body, ultimately it leads to many of health problems like mental health, stress, anxiety, depression. It leads to cardiovascular disorders, in the long run. Also, reproductive health is affected. Then brain health as the gastrointestinal disorders, and there is increased prevalence of cancers in those individuals who are regularly not taking good sleep and not eating. So that's what we say, that lifestyle is very, very important to prevent the onset or even to slow down those diseases. So here the role of circadian rhythms, I will explain to you with the help of one disorder. This is called shift work disorder. So in those people who have night shifts, so it's a clinical observation that night shift workers, they have many of these health issues in the long run. The reason being, when you have shift work disorder, there is a conflict basically between the body's circadian rhythm and the work schedule. So basically what happens that when the person is at work, the body wants to sleep because nighttime body wants to sleep. That's a biological clock regulation. But when we go to sleep during the day, that's the time when the body expects us to be awake. So there's basically a conflict between our biological clock, circadian rhythms, and our body functioning. So that conflict in the long run leads to many of these mental and physical health problems. So here I again will share with you another important study which we have made, the anti-cancer activity, anti-brain cancer activity using the plant. So we used the cancer cell lines, glioblastoma and neuroblastoma cell lines, and we treated those cell lines with the plant extract, and we could see that these and the extract has anti-proliferative activity, or it reduces the growth. And we all know the growth, the cancer is all about cell division, a very high uncontrolled cell division. So the plant extract could induce apoptosis, which is programmed cell death, and also it arrested the cell cycle. So basically anti-proliferative activity. We use flow cytometry technique to study these two parameters of anti-apoptosis and cell cycle arrest activity of this plant extract. And also we using immunostaining using immunofluorescence method, we could detect a very important upregulation of the neuron specific proteins, also along with the change in the morphology. So you can clearly see here, these are the controlled cancer cells, and they have lost their normal differentiated morphology. But when we treated them with the 200 microgram per ml or 300 microgram per ml of this plant extract, the rate of growth was reduced, as you can see the number of cells, and also the cells showed very nice differentiation. So we used other markers like NF200, MAP2, NeuN. These are all neuronal cell markers. So using these specific cell markers, we could clearly see very nice differentiation, neurodifferentiation. And I must tell all my youngster students listening to my talk here is that for brain cancers, differentiation based therapy is very, very important, because our brain is all about synapse or neuron connections. So if we are able to bring those brain cancer cells back into the differentiated morphology, they can again enter into the formation of new synapses, and the lost function or damage to the brain can be recovered. So differentiation based therapy for brain cancers is very, very important. So this plant extract we also tested using this migration assay in the wound scratch injury assay. We showed that this migration of the cells in the scratched area was inhibited, and also the cells were showing the very nice differentiation. Whatever small number of cells which were migrating, they were differentiated, unlike control where they were present in very high numbers and they were undifferentiated. Okay. So this is just the overall view of what different pathways, cell death, apoptosis, and then the plasticity and stress chaperones or stress response proteins expression, reactive oxygen species, production, mitochondrial dysfunction, which are all the hallmarks of the neurodegeneration, and all these were protected by the use of or by treatment of these cells with the plant extract. So here we challenge the cells, the neurons, primary glial cells, the primary neurons, cerebellar and hippocampus neurons, with the glutamate, very high concentration. And all neurodegenerative disorders, glutamate is one of the important neurotransmitters which is controlling or regulating brain function. But we say excess of everything is bad. So excess of glutamate, overstimulation of glutamate receptors, NMDA receptors or AMPA receptors, it leads to neurodegeneration or cell death. And the underlying causes are shown in this figure. And we could clearly see the butanol fraction of the Tinospora cordifolia extract. This particular fraction was very effective in controlling the neurodegeneration. So they showed very good regenerative effect. So you may ask, what is neurodegeneration? Okay, so neurodegeneration is basically a progressive loss of structure and function of the neurons, which ultimately leads to neuronal death. So this impairs the nerve function, loss of control abilities like memory and decision making. So degeneration of the neurons, which is a hallmark of the neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, Huntington's disease, and it can also be the outcome of stroke or other autoimmune diseases like ALS or the MS. So all these diseases affecting our brain, our brain disorders, they are having the neurodegeneration as the central point, which is the outcome of aberrant intracellular signaling. There is neuroinflammation can lead to neurodegeneration, protein aggregation. We have those diseases like Alzheimer and Parkinson's, where there is aggregation of the top proteins in nuclein or beta amyloid proteins, and then oxidative stress, releasing the reactive oxygen species, which also plays important role in mitochondrial dysfunction. All these factors ultimately lead to degeneration of the neurons, and that leads to loss of function and leads to the disease states. So again, this is one very important observation we made with this plant was we grew those explant cultures, primary explant cultures from the hippocampus region. And hippocampus is one brain region which is responsible for learning and memory. And also it's a very rich site where the neurogenesis, new neuron formation is continuously occurring. That is how we are able to make new memories, and the basis for this is new synapse formation, and that's what we call brain plasticity, that brain keeps on forming new structures, new synapses. So in this hippocampus region, we took small parts of this hippocampus, put them in the culture dishes, and treated one set with the glutamate, which led to degeneration of the neurons. But when we treated them with the butanol along with glutamate, we could clearly see the regeneration of neurite outgrowth and the migration of cells from these explants. We have immunostained these cells with PSA NCAM and NCAM. These are the two very important plasticity marker proteins in the brain, and associated with the differentiation, neurodifferentiation. So you can clearly see the positive role or the beneficial role this plant extract plays in the regeneration of the neurons. Same is shown here, that was explant, and this is primary cell culture. So again, you can see glutamate treated cells underwent degeneration, whereas they were eschewed when they were along with glutamate, they were pre-treated with the plant extract. So these are some of the observations which we made using this plant extract and testing it for various types of lifestyle conditions like sleep deprivation, high-fat diet-induced obesity. Then we used it for anti-brain cancer activity and glutamate-induced neurodegeneration. So all these parameters, apoptosis, which are markers for BCL-XL and BAD, then senescence markers, the HSP70 heat shock proteins, then inflammatory cytokines, and then the cell survival AKT pathway. All these pathways signaling pathways. Our students have studied in the lab and published the work in very high impact journals, and till date 20 students have done their PhD from the lab, and 4 more students are currently enrolled who are continuing their work on different aspects, working with the FDA approved mimetics, PSA mimetics, and also with these medicinal plants. So again, I'll come back to the general discussion about the lifestyle. What is unhealthy lifestyle? So we sometimes think it's only like if I don't do any physical activity, or I'm not taking good food like healthy diet, I'm taking poor diet or junk food, which is high salt, high sugar, fat diet, and sleep deprivation. So that is not all. All these factors, your poor diet, your sleep, your screen time, more of and this screen time, the glare of the screen also has impact, and when especially at night when we are using watching TV or working with our computers or these smartphones, that impacts your sleep patterns and impacts your sleep. So the quality of sleep is compromised. Then lack of physical activity, then the toxic negative thoughts, always be positive. That is one of the mantras. Then insufficient exposure to sunlight. We all know the importance of sunlight for the synthesis of vitamin D3, which is very, very important, not only for our bone health, but also for our mental health. So then similarly, alcoholism or smoking, and then 2 important factors like poor relationships. We all know how stressed we feel when we have a fight with a near or dear one, or we are isolated. So that impacts our mental health a lot. And then mental health has direct impact on our physical health. And then lack of communication, social interactions. This is very, very important factor, that we have good communications and we keep good relationships and interact socially and serve our community, contribute to our community. So not just taking care of your diet and physical activity and sleep, these factors play a very, very important role in your healthy life. Okay, so here in this context, I will share with you one study which was done by Dr. Julian from Brigham Young University of USA. So what she did, she took thousands of subjects, people, middle-aged people, and she did a longitudinal survey, and she studied these factors: clean air, hypertension, high blood pressure, body weight, physical activity, cardiac rehab, or flu, or boozing, smoking, close relationship, and social integration. So from these bars, I'm going to share with you what these bars actually indicate. So what she did, she took these parameters, studied, recorded these parameters in the form of a questionnaire from about 10,000 people, and then waited for 7 years. So this is middle-aged group, and she wanted to see which of these factors play the most important role in living longer. So 7 years after that first survey, she again surveyed with the same factors, and she analyzed her results, and this is the outcome, where she has shown that social integration and close relationships, these are the 2 key factors in living longer. And not just living longer, we are not concerned more about lifespan. What is important is health span. So whatever life we have, we should live healthy. And if you are living healthy, you have health span, your lifespan, longer life is a bonus. It's automatically going to be there. So social integration means how social you are, how, like when just going down the street, if you happen to see a sweeper or a vendor, so you say hello to them, or you see somebody, some lady going with their dog, do you stop and ask for her well-being? So it's not just your close relationship, your friends, your known people. Unknown people, do you interact with those unknown people? That really gives you happiness, so that we call feel-good factor. So close relationships are those people on whom you can depend, mid of the night, if you require some help, they are there. They always stand by you. So a small set of people we call close relationships, we can depend on them, and then social integration. It played very, very important role. So although your body weight as well as physical activity, that is your diet and physical activity also is important, but these factors collectively, all these factors are very, very important for leading a healthy life. Okay, but the thing is, for adopting a healthy lifestyle, the change starts in the brain. That means first thing which is required is your determination. So you should be determined that I'm going to have a healthy lifestyle. So the change starts in the brain, not in the gym. Well, yes, our brain is plastic. So nature has provided us with this gift that we can adapt, we can even change, or we can intervene in the onset of diseases by adopting healthy lifestyle. And as Dr. Behari has also pointed out about the brain, it's common saying, more you use, less you lose. So it's very, very important to keep these factors in mind. And dear friends, it is not a rocket science. You only have to be very, very careful what is right and what is wrong. We all know this is not right. And if we are like this, we are going to raise the levels of cortisols and all other stress hormones and neurotransmitters in our body, and that's going to play havoc with both our mental as well as physical health. So be like this. Again, do this. Don't be a couch potato. Don't just sit down with a bowl of chips or French fries or burgers and beverages, and just sit down with this remote in hand. Go out. Try to be with nature. This is the time. See, with the advancement in technology, with the advancement in medical science, with the inventions of antibiotics and the vaccines, we have taken control over our infectious diseases. We call communicable diseases, okay, contagious diseases. But our healthcare system is now having again to task to deal with the lifestyle diseases we call non-communicable diseases, like diabetes, like cardiovascular diseases, hypertension, anxiety, depression, cancers. These are lifestyle disorders, and this is very much in our hands. By adopting healthy lifestyle, we can take care. So eat this, eat healthy, not the unhealthy food. Okay, so in this direction, our lab has done quite extensive work with the dietary restriction regimes. So we asked a question like, do we really need those life extension movements? Like that's a hype in the media and in our social life, that we come across people suggesting anti-aging diets or anti-aging cosmetics, lifestyle diets or anti-aging medications. These are all fake, I must say. So what we need is to adopt healthy lifestyle, eating healthy controlled diet, improve your lifestyle habits, do little exercise, be very social, communicable. Okay, so all these things play a very, very important role. And we studied the role of intermittent fasting, we call periodic fasting. And if you go through the literature about this fasting concept or intermittent fasting, or these are called time restricted fasting also. So this fasting is not like Monday fasting, Tuesday fasting or Friday fasting. This fasting, which has scientific validation by many groups in India as well as abroad, they've shown that if we restrict our diet, our meals, during the daytime and evening, we follow the circadian rhythms. We eat early dinner and eat light in the evening, and then about 12 to 14 hours we don't eat anything. In any case, 7 to 8 hours we are going to sleep. So you, instead of eating late night, start eating early, say 7 o'clock. Eat something light which is digested within 2 hours, and then take a break of about 12 to 14 hours. So that is recommended, and that gives time to your body, to your cells to process the food which we have eaten during the day and also eat healthy. These are the mantras for healthy eating. And the concept of energy balance, always we should keep in mind that we should know how much I am eating, how much energy I am taking, and what is my daily expenditure of energy. If we don't keep that balance, that excess of energy we take, excess of food we eat, it is going to get accumulated as fat in our body, and fat around your body organs, internal body organs like your digestive tract or your reproductive or your heart, it is going to play a very negative role, because this brown fat especially, which is white adipose tissue, which has a lot of macrophages and is a source for inflammatory cytokines. So those inflammatory cytokines are very, very damaging to the functioning of these various organs, especially our brain. So caloric restriction or versus dietary restriction, generally we keep struggling. What should I how do I restrict my diet by working on my size of the meal or frequency of the meal? So to all these issues, problems, or the questions, the one answer is that go for intermittent fasting, as I just told you a minute before, that intermittent fasting, by skipping the dinner, heavy dinner, eating early, and following the circadian rhythm, eat early, sleep early, and get up early. That is one very, very important factor for maintaining good physical and mental health. So this intermittent fasting, it's like challenging your body intermittently, and that helps to improve the health. Here I'll also apprise you about one term and phenomena. This is called hormesis. Hormesis is when we give a mild stress to our body, it prepares our body to meet a major stress. So this intermittent fasting is again a type of mild stress. Same is true with the physical activity. Exercise leads to strenuous exercise or even mild exercise leads to generation of free radicals, and that free radicals are a sort of stress, but that is very low dose. So anything in low dose is good for health in the long run, which prepares our body to have to meet the major stress. Okay, so this hormetins, we call those factors which have hormetic effects, like fasting is one of them, spices, Indian spices in moderate quantities, another hormetin. Then we have little high temperatures sometimes, like that is what our saunas are very famous, that you have steam bath. So that again is type of hormetin. And then the physical activity or exercise. So all these are hormetins which induce mild stress in the body, and that mild stress ultimately prepares, because it has its basis in upregulation of those stress response proteins, which are the molecular chaperones, and they help in refolding or correction of the damaged proteins in the body, and it helps the physical as well as the mental health. Okay. Now the issue is that whether we talk about our ministries or faculties or the departments of health, as well as most of our biomedical research, that is dominated and it supports the disease-directed thinking. Most of research funding also goes in finding the treatment for the diseases or looking for biomarkers or diagnostics of the diseases. That's important, no doubt. But side by side, it is very important, like when we have taken control over the contagious or infectious diseases by use of vaccines and antibiotics, now these non-communicable diseases or lifestyle diseases, they are very much in our hands. If we lead healthy life, if we have or if we adopt healthy lifestyle, so we must also give importance to the self-management interventions so that we are able to sustain good health and optimize the functional capabilities of our body. Okay, so again, I'll say yes, your brain is plastic, but for the brain to remain healthy or plastic, it is important that we spend or we have healthy lifestyle. So healthy lifestyle, again, we have discussed, like your healthy diet, physical activity, your sunlight, your meditation, yoga, your close relationships, your social integration. So all these factors are the mantras for healthy lifestyle. So with the help of this cartoon, let's move off the illness path. Let's take out this thing that we will be having suffering from one or the other disease. Let's not move or think about that. And also by simply adopting the healthy lifestyle, we can prepare ourselves for self-management, self-help, for mental health. So this is very, very important for our youngsters. Again, keeping in view the fact that currently India's majority of the population is young. So the future of India is right now in the that youth group, and for the future, it is very important that our youngsters, our children, as well as the young adults, they keep all these factors always in mind, that if I keep a good lifestyle, if I develop good lifestyle habits, in the long run, I'm going to be benefited. So why to create pool of diseases and then look for the treatments and diagnostics? Why not prevent? Why not intervene right now and prevent the onset or slow down the onset of these diseases? Okay. So again, I go back to what WHO says or defines the health. So WHO defines health as a state of complete physical, mental, and social well-being, and not merely absence of disease or infirmity. And we know very well there's no health without mental health. So it's very important. I always like to talk about these 6 best doctors. And what are those best doctors for us? One is our healthy diet. And by adopting skipping the meals, especially the dinner, it's commonly said that eat your breakfast like a king, your lunch like a queen, and your dinner like a beggar. Why like a beggar dinner? Because beggars are never sure whether they'll get the next meal or not. So always have very light dinner, early dinner, and follow your biological clock. Go for physical activity, light physical activity at least every day. Go for sunlight. Respect your circadian rhythm, sleep early and get up early. Practice yoga and meditation, and then the support of your family and friends. Okay. Now, out of these 6 best doctors, I would say 4 doctors are India's heritage. How? Fasting is India's concept, heritage. Then the Ayurveda, which we have been talking, is India's heritage. Then meditation and yoga has origin from India. So again, India's heritage. And you know, this family and friends culture, any time of the day or night, you just knock at the door of your family or friends, you're always welcome. And you know, this type of culture, this type of welcome to the guest is nowhere in the world except India. So if we have so many doctors, best doctors, our heritage, what really prevents us from leading a healthy life? There should be no reason to say that still I am not able to follow. So let's start with taking the baby steps. Do simple things to take steps towards your health span. And again, if we talk about the current times, COVID-19 times, lot of stress, everyone, especially with small children at home, parents are under so much of stress, because earlier they used to get this respite of going out for some, like the students, children will go and parents can spend their leisure time. But now with children at home, it's too much of stress. And then what is going to be the future of this COVID-19? It's everybody's guess. It's every day we keep hearing in the news or newspapers that those figures are very alarming, and we don't know. We don't have any medicine in hand. We don't have any vaccine in hand as of now, although a lot of work is going on. So stress is there, but if we take it as a stress, and if we start just thinking about and getting upset with this time, or with the scare of the corona, then it's not going to help. Rather, it will spoil our physical and mental health. So what we can do during these times is to upgrade ourselves. We can make ourselves more physically and mentally strong, more fit, emotionally more balanced, and energetically more efficient. So this is very important, like how you take the stress. That's what I was talking. Mild stress, I don't say this is a mild stress, but after all, it is there, and nothing is in our hands at the moment. What is in our hands is to take care of ourselves. So we must take this time as a positive aspect. Also, this one positive aspect is like we are everybody sitting at home and attending those conferences. So a lot of these webinars are going around these times. So as far as the availability of good quality Ayurvedic products availability is concerned, I have one major concern. Like these, we have a lot of scientific validation, as I've shown you my work with the Tinospora cordifolia. But when we go to the market to get a good quality product in terms of its good components and right proportions, we are never able to get a good quality product. Why? Because there are many factors which count towards raising a quality raw material for these formulations. What time of the season the plant is harvested, what was the age of the plant when it was harvested, and there's a need to develop elite varieties with high content of the active ingredients. So, but our herbal industry and our say administration, they are not giving much attention towards these aspects. So people from herbal industries are getting the raw materials which is not tested. It is also tested for the heavy metals and any contaminations, but it is not tested for the concentrations of these active ingredients in these plants. And also there are no directions to the farmers that they should they should raise the plant for 2 years or 1 year, or what should be the season when the plant is harvested. So all these efforts are required to get a disease-free quality product, and by the Ministry of Ayush and the Government of India, they have taken the initiative by setting up herbal health research consortiums. So we have one such facility at Amritsar also, because Amritsar is the hub for raw materials and herbal industry. That's the biggest market of India, in Amritsar, for the raw materials as well as the herbal products. But the scientific validation is still a question mark. So what we need is a collaboration. We need to join hands between the research labs, between the industry. They should get quality product, and then we should get the quality raw material tested by the consortium facilities, and then the practitioners should also be priced that they use only the quality product for prescription to the patients. So it's a lot of potential in Ayurveda. It's time tested, thousands of years of testing, traditional knowledge is there, but still we are not able to get a good quality product. The reason being lack of cooperation between these sectors, which we need. So with this, I close my talk and thank the organizers. These are all my very lively students who have participated in all the studies which are being carried out in the lab. Some of them are already completed and gone for greener pastures to India, other places and abroad. So this is Golden Temple at Amritsar. You are all welcome. And this is my beautiful campus, Guru Nanak Dev University at Amritsar. This is the entrance of our university. Very happy to share that our university was ranked ranked 2 in clean campuses across India last year. So this is springtime in our university. So thank you very much for patient hearing. And I now hand over to Dr. Jaswinder.
Thank you, ma'am. That was a very informative session. I would now request our moderator, Dr. Smita ma'am, to take up the questions.
Thank you, Jaswinder. Thank you, Professor Kaur, for stirring the mind of all of us so deeply with your wonderful lecture in every sense. Going back to our root seems the mantra for the well-being of all of us.
Yeah.
Now, we would now take up the questions. Ma'am, the first question is there a significant benefit of vegetarian diet for cognitive performance?
See, vegetarian diet certainly, because when we take a vegetarian diet, we have a balance of, say, vitamins and minerals, and of course, a lot of fiber, which is good for digestion and clearing of the digestive tract. And especially, I would say, we take non-veg diet, non-vegetarian food, although it's a good source of protein being one of the important macronutrients of our diet, but it takes very long for the body, for the cells, for the digestive system to digest the proteins. Whereas the vegetarian diets are very easily digested. So that way, there's no bar on taking the non-veg diet, but it should be restricted. And also, especially in the evening, the dinner, one should avoid taking heavy, high protein diets. So a vegetarian diet is always good because it is good for your digestive system, easy to digest and nutritious also.
Sure, ma'am. The second question is the herb Tinospora cordifolia a nutraceutical?
Yes, you can say it is nutraceuticals because some of the components, like especially the antioxidant property of this plant and also immunomodulatory property of this plant, berberine is one of the important constituents. There are many alkaloids and polyphenols and other such natural compounds which are present in the Tinospora cordifolia. So if we are able to identify individual active components, and they can be even supplemented in some of the food products and can be recommended, of course, as formulations, herbal formulations available with say Patanjali or with Dabur or Himalayas or others, they are using it as a nutraceutical as well. Because see, anything which is good as an interventional product or intervene with the disease process is good for health, or it can be recommended as a nutraceutical. And here also I would like to add that let us make our food as medicine, not medicine as a food. Okay, so when we have good food, balanced diet with these natural products supplemented, so that automatically acts as a medicine. So we don't have to depend on medicine. Basically, we can check the onset of the diseases. So our food becomes our medicine.
Next question is, ma'am, does the plant affect in adipose tissue anyways?
Yes, yeah, that's what I showed in one of my slides, that it reduced the adiposity index. That means adiposity index is calculated by calculating the total amount of this white fat around the body organs, internal body organs. So when we looked at the total amount of fat which was present in the rat in the high fat diet fed and the one which was fed high fat but supplemented with the extract, so there was reduction in the total fat content which was accumulated in the abdomen. So adiposity index certainly was reduced. So that's why we further went on to study the lipid metabolism to look at where exactly it acts on the degradation or in the synthesis of the lipids from the liver.
Right, ma'am. The next question is, does the plant extract have any toxicity?
No, it doesn't, because because of lack of time, I didn't show my results. But when we were doing these experiments, first thing we tested was on liver function and kidney function. So we did all those liver parameters like SGOT, SGPT, these enzymes, which are the markers to tell us about the toxicity. And then we did kidney function, urea, creatinine, and all these oxidative stress markers. So all these markers study showed that yes, this plant, the dose we used was not toxic. That was very important first to find out that the plant doesn't have the extract or the quantity or dose we are using is not toxic to the body. So we did all liver function, LFT, RFT functions before that. And it was not toxic.
One of the participants is very eager to know about giloy leaves. So the question is, please let me know whether study was also done with giloy leaves or only the stem was used, and the concentration which was used for the immunofluorescence studies.
See, we have used only the stem because from a traditional knowledge, use of this plant for Ayurvedic formulations, the stem of this plant. And again, I must share here that we use the stem of this plant. The thickness of the stem should be your finger thick. That thickness about 1 centimeter thick and brown in color, not the fresh stem. And then when this creeper is grown with the neem tree, so since it has aerial roots, so it also acquires some property of the neem. So we all know neem is very important medicinal plant. So stem, finger thick stem and brown in color. So that has the maximal medicinal properties. There are reports with the leaf of this plant is well, but we have used only the stem because from the traditional knowledge we could gather that yes, stem is more useful for medicinal properties.
We'll take the last question for this session, and this is, these experiments were performed in vitro in cell culture. So if the plant extract, crude or purified form, is supposed to be administered to the live animal, how could we confirm or maximize its availability to the nervous tissue, as the nervous tissue is physically separated from the other bodily system?
Yes, that is very good question and a very elaborate one also. First, let me answer the first part. The experiments were performed in vitro in culture conditions. So when we administer it to the animal, like we have studied the effect of this stem extract or supplementing the stem powder in the diet. So we have studied both the peripheral organs like liver and kidney, body fat, and also we have looked into the brain functions like cognitive impairments and motor coordination and brain plasticity. So we could clearly see the effect of the plant given in the diet. That means some of the active ingredients are crossing through the blood-brain barrier. You know, the blood-brain barrier is the one which is separating which is basically there to control the entry of any molecule which may affect the functioning of the brain. So this blood-brain barrier, which is constituted by the very tight junctions of the capillaries of brain, capillaries of the blood supplying to the brain. So that capillary nature, the wall of the capillary is such that it does not allow any molecule. It is you can say not permeable to all types of molecules or cells to cross and reach the brain. So basically it's a defense mechanism for the brain, but yes, because of this blood-brain barrier, the entry is not open to all the molecules, but it does allow small lipophilic molecules which are say about less than 400 Daltons in size. And for our studies, we have not yet looked into what really from this extract, because there are maybe around 50-60 different components. So which component or which of these components are actually crossing and affecting the brain function, we still need to look into that. But if we just go by what we have done and we have seen the effect on the brain, that means some of the important constituents, they are actually passing through the blood-brain barrier, reaching the brain. Another important aspect here is that they are controlling the peripheral inflammation, and peripheral inflammation ultimately affects the brain by causing neuroinflammation. So that is another route by peripheral, means how the brain function can be controlled, or brain function can be like impairment can be prevented. So if it is like in high fat diet obesity animal, if it is controlling the inflammation, peripheral inflammation, so automatically neuroinflammation is also being suppressed. So there are various explanations for, but to really look into which component, we have to do some analytical methods to find out which of these components have crossed through the blood-brain barrier and they have reached the brain. Or we need to check individually on the brain cells under in vitro conditions.
Thank you, ma'am, for taking up so many questions and answering so patiently.
Thank you. Thank you very much.
Thank you for such an interesting and informative lecture that gave us all a food for thought. Health is achieved as an equilibrium of physical and mental well-being, and that Ayurveda indeed holds the key to all answers in the neurotherapeutic approaches. That was such a wonderful piece of very useful information for all of us, ma'am. It was great to know the use and efficacy of tea cordifolia for pharmacological treatment of neurological diseases and for treatment of fever, diabetes, anxiety, immunodeficiency, memory deficit, and psychological problems. We all had faith in Ayurveda, but your talk just reaffirmed our belief in it and its use in treatment of something as complex as a neural disorder. Thank you for reminding us that nature has given us all that we need, if just so much as look for it. It is such a pleasure and privilege to have you here among us. We are indeed obliged that instead of the busy schedule, you accepted our invitation and gave us the opportunity to listen to such an informative and knowledgeable lecture. On behalf of Gargi College fraternity and the entire team of NeuroEunoia, I extend my gratitude to you. Thank you, ma'am, for your gracious presence. Thank you.
Thank you. Thank you so much, Samita, and the whole team. They are doing a wonderful job, and really I admire your patience, and in spite of those hiccups, no, you guys are doing so well. So all the very best, Jaswinder, and
Thank you, ma'am.
very happy to see you all online.
Thank you so much, ma'am. You're such a great speaker.
Yeah, I'll keep in touch. Yeah.
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 Okay. Bye.
Okay, ma'am. Bye.
Bye.
Udita, would you like to say a few words? Udita? Udita?
I now request Udita Mukherjee to take over. Thank you.
Hello, everyone. What a wonderful day of talks it has been. And I must extend my sincere apologies to all the participants for the technical snag. Also, I must express my sincere gratefulness to all the speakers for their perseverance and fighting through the virtual difficulties, for sharing your profound knowledge. Apart from the insights that were given to us for neuroscience, it would not be an exaggeration to say that we have learned more from your perseverance. Words fall short to express how much it means to us. Starting with the all-rounded keynote address that told us everything about the brain and gave us the glimpse to the future. Then Brahma, the Indian brain template, was truly enchanting. We were made to realize the true complexities of our brain by knowing how it remains aware, rather on guard, even when we are sleeping. It was rather intriguing to know that the cognitive processes remain up and running, involving different brain networks during sleep and waking state. As we proceeded with the day, the talk on finding biomarkers associated with Alzheimer's disease, and then the use of Ayurveda for potentially curing some of the mental disorders have been so wonderful. This day has been nothing short of enlightening for us. Our heartfelt gratitude to each one of the speakers for their time and patience. I thank everyone for being with us today. We greatly appreciate your enthusiasm and hope to see you all tomorrow on the final day of NeuroEunoia 2020. Thank you. Have a great evening.
Thank you. Thank you so much.
Can we all just switch on our cameras for some time to take a snapshot? All the participants, please.
This Corona has taught us so many things, huh? So many new adventures.
Yeah. The picture is getting better and better. Thank you, everyone.
Thank you so
Have a
much.
great day.
Thank you. Thank you so much.
Mr. Anuj, can you please close the meeting?
Okay.